Dear honored guests and participants, it is my privilege to welcome you all to the 4th International Genetics and Bioengineering Congress held by the Yeditepe University Biotechnology Society. We are very fortunate to be among the most esteemed scientists who have accomplished groundbreaking works in their respective fields and will share their expertise with us today. We would like to thank the rector of our school, Professor Dr. Canan Aykut Bingöl, our Vice Rector, Professor Dr. Fatma Yeşim Ekinci, the Dean of the Engineering Faculty, Professor Dr. Ahmet Arif Ergin, and the members of the academic staff of Yeditepe Genetics and Bioengineering Department for their support in our Congress. In this Congress, we hope to involve the latest achievements and developments in the Yeditepe bioengineering field and discuss thought-provoking new ideas and to broaden our horizon. We also believe that the young and aspiring engineers of the future will be able to put a face to accomplished researchers in their fields while being encouraged to ask their own questions and benefit from our Congress. I invite Hüseyin Çimen, who is our advisor for the Biotechnology Society and a member of the academic staff of the Yeditepe Genetics and Bioengineering Department in Yeditepe University, to deliver his opening speech.
Thank you, Sarah Elif, for the nice introduction. So I welcome you all to our 4th International Genetics and Bioengineering Student Congress Days. In the hope that this is our last time organizing this meeting online, so we can meet with our guests and with our attendants on our campus, Yeditepe University. And also I'd like to thank to every and each member of our organization for their contribution, for their great effort to put everything together for this nice, the scientific day. And also I would like to extend our appreciation to our speakers who accepted our invitations and they joined us our meeting today for the Congress to share their, the experience, their, the extensive knowledge in their own fields. And I hope that this is going to be one of the most fruitful day for every registered, the member to our meeting. And let me say a few things in Turkish as well. Herkese hoş geldin diyorum. Bu güzel cumartesi günü öğleden sonrasında birbirinden değerli konuşmacılarla birlikte günümüzü geçireceğiz. Bu güzel organizasyonun bir araya getirilmesinde çok fazla kişinin emeği var. Ve bu kadar 500'e yakın bir katılımcımız var. Umarım herkes için çok güzel, çok verimli bir gün olur. Şimdiden katılımınız için herkese çok çok teşekkürler. Sözü tekrardan Sara'ya sana bırakıyorum.
Thank you for your kind and encouraging speech. I now invite the president of the Biotechnology Society, Aslan Demirkıran, to deliver his speech.
First of all, I am really honored to have a chance to say a few words among really respected scientists that we are hosting today. And I just want to use this chance to thank all the participants who are listening to us right now and our honored guests who graciously accepted to come. And but I especially want to thank two friends in the Biotechnology Society who worked tirelessly to put up this amazing event. And I wish you all a great Congress.
Thank you for your kind speech. We're now waiting for Assistant Professor Erdinç Sezgin. We are now having some difficulties since our guest speaker is having problems joining the Zoom platform. We hope we we thank you for your patience. I invite the first speaker, Assistant Professor Erdin Sezgin, to deliver his speech titled Seeing the Microcosmos of Life with Advanced Imaging. He is currently a researcher at the Karolinska Institute with the main research area of understanding the physiochemical principles underlying cellular processes using advanced imaging techniques. He will inform us today of the state-of-the-art imaging techniques whilst commenting on certain limitations of these technologies and how they can be overcome. Welcome and thank you for accepting invitations. If any of our participants have any questions during the duration of the speech, you may write it in the YouTube live stream chat or through Instagram DM.
Merhabalar.
Hi, sir. I
Hello.
assume they've checked your speech and
Yes,
if you're
we're all
able to
good.
present your screen. Okay.
Yes, we're all good. Can I just go ahead?
Yes, you may. Thank you.
Thank you very much. Thanks everyone for inviting me. It's a great pleasure to be here. I will talk about imaging technologies and what they are good for. I will start a bit on the story of light, but first I want to tell you a bit why it is so important to understand the microscopy technologies. So I think you can appreciate now these days in life sciences, not only in life sciences, material sciences, chemistry, it is really important to directly visualize what you have in your tube, in your sample. That is why it's very important that we develop advanced technologies so that we can understand what is our sample. So it is important also to understand that these imaging technologies can work to visualize not only the microcosmos, but also the macrocosmos. If you understand imaging technologies, such as microscopes that I'm going to talk about, you will also understand how we are using these technologies to understand the universe with telescopes. It is basically more or less the same principle. So you can understand, with using the same technology, you can understand a cell, how the cell is organized, or how a planet is organized. So that's very important to understand the basics of optics and imaging in this sense. So it is really also fascinating for me to see the story of light, historical story of light. How it starts with basically Euclid that you could be familiar with from your high school geometry lectures. Euclid explains the light, how we see things, by the help of light, but Euclid thinks that we are sending light from our eyes so that we can see things. And he says also it's because we send light, and if there's some objects in between, we have shadows. Euclid explains this kind of phenomena, but now it feels a bit absurd to feel that we are throwing something from our eyes. Although it sounds absurd, this idea was valid for hundreds of years till Alhazen. Alhazen actually fixed most of the Euclid's shortcomings in the explanation. You explained that we don't see by eyes throwing something from our eyes, but light sources outside, such as sun, brings the light and everything is reflected, and we can see this reflected light. So if you didn't read much about Alhazen, I really recommend to read a lot about Alhazen, because Alhazen is considered as according to some philosophers, science philosophers, he's considered the first scientist, because he establishes not only great scientific concepts, but also scientific paradigm, like how you should be critical to yourself, how you should be critical to what people tell you as a knowledge. So that's why I think it is really worth reading about Alhazen. Well, then after this, we know now, because of how Alhazen's experiments, like camera obscura, for instance, light goes as straight rays and then reflected from the objects, and we see this. But then at some point, some physicists, they were trying to understand what is the nature of light. Is it just a ray? It goes as a ray. And then Newton, Huygens, and Thomas Young, they were doing some experiments. They figured out that light is composed of waves. It may not, there are some phenomena that light is undergoing that cannot be explained by just the nature of a straight ray. So I'm telling this duality, if the light is a ray or a wave, is a particle or a wave, because it's very important for microscopes. For some aspects of light is really explained by the waves. Some aspects of the microscopes can be explained by particles. So that is really important to understand this historical progression of the light explanation. So, this prominent scientist trying to explain light as wave, but then there was a mathematical framework by Faraday and Maxwell. They figured out electromagnetism and light and electrons, they're somehow intertwined, and they called it electromagnetic theory. And they said light is only an electromagnetic wave, nothing else. And by then, people thought everything was solved. If we solved light, everything was solved. In a way that Lord Kelvin, some of you might remember Lord Kelvin by the temperature unit that we're using in science. He said, all the physics is now known. Don't send your kids to the physics department anymore, because there's nothing else to figure out. Physics, only thing that we should figure out now is like very small things that is not even worth it to have physics departments anymore. Well, this might be familiar to you, because you might also hear from some people that there is nothing big being to be discovered anymore in genetics, bioengineering, biotechnology. But fortunately, history shows us opposite, that Max Planck and Einstein fortunately didn't listen to this advice. They opened completely new window. They were saying, yeah, great, light is a wave, but it still carries energy. When you stay in the sun, it still heats you up. So there must be some particles. So Max Planck calls them quanta. Then currently we call photons, but then this quanta started actually this how quantum physics took off in the beginning of 900s, right? So, and then Einstein explained this with photoelectron theories, which brought him the Nobel Prize. This is also very interesting, because Einstein is known for relativity, but relativity didn't bring him Nobel Prize. The theory that brought him the Nobel Prize, but photoelectron theory, which explained how light, electrons, and matter is interacting. And then it creates this duality. Light is a wave, it propagates as a wave, but on the other hand, it carries particles that have energy. That is why I'm going to refer to this point, because it's really important for the microscopy to see what is limited by wave nature of light and what is limited by the particle nature of light. So this was just a brief story of light and how we came to understanding of light and how we use light, because in microscopes, this is one thing that we cannot do without. It is the light. But what do we see? Why do we need microscopes? So first of all, with the eyes, our own eyes, we can see millimeters. We can see very small scale, but we cannot distinguish if they're, let's say, two things very close to each other. By eyes, we can do if they're millimeters apart from each other. But then if there's small things like micrometer range, we have the microscopes. But then even the microscopes are limited because of the wave nature of light. We call it resolution limit, that I'm going to go into detail a bit more later. But even the microscopes, conventional microscopes that you can see in normal labs, they are actually limited by the light limit, the wave limit. So this is limited by half of the wavelength that they're using, that I'm going to go into detail later. But then we have a big realm below this limit. What are we going to do to understand this part? So this part is very important to understand. And that's why we constantly work in the lab to develop better and better microscopes to break, not to break a physical law, but to circumvent a physical law. So this wave nature of light imposes us this limit, which is called the resolution limit or diffraction limit, but we circumvent this to get better resolution, to be able to distinguish entities like smaller than this 100 nanometers, for instance, like viruses. So a bit on the evolution of microscopes. So the microscope starts with Leeuwenhoek, which is just a tailor trying to understand the fabric, the texture of the fabric that he's using for his clothes. But then the first scientific microscope is probably can be classified. First scientific microscope is Robert Hooke's microscope, who was in Oxford and trying to experiment on small setups like light source. Again, in a microscope, you always have this light source, which is, in his case, is a candle, as you can see. And there you then have your sample. You focus the light onto your sample by a system of lenses. Here, it's only one single lens here. Here you have like two lenses, and then you have to look at the sample. This is very important because then he was seeing this kind of structures drawing by hand, and contemporary microscopes look like this, but the principle is exactly the same. You have the light source, you focus the light source with the help of lenses to your specimen, and then you have to detect the signal by camera, your eyes, or some kind of other detectors. So this box is full of lenses that is doing exactly the job that this microscope does, but of course with better and better quality that we cannot even compare anymore. So, but then if we have this amazing microscope, can we just put a dead mice on a microscope and see all the molecules and then get amazed by it? Unfortunately, this is not real life. As you can imagine, you cannot just put a dead mice and see single molecules. That is not possible. So here, it is really important. If you don't remember anything from this talk after today, this part that you can remember, it is called pyramid of frustration. Pyramid of frustration tells us that we cannot achieve everything at the same time in a single microscopy type. So let's say you want to keep your sample healthy. Let's say you want to look at live cells and dynamics in the live cells, but you also want the big temporal resolution, which means you want to take pictures every millisecond, and you want good spatial resolution, which means you want to see a few nanometer details, not micrometers. And then you want to do in a whole animal, so your sample size is centimeters, and then you want really good signal. All of them you cannot achieve at the same time. So you have to compromise from one or two if you want to gain in another one or two. So if you want really good sample health, you have to give up on resolution and signal to noise ratio. So this is a big problem for microscopists, and microscopists are at the moment working very hard to get microscopy technologies that would get multiple of these properties in the same microscope so that you don't have to use multiple different types. But currently, some microscopy types are good at certain aspects of this pyramid. That is why I'm going to talk about today different microscopy types, what they are good at, and if in your current research or in your future research you need microscopes, you are at least informed which kind of microscopes are available and what kind of microscopes you could use to answer your biological question. So first I want to go into this axis, like what these axes mean. Sample size is a very important question. If you are about to investigate the whole animal, right? So whole zebrafish, whole Drosophila, this is a very big sample size. We are not dealing with a single cell anymore. We are dealing with a single organism, which means a very, very big field of view, right? So you can do this, but you need certain microscopes and you have to give up from some aspects such as resolution. But then you can go a bit more into detail. For instance, you can see not the whole organism, but a developing organism, which is less complicated. But you want, for instance, you want to see a time lapse. You want to see the whole development of an embryo. And then you need then some other certain microscopy technologies. But you can also do it, right? I'm also showing this to you so that you can see what you can do. There's basically almost no limit what we can do, just you cannot do everything at the same time. So you want to focus on a bit, let's say from the scale, you want to go into the cellular level, then you can do cellular imaging with a lot higher resolution. Now you can see single cells and try to identify different cell types in a tissue, for instance. This requires a bit more resolution. If you want to go into subcellular imaging, of course, you need better resolution technologies so that you can resolve multiple structures at the same time, so such as internal architecture of the cell. As you know, there are a lot of organelles inside the cell. It is very, very crowded environment, so that is why we need good resolution, good contrast, so that we can resolve them. But then when we go there, unfortunately, we hit a big barrier. Let's say you want to see organelles like lysosome. I don't know if you're familiar with organelles, but lysosome is one of the small ones. It is around 100 nanometers. But unfortunately, we cannot see entities that are smaller than half of the wavelength that we are using for imaging. Let's say you're using, usually we use visible light for imaging, and wavelength of visible light is around 500 nanometers. If you're using 500 nanometers light to visualize objects, you cannot distinguish two objects as two separate objects if the distance between them is less than half of your wavelength, which is 250 nanometers. So if they are apart from each other, more than 250 nanometers, you can distinguish as two different objects. But at some point, they get so close that you cannot really distinguish anymore. And this is because of the wave nature of the light. If you put an infinitesimally small particle under a microscope, let's say 0.001 nanometer, which doesn't exist as such a particle, but imagine you put it there, what you still see is half of the wavelength as a size. So if you're using 500 nanometer, your image is still 250 nanometer, although your object is very, very small. So this is called diffraction limit. This is because light propagates as a wave. So for this, what are we going to do? Are we going to just give up? So we accept that we cannot see this part of the realm? No, we actually don't. What we do, we develop super resolution microscopes. As I said previously, we are not breaking a barrier because we cannot. This diffraction limit, this resolution limit is like gravity. You cannot just break it, but you can circumvent it, right? You can create some gravity-free environment. It doesn't mean that gravity doesn't exist anymore. But this is exactly the same thing. This conventional microscope, this is exactly the same sample in a conventional microscope and in a super resolution microscopy technique. So what is the big advantage here? For instance, if you look at here, this part of the image, you don't see what is happening. You cannot really distinguish anymore what is there. But if you check here, you see like 1, 2, 3, 4, 5, 6, 7. You can count the molecules. This is now single molecule imaging, right? So you cannot do it with conventional microscopes, but you can do with a super resolution microscope. You can distinguish very much in details the architecture of your sample. So I hope you would understand here that this, in summary, conventional microscopes, any microscope is limited by the resolution that we can circumvent by super resolution technologies. But what do we need? Which microscopy technologies we need? And what are their applications? I think this is quite important for you to know that so that you have kind of a guide what you want to use in the future. So first of all, the most widely used microscopy technique is called confocal microscopy. So I mean, probably I would say most widely used technique is fluorescence, normal fluorescence microscopy. But now we are talking a bit more advanced microscopy. So the most widely used advanced microscope is confocal. It probably any institute would have it in the microscopy facility or somewhere in the lab. And this is very good for 3D sectioning. So here you see, for instance, an embryo that I imaged, for instance. This is cells labeled, and then you can see the yolk, you can see the cells. Or this is our synthetic cells. I'm not going in detail into biology. I just want to show what is available by microscope. So, but this is our synthetic cells showing two-phase coexistence, green and magenta, as you can see. This is a 3D image. You can do 3D sectioning with confocal microscope. And then you can also do dynamic imaging. Here, for instance, it is half an hour movie that, again, I show this phase coexistence, that you can watch what happens to these phases, that they can divide, they can merge. So you can do both 3D and dynamic imaging, but main thing about confocal imaging is you have to put a lot of light in order to gain good signal, which means if you're doing live cells, you cannot image very long. And this comes from the particle nature of the light, because each particle carries energy. And when you image something, you always put energy in your sample. So this means that you cannot do very long time imaging. Your cells will probably die, we call it phototoxicity. So the photons are toxic because you put a lot of photons in it, and it becomes toxic, and the cells go apoptotic, for instance. So although it is very good, very useful for certain applications, we cannot do everything, long time imaging, for instance. And for this, we use new microscopy modality, which was developed in the last decade, actually. It is called Lightsheet or lattice Lightsheet, depending on the sample size. So lattice Lightsheet is an excellent technology because you use very low dose of light. Of course, you compromise from the resolution a bit, spatial resolution, but you gain a lot from the temporal resolution. For instance, you can see how cells move. This is like 3 hours movie. You can see how the cells move, how intracellular structures like mitochondria, for instance, move around. And then you can actually do a lot of research on the dynamics of the cells. So if you want the small cell scales, you use lattice Lightsheet, or if you have big scales like organoids and see how different cells, this is a tumoroid model, for instance, and immune cells invade this tumor tissue and then go around and then move. And this is a bigger scale for lattice Lightsheet, but this is a perfect scale for normal light sheet imaging. But again, you can do almost a day, maybe more than a day imaging without actually causing any phototoxicity, any kind of damage to your cells. And another advantage of Lightsheet or lattice Lightsheet, fast imaging. So this is real-time 3D imaging. This is the magenta circles are synthetic cells, and green dots are virus particles. And we want to see how many virus particles bind to certain synthetic cell. And then what you do, you can do very fast imaging so that you can have more than, I don't know, 100,000 synthetic cells. So you can count 100,000 synthetic cells. It becomes very high throughput technology. It is almost like a fax technology, fluorescence sorting technology, but now here you can also image, you can also see them. But again, this is fantastic technology for dynamic imaging, 3D imaging, but the resolution is still really bad, spatial resolution. As I said, it is limited by the half of the wavelength of the light. If you need subcellular structure imaging, such as mitochondria, and you want to see the structure of mitochondria, you cannot do with normal microscopes, either with confocal or with lattice Lightsheet or Lightsheet. You need super resolution microscopy, and this is, for instance, our STED imaging. STED is one of the super resolution microscopy technologies we use in the lab quite often. It's called stimulated emission depletion microscopy. And here you can see in normal microscopes, you can see the details, but then you can see the subcellular substructures of mitochondria that you see in this textbook, the crystal structures of the mitochondria you see in the textbooks very clearly. And of course, you can see, you can increase resolution in 2D or in 3D as well. So this is 3D, for instance, if you look at the cell, it's a profile picture of a cell. You can see with normal microscope, you can't really resolve structures, but in STED, for instance, STED imaging, super resolution imaging, you can get this endocytic vesicles. These are the vesicles that capture something and go into the cell, so that it's basically material transfer for the cells. So you can capture this details very much in real time with very much in details. So this is where the super resolution imaging is good for. So with this, I'd like to finish here without going much into details. And take-home message from this talk is there are multiple microscopy techniques. There is not a single microscopy advanced technology. These are the ones that I mentioned today. These are the ones that are more mainstream ones. There are also other technologies that we don't use very often, but they're also good at certain aspects of imaging. And they are all good for certain applications. We have plenty of room for new technologies. And that is why I think I encourage you, since the audience here, mostly students, I really encourage you to go more into interdisciplinary science in your future, because as you can appreciate, we need physics knowledge, optics mostly, to be able to push these technologies. We need biology knowledge so that we know what is needed. And we need chemistry knowledge because you need all these fluorescence probes, biosensors, so that you make stuff visible, otherwise not visible. Computer science, of course, since we have a lot of data now, we have to analyze this data. Everyone in my lab can do programming because it is important. So you get the imaging data. You cannot do much if you don't know, especially if it's new type of imaging modality. You don't know what to do. You have to have analysis methodology. So you have to write scripts to be able to analyze. And that's why I also encourage you to go into fields of biophysics, especially biophysics, because biophysics field is really enriched with this kind of research where you need chemistry, physics, and biology at the same time. So with this, I'd like to acknowledge my lab, all my collaborators all around the world that we collaborate on very, very different biological topics. I'm not going to go one by one, but more importantly, I'd like to thank you for inviting me and thank you for listening. I'm happy to take some questions if you have some.
Thank you so much for your speech, Professor Erdinç. We have some questions that we're going to ask you. One of our participants asked that are advanced microscopes today made according to the law of classical physics or quantum physics?
Very good. I mean, for most parts, we are still using classical physics. But of course, as you go deeper and deeper, as you go more into resolution, that scale doesn't work anymore, right? I mean, we need our bottleneck is at the moment is coming. Because for now, the bottleneck was resolution. So we solve resolution. Now we are coming to data analysis. We are solving it, but the big data comes, right? With the big data, we cannot even, for some microscopy technologies, having the data real time, you produce so much data, normal computing might not work anymore. So we might need quantum computing. But I think it will come in next decade. Maybe we'll use quantum computing, for instance, to acquire images even.
Other question, how do you get so many colors in microscopes?
I mean, getting colors is not really a big deal because what you need is different fluorescent molecules. different fluorescent proteins, different fluorescent dyes that has different excitation and absorption spectrum. So you can get infinite number of colors. The point is to separate them, right? How you can separate yellow from yellowish green from green. So then we have a lot of tools like spectral unmixing tools that we can go up to 60 colors, for instance. But main thing is getting those molecules are easy, but separating them might not be so easy. We need more algorithms to get this.
Thank you. And when imaging lipids, do you stain using organic dyes? How does the lipid preserve its natural behavior?
Ooh, that is a hot one. Yeah, no, that is one of our very big challenges. Labeling proteins, just to give a background for people who cannot picture lipids, labeling proteins with a fluorescent tag is easy, relatively, because you just put a fluorescent protein next to your protein of interest, so your protein becomes green or red. But lipids are so small compared to proteins. If you put the fluorescent tag, it is as big as the lipid. So that's why we really screen hundreds of different strategies to label lipids and try to see if the lipids preserve their nature. And of course, we have some guideline after working on this for years. We know that the dye should be very small, smaller than the lipid, and the dye should not be hydrophobic, because if it's hydrophobic, it interacts with the lipid itself, so it's not very good. And then we should have a space between the lipid and the dye. We usually put a spacer so that dye is a bit outside the lipid. But it is definitely still ongoing, so we need better and better fluorescent lipid probes.
Developing the microscope is an interdisciplinary field, but in which fields are there many people?
I believe this question is which fields are there many people? So first of all, I will reply as much as I understood, since I cannot ask what they really meant. So first of all, yes, it is an interdisciplinary endeavor. So you need physicists, optics to be able to set up the microscopy technique, right? But it's not enough. Sometimes, usually even people from electrics and electronics are going into this direction, because half of optics is basically electronics. So you can also make use of your electronics expertise in optics. There are more biologists in the field, of course, trying to answer questions, because there are more biological questions than more ideas of microscopes, right? So I think it's probably a bit more advantageous to push a certain microscopy techniques. But then the main problem there, you have to make a very good technique, so a lot of people use, right? So we have a lot of microscopy techniques that are very specific to certain labs. They have it, and that's it. No one else has it. Maybe another lab can build it. But the main idea is to build a microscopy technique that would be applicable in many labs. So I think that is why the field is populated by every single field. So it is very important that you find your own niche, but as long as you have interest on interdisciplinary science, I think that is the main key. Begum, I can't hear you. I can't hear you. You're muted.
I'm sorry. I'm so sorry.
Nice.
Are we able to manage to pass the fraction limit that you just mentioned? Light by using, instead of using light nature, then the particle nature of the light. I'm not mentioning the electron microscopes.
Yeah. No, I get it. I mean, we cannot really pick a certain nature of light overall, right? Light is a light. So we have light, we have to work with it. And it's a wave. Whenever it comes as a wave. What we do to circumvent it, we are not actually picking a property of light, but what we do, so there's one simple rule. Since light, the resolution limit is half of the wavelength, you can just go very low wavelength light, right? So you can go to UV, but then you kill your cell in a second, right? UV is high energy. As even you can go a bit more, right? So people use this X-ray imaging, right? X-ray imaging is really low wavelength, and then your resolution is super high. but then, you know, X-ray imaging will not be very good for any live tissues. The point is then, how are we going to do it? So we have smart technologies, smart ideas to do this. That's why they got the Nobel Prize in 2014, because they're very smart. So first of all, it was also, they got the Nobel Prize in chemistry, super-resolution microscopes. That also highlights how interdisciplinary the science is. Imagine then why super-resolution microscopy gets chemistry Nobel Prize, because chemistry is very important. The dyes are very important. The probes are very important to be able to see things, because most of the smart technologies are because of the dyes. We can actually switch on and off the dyes, so that even if two things are very close to each other, we switch off one, we know where this one is, and then switch off this one and this one is, and then we can fit and then find the center. Once they are isolated, it's very easy. Once they are at the same time emitting, that is not easy. That's why we switch on and off. And this switching on and off mechanism is a good mechanism to get super resolution imaging, for instance.
Thank you so much. And during the experiment in the lab, you mentioned that there will be some other issues with the labeling the lipids. How do you manage to do the optimization process for those dyeing processes?
Yeah, it takes a lot of time, to be honest. I mean, now in the lab, most of the optimization is done by myself, because this might not be the fun job for everyone, right? You get a new dye and then you try to understand how it behaves. So we get a new dye, either we make it or we buy it, or we collaborate with chemists, so they synthesize new dyes for us, new lipid dyes. So we take it to the lab. First thing is to see how they behave in the assays that we know how lipids would behave. So if it's a saturated lipids, we know how saturated lipids behave in a certain setting, and we see if they behave as a saturated lipid. And then next, we have specific lipid assays. Like if it's let's say, is it a ganglioside? If it's a ganglioside, we have specific binding assays. Gangliosides, some toxins bind gangliosides, right? Do these toxins still bind this ganglioside? And then we have diffusion assays. We know how molecules diffuse more or less in the membrane, and we check the diffusion, if this diffuses properly in the membrane, if it's like a single lipid or it forms clusters, and so on and so on. And we usually eliminate a lot of them. I mean, we have only a handful of lipid dyes that we trust out of, I don't know, maybe 1,000 I personally tried since my PhD. We trash a lot of them and just use only a few that works with them.
Thank you so much. And our last question is not related with your topic, but it comes to you. They said that you did your PhD in the Max Planck Research School. And how was your process when you're doing your PhD? How did you manage to, choose that particular related field for the young scientists?
Good. Begum, can you inform me, most of the participants are from Yeditepe or not?
Generally, yes.
Okay. So I graduated from Yeditepe University, genetics and bioengineering. I guess this is relevant information. And I really appreciated the support from our faculty. They were very supportive, very friendly, and they really thought about our future, our career. I'm sure it's still like this because some of them are still there, and the new ones, I'm sure they're also very supportive. But after third year, when I was in third year, they already started encouraging us to apply, because that was very good timing. If you're already at the fourth year, you would be a bit lost, right? It's fourth year, you're ending, and also most of the deadlines are already beginning of the fourth year, right? So you don't have to finish so that you apply. You have to apply before you finish. So I really liked the research of biophysics when I was at Yeditepe. So I was in the lab of Mustafa Chulha, who was doing nanotechnology, which was a interdisciplinary area. It was between chemistry, certain physics, not so much physics, but biology, definitely chemistry and biology. I was doing Raman spectroscopy of living tissues. And that already gave me a purpose a direction, because I realized that I really like this analytical technologies, right? So I want instruments, but I want answers of biological questions using these instruments. And particularly when there was a challenge on the instrument, I really like to troubleshoot this challenge, right? So if there's something new I can explore in the microscope, Raman spectroscopy, it was really very good for me to go into it, to read about it. So I realized that this is what I like, and I applied to labs that are doing mostly Raman, but that was actually not a good choice. I think Cosmos directed me to a right path by not giving me a lab of Raman, because that would be very limited for me. It was instead I went to a interview in the Max Planck Research School in Dresden, and there my PI, PhD supervisor, she's working on fluorescence correlation spectroscopy. It's a different type of spectroscopy, very widely used to understand mobility of molecules in cells, in solution. And then I realized that this could be an interesting endeavor for me, because it's a completely new technology. So I'm not doing exactly the same technology that I was doing before. But then biological questions, the nature of biological questions will change. It will open a new window for me. So that's why I decided to go to this field. And it was the field of biophysics, because there was more physics in the lab, but also a lot of membrane biology. And I really like membrane biology. Also, for me, membrane was also one of the things that I really liked when I was having like cell biology lectures, like DNA, for instance. It was never attractive to me. Somehow, I enjoyed this, membrane trafficking a lot more than DNA replication, histones. And I still, if you ask me all these questions, I might not remember much about the DNA, but when it comes to organelles, plasma membrane, mitochondria, I like it a lot more. So that's how I decided to go into this field. And I think overall, I feel like it was the right choice for me, but probably whatever I chose then, I would have felt the same way, that it was the right choice for me. So just keep your interest broad enough, within the range that you know what you like and what you don't like.
And also, sorry for my information that it will be a last question, because some of our participants still ask questions.
Yeah, but I still have time, and also we still have time, no, for the next speaker.
Yes.
Yeah, okay, please shoot.
You mentioned about the programming. Is it related with the R language or other languages? Could you please provide some details?
Yeah, we in the lab use mostly Python. I mean, we means my people in the lab. They're using mostly Python, because it is a very good language for image analysis. But of course, if it's big data analysis, like omics analysis, for instance, I know a lot of people use R. And they're both free, they're both open source, right? Depending on what you want. Do you want big data analysis? If you want to go to omics, for instance, to this kind of fields, I think R would be good. If you want to go more into writing a program that would work like with a graphical interface, and then probably Python would be a good option. But I think I would definitely suggest this at the moment. But of course, there are new languages coming up. Probably there'll be in 10 years, there'll be another one that is more popular. As long as it's open, that is very important these days, because like MATLAB was very big thing, but MATLAB is going down because it's not open. People have to pay. And now there's an alternative, which is free, like Python. Of course, everyone is changing from MATLAB to Python. Of course, R will not die, because R is a unique thing still, and free, open source.
Also, you mentioned in your speech that pyramid of frustration. There are parameters that affect certain imaging, and I want to ask, are there any softwares that to adjust according to your sample, according to your experiment?
Yeah. I mean, all the software is basically specific to certain imaging types. So we have a big software called ImageJ.
Mm-hmm.
ImageJ, Begum, you better get used to ImageJ, because you're going to use it a lot. ImageJ is it's like the most widely used software for imaging people. It is also free, of course. It is developed by the community, so you can develop some plugins, install it, and then it becomes available for everyone in the community. Of course, ImageJ though does things that are more or less the mainstream in the imaging. So if a lot of people need it, you can find a way to do it in ImageJ. But if your imaging technology or your outcome is very specific, then you have to write your own script. That is why most of the technologies that in the pyramid of frustration, you have certain technologies are good at something, you also have software doing this, right? But software cannot take one axis and get close to the other axis. So software can also do analysis of the imaging. They cannot change the optical limits, right? I mean, to a certain extent, but not fully.
Thank you so much for your speech. It was phenomenal.
Thanks so much. Thanks for inviting me. I really had fun.
Two of our friends will be in the breakout room for the evaluation part, the Congress. Again, thank you so much for your coming.
Thanks, Begum. Thank you, everyone.
Our session for the first speaker has now ended. We are now waiting for our second speaker. For all our participants joining, we have a Google Form doc for attendance in the YouTube chat, which is mandatory for you to fill out if you would like to obtain a certificate of participation. I would also like to reiterate this in Turkish. YouTube chat kısmında yoklamamız var Google Doc üzerinden. Katılım sertifikası almak için doldurmanız zorunludur. The Google form will be removed at 13:45. We have a short break for about 10 minutes before our second speaker joins. Our next speaker is Professor Seamus J. Martin, PhD, who is a researcher at Trinity College. Today, he will present us some of his in-depth research on molecular control of apoptosis and how this process is regulated from complex multicellular organisms such as humans to more simple organisms such as earthworms in a speech titled Regulation of Programmed Cell Death from Worms to Mammals. Welcome and thank you for accepting our invitations. If any of our participants have any questions for the duration of the speech, you may write it in the YouTube chat live stream or through Instagram DM. Thank you for joining. I assume they checked all your microphone and screen presentations?
Yes.
Okay, you may start your presentation now. Thank you.
Okay, thank you. Okay, can everybody see this? Yes?
Yes.
You see my slides? Okay. And you can hear me okay. So, all right, I will start. My name is Seamus Martin. I work at Trinity College in Dublin, and I'm going to tell you about programmed cell death, which, as you'll hear, is a really cool type of cell death that takes place all of the time in your body. So I work in Trinity College in Dublin, and this is the Trinity College campus here in the center of Dublin. Can you see my pointer on screen? Could somebody let me know, can you see the pointer moving on screen or not? No?
Yes, we can see.
You can, okay. So this is Trinity College Dublin. It's Ireland's oldest university, over 400 years old. And we're right here on the east coast of Ireland, this Dublin Bay out here behind the campus, and with the River Liffey running through the center of Dublin. So that's another view of Trinity. And this is obviously not Trinity College, but I just want to mention that I like fishing very much, and I was out fishing yesterday. It was a beautiful day in Ireland yesterday, and I hear it was a very nice day in Istanbul as well too. So I think spring is finally here, and that was the sunset. I put this up because people always think that Ireland is a place that it always rains. It does rain here quite a bit, but not quite as much as people think. So I'm going to talk about cell death, and this is the way we used to think of cell death. For many, many years, cells were going on, going about their daily business, and then suddenly something really terrible happened. And then the cell is dead. And while that is true in certain cases, that type of cell death is called necrosis, where cells really don't make a choice to die, but things just happen to them. And that type of cell death happens occasionally, and it's the kind of cell death that we see when you get a burn injury, for example, or you're involved in a very serious accident where there's a very sudden loss of cellular life. And that type of cell death is called necrosis. But in fact, most of the cell death that takes place in our bodies is very regulated and very planned, very coordinated. And as I'm going to tell you, there's an actual set of molecules, a cell death machinery, as we call it, that cells make in order to specifically kill the cell when the cell needs to die. So I'll start off with just making some comments about cells. Obviously, the cell is the basic unit of life. We tend to treat cells as quite simple things. They're not simple things at all. They're highly complex. Cells have thousands of different proteins. Obviously, our genome has about 25,000 different proteins, but with post-translation modifications, we make about maybe about 150,000 different proteins at a minimum. And so cells have tens of thousands of copies of each of those proteins, all working together in different macromolecular complexes. And we really have no idea really exactly how cells work in any kind of detail yet. We're decades away from really understanding that. So although cells are the basic units of life, they're highly complex. And we have 10 million cells in our bodies. 10 million, that's a lot of cells, 10 trillion cells. And in fact, during our lifetime, we make probably 1,000 times that number of cells because cells die in various tissues at different rates. And so we're constantly replacing cells in different tissues. Cells are beautiful, and as I've said, they're highly complex. I'm going to talk about just one of the many different types of cell death process or cellular processes that cells have at their disposal. And even that is pretty complicated, with about 100 proteins that have been identified that work in this process. And we still don't understand exactly all of the different pathways, how to switch on this machinery and how it's switched off in all the various circumstances. But we know a great deal more than we did 20 years ago, obviously. But as I've said earlier, we're still 20 to 30 years away, at least, from really understanding in any kind of level of detail how it all fits together, how cells really work. So if you consider the body as essentially a complex cellular society, where the outside of the body, the skin, and then the gastrointestinal tract running through it, and we have the lungs here, and then all of the other constituent tissues. There are certain rules that cells must obey in order to be allowed to live in this cellular society. And the rules are: do your job, stay where you live, so cells don't get permission to move around the body. Don't eat your neighbor. Obviously, in a multicellular society, it's essential that cells can trust one another, and so they don't eat one another. Whereas essentially we can eat almost anything else. We eat most of the other animals, obviously, on the planet, and most of the plants and so on. But in a cellular society, cells don't eat one another. Now, the reason I mention that is during programmed cell death, that rule breaks down. And in fact, permission is given within the body for cells to eat one another, only under those special circumstances. Another rule is don't divide without permission. So you're not allowed to make another cell unless you have permission to do that, and permission is given in terms of growth factors. Other rules are don't allow bugs inside. Clearly, our immune systems are constantly combating infection. In fact, our skin, lungs, and gastrointestinal tract are major barriers to infection. And our skin is very good at keeping out infectious agents. And if infectious agents get into our bodies, we spend a great deal of time expelling them. And then another rule is don't act weird, don't act strange. Now these are, in many ways like the rules in society, except in cellular society, if you break these rules, the punishment is death, essentially. As we'll see, the major punishment for breaking any of these rules in a cellular society is that you are killed by programmed cell death, or you turn on your programmed cell death machinery. Because our bodies obviously have a certain shape, a certain form. We all look pretty much alike, and that's not by accident. All of the cells in all of our tissues all do their jobs. They don't move to other tissues. So a cell in the kidney doesn't just decide to go on a journey to the brain, for example. And we intuitively know this. But this is maintained, this is enforced by cells not behaving the way they should are killed by programmed cell death. And we'll get to see. So that's really what I've said. Cells that are injured or infected or abnormal, dysfunctional in some way, breaking the rules. In the cellular society, death is the penalty. And the type of cell death we see in all of these situations is programmed cell death. And this is what it looks like. These are normal cells here. The two cells that I'm on the left-hand side here that I'm pointing at. This is a normal nucleus, and we've just stained the mitochondria here in green. But then these two cells on the right-hand side that are stained with blue and red, the blue are chromatin fragments. So the nucleus is broken up into these very small, strange pieces. And this is highly characteristic of apoptosis, which is the major form of programmed cell death in the body. And then the membrane of the cell is staining with a dye called an exon 5 that binds to a particular marker that only appears in cells in the programmed cell death. But these cells look really quite abnormal. Obviously, they're highly blebbed. They haven't burst open, they haven't ruptured, and that's for important reasons, which I'll get to. But these cells are undergoing programmed cell death. So programmed cell death enables us to eliminate cells with surgical precision in all of the situations where it's desirable to eliminate cells under conditions where cells are breaking the rules of the cellular society. Whoops. So really what I've said here is we're constantly making cells by cell division, because in various tissues in our body, there's a lot of cell death taking place because of injury, because of infection, because of aging, because of some other type of dysfunction. And cell death is the counterbalance to all of this new cell division. And in fact, obviously, if we didn't have cell death, so if cells didn't die in our bodies due to these causes, due to injury and infection and aging, we wouldn't need cell division taking place in the adult. So there is a constant throughput of cells in our tissues where new cells are born, and then they become aged or old or dysfunctional, and then they die by programmed cell death. And as I mentioned earlier, in our lifetime, we turn over about 1,000 times. We're constantly replacing tissues or cells within our various tissues. And as a result, cell death, programmed cell death, can be looked upon as the body's natural healer. It's a way of naturally eliminating these aged or dysfunctional cells. So I've said this a couple of different ways now. These are the circumstances we're going to be focusing on today. And we're going to look at how cells die under these different conditions. And as I've said, it's really important that cell death is regulated, because of course we understand that cell division is regulated, but it's just as important to regulate cell death so that there is this fine balance between the rates of cell division to balance the rate at which cells are dying throughout life. And if we didn't have that balance, then clearly there'd be either too much cell division, and that does happen in cases such as cancer, for example, or there'd be too much cell death. And that happens in, for example, neurodegenerative disease. So the body needs to strike this fine balance between cell division and cell death throughout our lives. And when that balance goes into disequilibrium, as I said, we get disease of various types. So here's an example. These are normal lymph nodes, these little white dots in a petri dish here, normal lymph nodes from a mouse. And this is the normal size spleen from a mouse. And these are the lymph nodes from a mouse that has a single gene mutated in the machinery that regulates programmed cell death. Now that gene is called FAS. I'll return to it later on to talk about death receptors and how they switch on the programmed cell death machinery, something that we've worked on for a number of years. But if you mutate the FAS gene in a mouse, within 4 months of birth, the lymph nodes of the mice are about 50 times the normal size. That's because there's an accumulation of extra T and B cells that normally go through this cycle of cell division and then cell death, but they don't die in this case. FAS is needed for the elimination of mature T cells. And now they keep getting born, but they don't die. And as a result, you have this huge accumulation of T cells and B cells. The splenic enlargement is also quite dramatic, because now you have all these extra T and B cells, and these mice then go on to develop an autoimmune disease as a result of that. So that's just in one tissue. So programmed cell death is used to eliminate cells at all stages of life. During development, you have lots of processes where cells are born only transiently. They're only needed for a short period of time. For example, in the development of the limb, you make extra cells as you grow the limb, and then you eliminate the cells you don't need in a process like sculpture, where you form the final limb by eliminating the interdigital cells that have been made transiently during development. We do the same thing in our developing nervous system. We overproduce neurons, and then the ones that make connections are the ones that are allowed to survive. And at least 50% of neurons die during development. We do the same thing when we're making our adaptive immune systems. We overproduce T cells and B cells. And in fact, about 95% of developing T cells die because they don't make the right receptors or they're autoreactive. So there's a huge process of cell death going on, or a huge amount of cell death going on during development in order to shape the final form of different cellular systems. And then in the adult, we have a lot of cell death going on in tissues most prone to injury and damage. And obviously the skin, for example, and our gastrointestinal tract are constantly being damaged by the environment, by things we eat and drink, and also by infectious agents. And so these tissues are constantly undergoing renewal. Cells of your skin, for example, they're completely replaced every 30 days or so. So one message that anti-aging creams don't work, right? The same happens in our liver and other tissues, such as our gastrointestinal tract, where there is a lot of damage constantly going on. And in response to that damage or infection, we see a lot of programmed cell death take place. So programmed cell death helps to keep the body healthy. It's involved in sculpting organs and tissues. It culls the weak, the sick, the infected, the strange, and maintains the vitality of our tissues. And there's just an example again from development. This is the development of the limbs during development. They initially occur in the developing embryo as protuberances. This is a mouse embryo. And then they get progressively larger. Those bumps, those protuberances, and then you get the limb paddle being formed. And now this is something that's approaching the shape of the final limb form. And then the interdigital cells die. And now you get the digits formed of the final hands and feet. But obviously you overproduce cells initially in order to do that. And the same happens when you're making blood vessels. You make a solid tube and then you cavitate out the center of the tube in order to make the blood vessel. So this is Xenorhabdus elegans, a small nematode worm that's been used to study programmed cell death. And as we'll get to later in the talk, Xenorhabdus elegans, there's 1,090 cells born, and then 131 of these cells die during development. They're disc-like cells, like button-like smooth surface cells. Now the worm is actually now at an advanced stage of development, and it's moving in the egg case, so you can't see it. But the why I bring this up is that a breakthrough came in the study of programmed cell death, realizing that cell death is genetically encoded during development, and genetic screens that was done in the laboratory of Bob Horvitz identified all of the genes that regulate programmed cell death in the worm. And as I'll mention a bit later on, this basic genetic blueprint has been conserved from worms to mammals. It's considered more complex in mammals, in ourselves, but the broad architecture of cell death is preserved from simple nematode worms that only have 1,000 cells through to mammals that have 1 trillion cells. And that's pretty remarkable. And in development of the worm, the programmed cell death that takes place, so over 10% of the worm dies during development, and the cells that die always die at the same times and the same places. So this is highly genetically encoded. Oops. Okay, so I'm going to go through the different modalities of cell death now to take us through some of the molecular mechanisms. And I posed a question here, so how do cells die? By accident or design? Well, both. There are accidental types of cell death, which I'll talk about, necrosis, and then there are programmed cell deaths, and predominantly apoptosis is what I want to talk about. So necrosis can be viewed as murder. In necrosis, cells essentially burst open very rapidly. They release their contents. That has a big impact on neighboring cells. That's really a damaging mode of cell death that it's best to avoid. When you need to eliminate cells, it's better for the cell to be involved in packaging itself and to enable it to be removed from the tissue quite silently without bursting open. So a lot of what happens during apoptosis is about allowing the cell to be removed without causing damage to its neighbors, and also, very importantly, without activating the immune system. So necrosis is a type of cellular murder. The cell doesn't participate in its own death. Events are imposed on it and the cell just ruptures. Apoptosis is more akin to a cellular suicide, where the cell's an active participant. Most of the time in apoptosis, the cell switches on its own programmed cell death machinery. And then there's a routine, a subroutine of apoptosis, where the cell doesn't switch on its own programmed cell death machinery, mainly usually because it's infected, or maybe there's some mutations in the genes. And then in this case, we have specialized cells called cytotoxic T cells or natural killer cells that can switch on the programmed cell death machinery within the target cells. And again, cells die by apoptosis in this particular case. So these forms of apoptosis molecularly are very, very similar, but apoptosis is very different in necrosis. Now, I like to sometimes consider this with a little joke here. This person is not dead, they're meditating, and this is an advanced form of meditation where you stay in this position for 30 minutes or an hour. And this person is in a superficially similar position, but he hasn't got there by design, whereas this gentleman has, and it's unregulated. And obviously this is considerably more damaging for the individual than this type of meditation. So apoptosis is highly regulated, necrosis is not regulated. And as I've said a few moments ago, it does matter how cells die, because although these are both modes of cell death, the difference that they make to the impact on them, the cells around them, and to the body are very profound. There are consequences for how, there are consequences for those left behind, depending on how a cell dies, because the immune system basically doesn't activate in response to apoptotic cells. The immune system ignores apoptotic cells. It deals with apoptotic cells very differently to how it deals with necrosis. Whereas when the immune system perceives necrotic cells, it gets very activated in a manner similar to how the immune system responds to infection. And that's because molecules released by cells that die by necrosis activate the immune system, and those molecules have been called damage-associated molecular patterns, DAMPs. So DAMPs, when they're released, they activate macrophage and dendritic cells, they cause the production of more cytokines, and they ramp up a process, they ramp up the inflammatory process. And really, you don't want that to be happening when cells are dying during development, or when they're dying in situations where there's only been mild injury to just one or two cells. You don't want full-blown activation of the immune system because it's very wasteful in resources. And also the immune system is damaging. Now that damage is normally balanced by the threat posed by infection. So when you get a full-blown inflammation, that also causes damage in your tissues and makes you feel sick, but it's worth it if you're fighting off a big infection. But it's not worth it if you're just eliminating a few cells that have died, because of a bit of DNA damage, or because they're aged or dysfunctional in some way. Now, just showing you an example of this is a laser has been used. This is by Lammerman and colleagues, and it was a paper published in Nature a number of years ago. And this is in vivo. A laser was used to wound, to cause necrosis in a tissue. And this is basically, you have fluorescently labeled neutrophils, and these are first responder cells of the immune system. And within minutes after laser-induced injury, you can see the immune system here has been massively activated as a result of necrosis. You get very rapid recruitment of neutrophils to the site of necrotic cell injury, just as if you would if it was an infection. And once those neutrophils go in, they release all sorts of nasty enzymes, and they cause further inflammation. And this is what happens when we get a cut. Typically, we get this massive kind of wound healing reaction, and our tissues swell. And really, you don't want that to happen if you're eliminating cells in a more natural way that happens during programmed cell death. So this is a movie of that. The laser injury has just been induced, and you can see these swarms of neutrophils going in, very, very quickly upon laser-induced wounding. And then they amplify, the neutrophils further amplify the inflammatory process in ways that I don't have time to explain. So we know what the key DAMPs are, the key damage-associated molecular patterns that are released from cells that have been damaged to undergo necrosis. There are molecules such as IL-1 alpha and beta and IL-18, IL-33. My lab has worked on all of these cytokines that are released through necrosis, and they then undergo proteolytic processing to activate many different cells of the immune system and ramp up inflammation. And I don't have time to tell you much about this family of cytokines today, but they're a very important cytokine family that are all released when cells die by necrosis. So what typically happens, obviously a live cell is not emanating anything, so immune system ignores live cells. Necrotic cells release stamps. That's indicative of danger, make an immune response, whereas apoptotic cells don't release stamps. And for reasons I'll tell you, they're recognized as a result of changes that take place in their cell surface. They're recognized and they're eaten by the immune system at an early stage, and there is no immune response to them. I'm going to skip these slides, but just to say that one of the consequences that would occur if we constantly had a lot of inflammation going on in our tissues as a result of the cell death that takes place in our tissues is that inflammation is a driver of many diseases, and inflammation has been linked with many chronic diseases such as cardiovascular disease, Alzheimer's, diabetes, cancer, neurological disease. These are all non-infectious diseases, but it's got to do with inappropriate inflammation taking place in our tissues for various reasons. And, you know, this would be much worse if we were mounting inflammatory responses in response to programmed cell death. So one of the major forces driving programmed cell death is to avoid this type of chronic inflammation. And that just really summarizes what I've said. So how is cell death regulated? Well, as I've told you, in necrosis, the cell doesn't choose to die. Things that kill it are typically severe trauma, direct compression injuries that causes very rapid rupture, high concentration of toxins, high temperature, very low temperature, high dose radiation. Really nasty stuff is what typically causes necrosis. So typically what happens, cells, they detach from the substratum that they're adherent to, they swell very rapidly and they burst open and release their contents. And this is what it looks like. So this is a movie we took a number of years ago now. These cells are dying in the space of 1 hour in response to hydrogen peroxide. And what you can see is these blebs forming on the surface of the cell. The cell just ruptures, it releases its contents. And all of the cells die very quickly, all, you know, at the same time within this 1 hour time frame. And that's very different to apoptosis, where each cell makes its own independent decision to die many, many hours apart. In a movie I'll show you in a moment. And these cells have released all of their contents. And, you know, cells have lots of nasty enzymes, proteases, lipases, RNases, DNases, carbohydrates. And if you release all of that together, it causes a lot of damage to surrounding tissue. So necrosis typically causes additional necrosis as cells release their contents. So in apoptosis, cells effectively choose to die. The things that cause apoptosis are natural triggers made by the body, molecules that are protein, they're not injurious in any way. So TNF or FAS or TRAIL, these are just proteins that bind to receptors on the surface. But also apoptosis is induced by mild damage or injury. So we cause DNA damage or mitochondrial damage or cytoplasmic, proteotoxic stress. These types of stimuli also evoke programmed cell death. The cell perceives the damage and actually switches on its programmed cell death machinery. And this is the best way for cells to die. As I've been saying, cells round up, they detach, the nucleus condenses, it breaks up into small pieces, the cell condenses, the cell actually blebs into these small pieces, and the cell still remains intact. It doesn't burst open, it doesn't release its contents. And most importantly, at this stage, you get small pieces of the cell shed off and they're eaten by surrounding cells or macrophages. And so cells are given permission to eat their neighbors during apoptosis. And that goes back to a slide at the beginning where I said that normally in a cellular society, cells are not allowed to eat their neighbors, but during apoptosis, they're given permission because there are changes on the cell surface. And the major change that we know of is the externalization of phosphatidylserine, a lipid that's not normally present on the cell surface, that externalizes during apoptosis. And then neighboring cells and macrophages recognize this change, and that gives permission for the cells to eat one another. And that was the stain I referred to at the outset. This red stain here detects phosphatidylserine exposure. So this is an apoptotic cell here in the center. You can see all these little blebs being produced. They're called apoptotic bodies. And this is a way to as I say, facilitate disposal of a cell that has to die, that needs to be eliminated, but without causing disturbance to the rest of the tissue. This is what it looks like. This movie is taken over 24 hours, so much longer than the other movie. And so this is several hours into the movie already, and only this cell is starting to undergo apoptosis. Now these other cells start, but you can see the process goes on for a long time. There's no bursting open of the cell. The cells are all independently initiating the process, many different hours apart. And so here we're 18 hours into the movie, and this cell still hasn't initiated the process. And so cells remain intact for very much longer, but already within an hour or two of displaying the blebbing phenotype, phosphatidylserine has been exposed. Even at this stage, phosphatidylserine will have been exposed. And that would enable, if this were in the body, a macrophage or another cell to eat the cell. And these cells are actually dying in response to a chemotherapeutic drug, daunorubicin. So many chemotherapeutic drugs that are used to treat cancer induce this type of cell death. So phagocytes capture apoptotic cells and eat them because of the changes that take place early on in this process. And as I've mentioned, that phosphatidylserine flip that occurs on the surface can be detected by an XM5, which we discovered about 25 years ago now. And an XM5 staining is very widely used for the detection of apoptotic cells. And there's an example. So here, cells are being stained for phosphatidylserine by looking at an XM5 binding. And this is a healthy cell population. And then early in apoptosis, you can see this population emerging. These are binding in XM5. And now virtually all the population are binding in XM5 as we go through the process. So phosphatidylserine is externalized during apoptosis, and we can detect this by probing cells with an XM5. And here we have cells that an XM5 is stained here in pink. Cells don't initially stain it, and then as they go through the process of apoptosis, you can see that the cell lights up with an XM5 staining, and also the blue stain is where a DNA binding dye now is getting into the cell during apoptosis. So I'll just play that again. Oh, I can't. Oh, yeah, there. So once the cell starts to manifest apoptotic features, you'll see now the XM5 staining lights up on the cell. And that would be normally, as I say, the signal for the cell to be eaten by its neighbors. So that's really the major functional outcomes when a cell dies by apoptosis. It makes changes to the surface, as well as releasing certain signals I don't have time to go into, and that facilitates its uptake by macrophages and other cell types, and you get either no inflammation or an anti-inflammatory response. Whereas in necrosis, cells burst open, they release stems, and then this is perceived by macrophages. They get activated, they make more cytokines, and this is an inflammatory response. Now there's a subroutine of apoptosis, and it's, as I mentioned, in a case where a cell cannot kill itself because it's infected with a virus. In this case, we have cells in our body that patrol the tissues. They look for cells that are infected with viruses or that are potentially cancerous, and they then deliver a trigger to those cells that turn on the cell's programmed cell death machinery. And so these natural killer cells or cytotoxic T cells are really important for eliminating cells that are infected with viruses. In fact, we've heard a lot about them with the SARS-CoV-2 pandemic and the coronavirus pandemic. Cytotoxic T cells are really good at getting rid of the virally infected cell by turning on their own cell death machinery. Here's an example. These are most of the cells in the dish that are flat, they're cancerous cells. The cells moving around, such as this cell here in the bottom left-hand corner, this is a cytotoxic T cell, and we've added them to the cancerous cells. And this cell here is going to be killed by this cytotoxic T cell. This, in this case, is natural killer cell. Now it's degranulating. It's delivering in proteins into the target cell. We know what these are. The major one is called granzyme B, and that turns on the target cell programmed cell death machinery, as you can see here. Now the cell is undergoing apoptosis, and it's happened very selectively just to this cell. You see the way this other cytotoxic T cell is moving around very rapidly searching for infected cells. So that's a type of involuntary apoptosis where a cell is executed. So how does this all work? What happens within the cell during apoptosis? Are there genes that regulate it? I know that I don't have a lot of time left, so I'll go through this very quickly. We know quite a lot about programmed cell death now. Work started in the worm, really in the 1980s, in Xenorhabdus elegans, this small nematode worm. And as I mentioned earlier, genetic studies were done in this worm looking at development to identify 131 cells that died during development. And then as a result of mutagenesis screens, the genes were identified by essentially doing mutagenesis experiments. And this framework was put together where 2 genes were found to be essential for cell death. They were called SED3, cell death defective 3, and SED4. So they were required to drive cell death. SED9 was normally blocked cell death, and EGL1 inhibited the activity of SED9, so it promoted cell death. And it turns out after work actually that went on in parallel in human systems and mouse systems, SED3 is a protease. It's a member of the caspase family of proteases, and it's switched on by SED4 in order to drive programmed cell death. And SED9 can block the activation of SED3 by binding to SED4. And as we'll see, this basic genetic framework has been preserved in mammals. We have caspases, which are the SED3 homologs. Now there's 12 caspases, or in fact 14 if we include the mouse caspases. And then the SED4-like protein in humans is APAP1. And the SED9-like protein is the BCL2 family. There's actually 6 members of the BCL2 family, or the pro-survival members. And there are 8 members of the BH3 only. This is the EGL1-like proteins. Now there's broad similarity, but of course it's also very different in the detail. It's not just more complicated, but there's lots of differences too. I don't want you to think it's exactly the way it is in the worm, but the worm is, roughly speaking, a good template for how mammalian programmed cell death is regulated. And both the worm system and the human system and the fly system have informed each other in terms of really elucidating how it all works. And this is a summary of human programmed cell death. We now know there's 3 major pathways to turn on the cell death machinery, either from the cell surface by engaging so-called death receptors. These are molecules within the TNF superfamily. And when the molecule such as TNF binds to this receptor, we know all of the kind of pathway involved and how that's routed and the multiple caspases that are activated. I'm not going to go into the details, but suffice to say that when these caspases are activated, they're proteases. They then cleave lots of proteins in the cell in order to coordinate the cellular execution process that we call apoptosis. We can turn this on with cytotoxic T cells, as I mentioned a few minutes ago. They inject in granzyme B, as well as other proteins into the cell. And we know how granzyme B works. It turns on caspases in 2 different ways. And then there's an intrinsic sensing mechanism that the BH3 only proteins regulate. And they sense lots of different types of damage and injury. And in response to that, they open up pores in the mitochondria to turn on the programmed cell death machinery. And so we've learned a great deal about how apoptosis is regulated over the last 20 years. There's still much to learn, but we know a lot at the moment. And the BCL2 family in particular has turned out to be very important as a major checkpoint in the regulation of programmed cell death. And often in cancer, BCL2 pro-survival family members, and there are quite a few of them, they're often overexpressed in cancer. And this is how a lot of tumors avoid switching on the cell death machinery. They overexpress BCL2 or BCLX or MCL1, and that allows them to become very difficult to kill. And as a result of that, these BH3 only proteins, they've been mimicked with anti-cancer drugs. So there was a drug that was developed again over the last 20 years called venetoclax. It mimics the BH3 only proteins in order to antagonize, to inhibit the BCL2 pro-survival proteins and promote programmed cell death. Now I've mentioned, I'll briefly mention the caspases is an area that we've worked a lot on. So apoptosis ultimately is about activating caspases. These are proteases. As I've said, there are 14 of them when you include mice, 12 in humans. They're normally kept inactive in the cell, but once they get activated, they cleave lots of different proteins. And we don't understand exactly what all of these proteolytic events do, but we know that they are required to turn on all the changes that take place during apoptosis. And there's lots of different substrates for caspases that we and others have identified using proteomics methods, for example. And this is from a review we wrote for Nature Reviews a number of years ago. If you're interested in this topic, we've explained exactly, what, caspases do in terms of activating downstream events. But I like to think about it like this. When caspases are turned on, they really wreck the cell, but in a regulated way. These are not caspases, obviously. This is 2 children of a friend of mine. He was painting and he left his children with the paint while he went to answer a phone call. And this is what he found when he came back. They had helpfully redecorated the sitting room. So caspases, really what they do is they make sure that a cell is shunted towards apoptosis so that it's immunologically silent. And if they're not activated, you get necrosis and it's much more inflammatory. Now I realize I'm running out of time, so I'll finish up by just mentioning for a moment what we're currently working on. We're currently working on a kind of related aspect to cell death. We're working on understanding when cells are stressed by things such as chemotherapy that could kill them, sometimes they make an inflammatory response if they don't die. And so anything we've discovered that can potentially kill cells can also trigger this stress-associated inflammation. We think it's very important in helping, for example, cells avoid the cell death effects of chemotherapy and in other situations as well. When we're trying to understand how this type of inflammation works, it's a novel type of inflammation. There are 2 major drivers of inflammation known: infection and severe injury via PAMPs and DAMPs. We know how these work. We don't know how stress-induced inflammation is regulated. And we've just been funded for 5 years by the European Research Council with an advanced grant to investigate this problem. And it's very important in cancer, this type of stress-associated inflammation, because we think, well, we know that tumors benefit from this type of inflammation, and we're trying to understand how it's regulated. So there's still a huge amount to discover. We know a lot about programmed cell death, but we know that we don't know everything. And this is actually not a cell, but this is a star cluster taken with the Hubble telescope. And it's obviously very complex, space is almost infinite. And this is not a galaxy, but looking at just a tiny fraction of the protein complexes in a cell. And as we zoom out, we see there are thousands of different protein clusters, all doing different things, all populated by different proteins. It's immensely complex, just like deep space. And there's so much more to learn. In fact, so much that this is sometimes still, I feel, the level that we're at when we're looking at cells, at a very crude level. And we really need to understand things in much more resolution. And a word finally on doing research. It's fantastic doing research. It's something that I really fell in love with when, in fact, already as an undergraduate level, I did my final year research project in a lab. And I just realized I loved working in a lab. No 2 days are the same. It's like having a hobby for a job. It's something that's very rewarding. You have to work at it. It's hard work. But when you reach a certain point, it doesn't feel like work. And, I feel immensely privileged to have a job like this. I definitely think it's a lot easier than working in a coffee shop or working as a taxi driver. Those are much harder jobs. So I would certainly encourage those of you who are interested in doing research to explore that further. And finally, I'll finish up by thanking the people and acknowledging the people who actually do all the hard work in my lab, who work at the bench. And I've had some great people over the years. And it's very gratifying to see them going on in their careers. And many of them now run their own labs in different universities in the world. So I'll finish up by just mentioning Essential Immunology, a textbook that I've been involved in for quite a long time now. And if you're interested in immunology and programmed cell death and immunity, I'd recommend the book. And thank you very much for your attention.
Thank you so much for your speech, Professor Siamas. We have some questions that our participants going to ask to you. First question, could increasing apoptotic activity be the solution to resistance to primary and secondary treatments in cancer?
Yeah, absolutely. Certainly, avoiding apoptosis is definitely probably the major mechanism of resistance. The whole point of cancer therapy, of course, is to kill cells. And we know that most chemotherapeutic agents and radiotherapy kill predominantly by inducing apoptosis in the tumor. And so by definition, if the chemotherapy fails, then clearly the cells have avoided, they've developed resistance to apoptosis in some way. So yeah, that's the goal of cancer therapy, to increase the frequency of apoptosis that's taking place. And that's where a lot of the focus has been placed over the last 20 years. And as I said, we have some agents now that can do that. There are BCL2 inhibitory molecules such as venetoclax and others in development, and they mimic the BH3 only proteins, and they lower the resistance of the tumor to chemotherapy.
If possible, by the way, thank you for your answer. Other question follows with, if possible, can you briefly touch on the crosstalk between autophagy and apoptosis? Actually, I wonder the major differences between them, one of our participants asked.
Between autophagy and apoptosis?
Yes.
Yeah, they're very different processes, really. So there's been some research, research goes in fashions at times, and there was a fashion at one point to look at connections between autophagy or autophagy and apoptosis. And in fact, they're very, very different processes. And there are some points of regulation. Autophagy really is a way that cells use to dispose protein aggregates or organelles that are damaged. And it's literally means self-eating. Cells also use autophagy to survive under situations of starvation. So it's not a death process at all. If taken to extreme, cells can die if autophagy goes on too long, but primarily it's a mode of survival. And in fact, normally what you find is if you inhibit autophagy, you get more apoptosis, because if you block the survival attempts, then the cells will die more readily. So autophagy is in a way, it's a similar healing process, but healing at the cellular level. The cell is eating its own organelles to survive, or eating protein aggregates because they're toxic. So it's a way of surviving. And then apoptosis is a way that the tissues heal, where effectively the tissues eat cells that are unhealthy. But in terms of the actual links between them, there aren't a huge number of actual specific molecular links. There is some overlap with the BCL2 family members that have been implicated in both processes. But the names are very similar, but I think the processes are actually quite distinct.
Thank you so much. Other question follows with, is it possible to make that cancerous cell to go apoptosis with producing anti or pro-apoptotic molecules such as BCL2 family molecules?
Yes, so as I mentioned, the BH3 only members of the BCL2 family, they normally neutralize the pro-survival BCL2 family members. I know I went through this quickly, but there are lots of good reviews on this topic, for example, by Anthony Latai and colleagues, LETAI, if you're interested in the topic. And the BCL2 family neutralization strategy is a major strategy for improving cancer therapy at the moment. Venetoclax, if you read about that, VENETOCLAX, venetoclax is a drug that specifically neutralizes BCL2 family members.
And other question is, thank you so much for the response, by the way. The other question follows with whether the apoptotic activity, increasing the apoptotic activity could be the better solution for the primary cancer cells and the secondary cancer cells?
I think that question was asked already.
Really?
About,
yeah.
I think, evasion of, therapy.
Sorry
So,
for the, I
no
need
problem.
to, in some occasions, I need to just retranslate because some of the questions are in English, so that's why I need to just flip-flop.
Don't worry. I'm sure it's difficult. Yeah.
Sir, I have a question for you.
Yep.
You just said that working in the lab, and especially in this topic, is easier than working in a coffee shop, right?
Well, in some ways, yes. Yeah.
Yeah. I'm asking this behalf of my friends from undergrad students who are watching us right now. Sometimes we get really overwhelmed by the work from our lectures. So
Yeah.
do you have any suggestions to, how can I say, do you have any suggestions that makes more motivated about in our jobs that we will?
Sometimes you get overwhelmed by the work, by the amount of work or the complexity of the work, or
sometimes complexity, but sometimes the amount of the work we do. But we always try to find time. Also try to put up events like this congresses to speak up with our academics, just like you. So do you have any suggestions how to organize in this area?
Yeah. I don't have suggestions. What I have is, all I can tell you what I've experienced and what I've seen myself over the years. And what I've experienced is, it's hard at the beginning. It's hard when you're learning a lot of new things. And then it gets easier, because now you establish the foundation that you have to build on later. And also you now can see the connections between things easier. And like anything, if you want to become a great musician, at the beginning, you sound terrible. At the beginning, it's very hard, you seem not to be progressing. And then you learn how to play, some songs and it sounds good. And then you get better, but you also realize, because now you're tuning in to how really good music sounds. And you realize that, no, this can be better. I can be better than this. And you do this for yourself, really. So it depends on what standard you want to set, and it depends on what kind of life you want to have. So, some people don't want to work that hard. They don't want to know. They just want a job that is easy. And they don't love learning. If you love learning, it's still hard, but it gets easier. And, at some point you kind of see the Matrix, I don't know if you've seen that movie, but at some point you realize, wow, this stuff kind of is easy. It's like intuitive. And, but I'm sure that's like that with anything where you have to put some effort and energy in. I make the comparison with music deliberately. Unfortunately, I love music, but I can't play any musical instruments. It looks very complicated. I have guitars here, but I'm terrible,
by the way, so I understand what you mean.
And you need to practice, and it's difficult. But once you reach a certain level of competency, now you can fly. That's kind of what I see. Now you can start making your own original compositions, and it gets easier. Like being an artist, I'm sure is the same. I love art. I really, I know enough about art to know the difference between a very good painter and not such a good painter. But and I know that I can recognize the skill, the craft. It doesn't come overnight. Nothing is easy at the beginning. So, if you're finding it difficult at the start, that's fine. Everybody does, and it's a question though is, do you enjoy the process? Because when I go through difficult things, and I do all the time still, things that I hate doing while I'm doing them, it's like I feel I'm trying to squeeze something out of myself. But when I get through to the other side, I'm proud of what I did. And when I look back at it, I kind of go, yeah, that's pretty good. I don't tell other people that, but to myself, I'm proud of maybe the effort I put in. And when I say that, it's easier doing science than working in a coffee shop or, building houses. I mean, what I mean is, I go to coffee shops every day and it looks like hard work to me, It looks like you're standing on your feet all day. You're doing the same thing over and over. It would be very difficult for me to stay motivated and enjoy that kind of work, you know. So that looks like a hard job to me. But with science, it's enjoyable. It's easy. I mean, I get to go to lots of different places in the world. You know, there's people working in the lab on lots of different things. I get to teach students. I get to write books and give talks, and it's quite a creative, stimulating profession. So, but it's not easy at the start. You know, it's not easy at the start.
Yeah, I'm sure
but it gets easier.
I'm sure my friends who think the same way, who are not overwhelmed by the work, will be guided. Thank you so much, sir.
Yeah, thanks for that question.
Thank you so much for your attendance to our Congress. We will basically want you to go back to the breakout room.
Okay. So I will see a prompt, I think. So have a great rest of your conference. And I wish I was in Istanbul, beautiful city, the cradle of civilization, great food, great sights. And I hope to visit again soon. Thank you.
Thank you, sir.
Now, am I in the breakout room or not?
No, you're still in the main room. There should be right next to leave the meeting an option with 3 dots saying more options, I think. Mine's in Turkish. I have difficulties saying what's what.
I just see leave the meeting.
If you click on the information bit with the 3 dots, there should be join the break room.
Yeah, I'm not
Professor Martin, if I may, are you using a Mac OS device or a Windows PC? The
I'm
interface.
using my Mac.
All right, so next to reactions under your bar, when you hover your cursor, there should be breakout rooms between record and reactions.
Good, yeah, I see the break.
Our next speaker today is Dr. Nathaniel Hakanakorn from the University of Edinburgh. Today he will enlighten us on the topic of cell-free synthetic biology, which has been fundamental to the development of modern molecular biology, in vitro transcription and translation technologies. He will describe 2 projects which combine cell-free systems with microfluidic technology in his speech titled Synthetic Biology with Cell-Free Systems. Welcome and thank you for accepting our invitation. If any of our participants have any questions for the duration of the speech, they may write it in the YouTube live stream chat or through Instagram DM. Thank you.
Thank you. Can you all hear me and see my screen?
Yes, it's great.
Perfect, great. Well, thank you so much for the kind invitation to speak at this conference. So yes, the title of my talk is Synthetic Biology with Cell-Free Systems. And I am at the University of Edinburgh. I recently started my lab, actually about 2 years ago now. It seems quite recent, but I think with the situation of the last 2 years, the time has somehow sort of contracted. So I'll start with a quick background about myself. So my background is actually in physics. So I studied physics at the University of Cambridge, and I focused on nanopore and single molecule biophysics. After which I moved to Switzerland, to Lausanne, where I was introduced to cell-free synthetic biology and microfluidics. So I worked in the lab of Sebastian Merkel. And I moved to Edinburgh in 2019 to start my own lab, which basically builds on a lot of the skills and background that I've acquired in my previous career. So cell-free systems, the fundamental aspect of cell-free systems is that they harness biological processes outside of cells. Right, so you can imagine a simple biological process like an enzymatic reaction. And you can easily see that if you take an enzyme and combine it with its substrates in a test tube, you might get some activity. which may be useful. You can also do something more complex. So if you extract the cytosolic components of an E. coli cell, for instance, and combine it with the appropriate biochemical energy and substrates, as well as DNA instructions, you can carry out in vitro transcription and translation to make protein inside a test tube. And this is obviously a more complex example of in vitro processes. In my lab, we're broadly interested in both the understanding aspects of this approach, as well as the biotechnological aspects. And the two projects that I described today will address each of these items. So the first one is understanding. So cell-free systems have been used for a very long time to discover things about biology. And they're useful for this because, first of all, you can construct and test hypotheses by directly experimenting on specific biological processes in open reactions. Right, so you're no longer constrained by the barrier around the cell, but you can actually try to isolate the individual processes in a test tube, which you can then perturb. And you can change the conditions and try to identify the important factors in this reaction. The second way of understanding is actually a constructive process where you try to build minimal systems. So you ask yourself the question, what are the components that I think are responsible for a particular process? You isolate those, and then you try to make that process happen in the test tube. So that's a direct test of your hypothesis. So a very famous example, so both of these processes have been both these approaches have been very important for molecular biology. A really important example of the first is the elucidation of the genetic code. Which was done in the 1960s. And here, what the researchers did was they carried out cell-free translation using E. coli cells, which have been broken apart in exactly the same way that I described before. But what they observed was if you actually put synthetic mRNA into the system, you get a polypeptide, right? And if you design your synthetic mRNA to contain only certain elements, so poly-U, for instance, you get certain types of polypeptides, like polyphenylalanine. And by doing this very carefully, by changing what mRNA you put in, you can slowly decipher the mapping between the genetic code on the mRNA and the polypeptide that you get. So this is possibly one of the most famous examples of the use of cell-free in molecular biology. In terms of the minimal systems, one way you can think about this is that instead of taking the E. coli cytosol, which contains numerous, thousands of factors, you can ask yourself what components are important for protein synthesis, let's say. And these might be the ribosomes, polymerases, and so on. And you can extract and isolate all of these components and then try to construct a purified enzymatic system. Right, and an example of this purified system is something called PURE, which stands for protein synthesis using recombinant elements. It was developed by Shimizu et al. Back in 2001. And it is a minimal system for in vitro protein synthesis. So it contains 36 components, which may sound like a lot, but protein synthesis is actually a complex process, and the fact that you can do it with only 36 components, maybe actually it's quite impressive. So in protein synthesis, there are 4 main processes which have to happen. So you have transcription and translation, obviously, to convert DNA to protein, but you also require amino acylation, and this is the charging of tRNAs with amino acids. And because all of these processes consume biochemical energy, you need an energy regeneration system to regenerate your ATPs and GTPs. And so if you take your 36 protein components, which make up these 4 subsystems, and combine them with the appropriate substrates in your test tube, what you see is you can make protein from DNA. So this system is impressive. I say it's a tour de force of in vitro reconstitution. It's minimal, by which I mean if you remove any one component, the activity of the system suffers dramatically. And it's completely defined. We know exactly what goes in the test tube. So this shows that we really understand what's going on about this fundamental biological process of protein synthesis. So a natural question is to ask, what other processes can we study in this way? Well, we've actually been, in molecular biology, we've adopted this approach to study numerous processes, not just protein synthesis, but things like DNA replication or the biosynthesis of membranes, the development of cellular structures like using cytoskeleton, metabolic pathways, and so on. The point is that all of these processes are carried out in isolation. We're developing minimal systems to figure out what are the smallest number of components we need for this process to happen, right? If we actually adopt a slightly different viewpoint, that of bottom-up construction, which is one of these underlying principles behind synthetic biology, the idea is that if we can slowly combine these processes together to make more complex processes, might we be able to one day make something like a synthetic cell? So this might be a construct where you have protein synthesis. These might make a number of enzymes, which may be responsible for synthesizing the membrane of the cell, and eventually allow the cell to grow, right? But this is a much higher level of complexity. You need to put more processes in. Obviously, natural systems are very complex, far more complex than any of these in vitro systems that we've constructed. So the hope is that by trying to build complexity using these simple processes, whether we're successful or not in eventually creating a synthetic cell, we can learn something about how complexity needs to be organized to carry out these more higher level biological functions. So an example of a more complex process is something I call regeneration. So this is literally the idea that you can make more of the components that you originally had in your system, right? Regeneration of internal components. It's a fundamental biological process because cells need to regenerate all of their internal components in order to maintain themselves at steady state, but eventually grow and also replicate. So you actually need to make more of your starting material. So specifically in terms of our cell-free approach, the question we want to ask is, can we direct the pure system, this is the purified enzymatic system, to produce more of itself? So instead of using the pure system to create any old protein, instead if we program our reaction with the DNA templates, which themselves encode for these components of the pure system, can we make more of itself? So it's a simple question, and it's the first step towards trying to make something more complex happen. So this is the first project I'll talk about, which was carried out in collaboration with Sebastian, my postdoc supervisor, and a PhD student in his lab, Barbara. So precisely what we mean is this. If we take our pure system in a test tube, we add all of the substrates it requires, as well as DNA templates encoding for its components. Over time, can we double the amount of pure system components we have? Right? And actually, if you think about this problem, there are two main objectives you must meet. So first, the pure system itself must have sufficient capacity to make all of the proteins at the original concentrations if we want to double it. And secondly, the proteins produced must be functional. Otherwise, you're not really making the pure system. You're making some non-functional system. So the approach that people have used to test these aspects is something called the complementation assay. So you start with a pure system, and you allow it to produce GFP. So this is a fluorescent protein which you can measure. So you see that the system is active. It's synthesizing GFP from a DNA template. If you remove one component of the pure system, we end up with something called a delta pure system. And we already said that because the pure system is minimal, if you take away one component, it's no longer functional. Right? What you then do is you set up a second reaction where you have another pure system, but now you have a DNA template encoding for that missing component. And then you add that missing component, you complement your original delta pure system with that missing component, and if it works, you can restore the activity. So this allows you to test how much of this component you need and whether it's functional or not. So people have tried to do this. The approach is quite laborious. So what we did in our lab was we developed a system to do this quickly using microfluidics. So microfluidics is essentially devices which manipulate very small volumes of liquid. It's usually made from this polymer, PDMS, which is a soft polymer, and glass. And the polymer is micro-patterned with these this complex network of channels. And essentially you have two layers of channels. In here, the blue layers is called the flow layer. And so you introduce your reagents, your reactions in this blue layer. And the purpose of the red or control layer is to act as valves. So you fill the red layer up with water and connect it to a pressure source. And then when you pressurize these different lines, you can close off valves in the blue layer. So then you can move liquid around in the device. This particular device is a chemostat. So what this means is that you can fill up your reactor. So the reactor is this rectangular structure. So if you look top down at the device, you can fill your reactor up with the three things you need for your cell-free reaction, which is your small molecules, your protein components, and your DNA. And you see that these are actually filled up in a specific ratio. And once you fill up the reactor with these components, you incubate it. You mix the components together, and then you allow it to sit there, and the reaction will proceed. But periodically, you replace a part of the reaction with, again, these three components in exactly the same ratio. But what happens here is that if you do this periodically, you end up with a situation where your reaction keeps going continuously. So it's periodically replenished. And this allows the reaction to go on at steady state. And so what we see is that we maintain a steady concentration. We maintain a steady concentration in the reaction of all the components. And we allow our cell-free reaction, in this case producing GFP, to eventually saturate at a steady state when the synthesis rate is equal to the rate at which you're exchanging these solutions. So we have steady state gene expression. So then what can we do in terms of our complementation assay? Well, if you allow GFP to be synthesized at steady state, the reaction will eventually saturate at the steady state and keep going. Right? And this is the case where we're replenishing the reaction with template DNA for GFP, as well as the pure system, the protein components. If you, however, switch the reaction and replenish it instead with a delta pure system, which is now missing one of these factors, the reaction will slowly over time consist purely of the delta pure system, which is inactive. So in that case, even if you add the template DNA, what you see is that you lose your activity over time. This is a cartoon to show what we expect. If, however, you actually add DNA encoding for this component as well, what we see is that the reaction, even when you replenish it with delta pure, remains active. And it's active because you're actually also making the second component, this missing component inside the reaction. So this is how we test for self-regeneration of these components. And in terms of the data, this is what it looks like. So we tested it for a large number of components from Pure. In this slide, we're looking at amino acyl tRNA synthetases. So these are enzymes which join amino acids to tRNA. So it forms this process of amino acylation I talked about before. So normally, if you remove one of these enzymes from the system, your system completely loses activity. But we can see that we can keep regenerating these enzymes to maintain the activity in the system. So we can test these synthesases and demonstrate that the Pure system is capable of synthesizing functional synthetases. And importantly, what we observe is that there is a minimum synthesis rate. So if you don't synthesize these enzymes fast enough, the steady state concentration of the enzymes is too low for it to function. And then the system is non-functional. So this is actually a key point. You actually, and the other point that I mentioned, you need to synthesize these components with a sufficient concentration. Right? So how do we ensure that we're above that rate? Well, normally in cell-free reactions, the more DNA template you add, the more protein you get, up to a point. So when you add template DNA for GFP, you see that you get more and more GFP synthesis, and then eventually it saturates. And this is the fundamental limit of our protein synthesis system. This maximal synthesis rate of around 4 micromolar worth of peptide bonds per minute. And what happens, what this is due to is that we have a fixed resource pool. So normally, if these red dots represents resources, as you add more template DNA, you accelerate the reaction until the point when you're using up all your resources. And then actually the more DNA you add, you don't get any more protein. So you've saturated the reaction. And actually what this means is that this limit is the obstacle for us, which prevents us from synthesizing many components together. So we demonstrated that we could synthesize up to 7 of these synthetases at the same time, because when you put 7 in, what happens is that the maximum synthesis rate is shared between each of these enzymes, so that each of the enzymes is still being synthesized at above its minimum synthesis rate. But when you try to synthesize too many components together, the system fails because you no longer have the capacity to make this to synthesize everything at the correct concentration. So the takeaway from this part is that we had a chemostat device which allows a rapid test of the various functionalities and synthesis rates required to generate these individual components in the Pure system. But that the main limitation is actually the synthesis capacity, this number of 4 micromolar peptide bonds per minute. And that's the obstacle at the moment, which is preventing us from synthesizing all of the Pure system. It's one of the main limitations. And so the future work in my lab at the moment is focusing on this problem of how do we improve the synthesis capacity of the Pure system. And so this is a research program which we're just launching this year in our lab. So to switch tack slightly, we'll go back to talking about the other aspect of cell-free synthetic biology. And this is the use of these cell-free systems as a biotechnology. So as a biotechnology, you can imagine that you have this test tube and you can program it with DNA instructions, synthetic DNA instructions. And these DNA instructions can encode for things like RNAs, proteins, or maybe a large system of interacting components. You can just add the DNA templates for all of these different elements into your system. And then these products can be used directly. You can use cell-free systems to produce RNA, for instance, for RNA vaccines. or proteins, or you can use systems based on cell-free synthetic biology, for example, to carry out things like diagnostics or biosensing. Quickly, I'll just mention the feature, to reiterate again, the features of cell-free reactions, which are actually different from gene expression inside cells. The first is that the reaction is obviously non-living. It's a biochemical reaction. A really important point of the reaction is that it's open. So you can control the conditions very carefully, and you can control its composition. And it's simplified. There's no biological elements, biological processes such as regulation or evolution, which normally when you work with synthetic biology inside cells, you have to take into account. So for these reasons, people are interested in using cell-free techniques for technology. So quickly, I'll mention three. The first is protein production, which is probably the most obvious technology from these systems. So typically in vivo, when you want to make proteins, you transform a host cell with DNA encoding for that protein, and then you grow the cells and then allow it to express its proteins. Now, the problem here, or one of the challenges here, is that the process of growth and production is intimately coupled together, right? So your protein has to be made while keeping the cells growing and alive. In the cell-free system, you can decouple these processes, because what you do is you first grow the cells, you make your cell-free reaction, and then you produce your protein in the cell-free reaction. And this decouples the two processes, which allows them to be optimized separately. But importantly, it also allows these cell-free reactions to make things like cytotoxic or proteins which are difficult to be expressed inside normal cells. So one of my students at the moment is working on using cell-free systems to produce therapeutic proteins, such as antibodies. A second example of cell-free biotech is metabolic engineering. So again, in the in vivo situation, what you normally do is you normally transform your cell with enzymes. So these can be, for instance, if you have 3 enzymes which convert a substrate A to a product. In order to make this happen in a cell, you might put these 3 enzymes into the cell. And the difficulties here are you have to control the amount of enzymes you make. And you also have to make sure the cell takes up your substrate A and expels your product in a reasonable way, right? In the cell-free approach, there are several ways to do this, but one way is to make a cell-free reaction where you express the enzymes of interest. And then in this one pot, you also add your substrate, and then you can immediately get your product out. And in order to optimize the reaction, you can change the amount of enzymes. You can adjust the amount of enzymes in your system by adjusting the amount of DNA that you add. And so you can use this system directly, or you can even use it as a guide for in vivo engineering. And another one of my students is working on these cell-free metabolic systems. The third thing I want to talk about is rapid prototyping. So in synthetic biology, we adopt this engineering principle of design, build, test, learn. So we design our system. Let's say it's a biosensor. You have to build all of the parts and implement them, and then you want to observe whether your system works as you hoped it would. Now, the challenge in synthetic biology is that this build, test, and characterization process is typically quite involved, because you're working with living cells. You have to clone them, which takes time. In the cell-free case, you can build parts on linear DNA, which is fast. You don't need to make any plasmids. You can immediately compose these reactions, and you can do this in an automated way, for instance, using microfluidics or liquid handling robots. You can test these designs and characterize them in cell-free, and you can go around this loop as many times as you need until you get something which is working. And you can use your cell-free product directly, or you can try to now implement it in vivo. But you've done a lot of the testing already in vitro. So what the benefit of this is that it reduces the time and cost of carrying out this process. So cell-free really can be a complement to traditional synthetic biology methods. So the second project I wanted to talk about is using cell-free to design a synthetic transcription factor library. And this is the work I carried out in my postdoc, along with Zoe, a PhD student who I worked with. So transcription factors control gene expression. And if you want a library of them, well, there are two basic approaches to finding transcription factors, to developing libraries, is to use existing ones, to mine existing organisms for transcription factors, or to design your own. And we used a library of zinc finger proteins, which are very common eukaryotic transcription factors. And the benefit, the really big strength of zinc finger proteins is that they're modular. So they consist of these repeats of these domains called fingers, zinc fingers. And the domains have these alpha helices. And on the alpha helices, you have a small number of residues whose identity determines what DNA sequence it interacts with. So you can start with a zinc finger consisting of 3 domains, which binds a particular target. But what you can do is you can have different zinc fingers which also bind different targets, but you can shuffle their domains around. So you end up with a combinatorial library of proteins which bind known targets, at least in theory. And then what you can do to carry out transcriptional regulation is to introduce these binding sites inside a promoter sequence. So a binding site in the middle of the promoter sequence will be a repressive site. So when the zinc finger binds, it blocks the binding of RNA polymerase and turns off that gene. Right? So we started off with a large library of zinc fingers with characterized binding affinities. And our first goal was to develop repressors, a library of repressors using these transcription factors. Again, we turn to microfluidics. Looks like a more complex device than the one I showed previously, but actually it's simpler. The function is simpler. So it consists of, again, 2 layers, a flow layer and a control layer. And here you can actually see the control layer in action. So when you pressurize a valve, it pinches off the flow layer here. So essentially this device is a high throughput batch set of batch reactors. So how it's made is we take a glass slide, we use a microarraying robot to spot DNA onto this glass slide with known composition. Right? So we can control exactly what DNA and how much we put on the slide. And then we take this PDMS, the soft polymer part of the microfluidic device, and align it on top of your glass slide. And when you bond it to the glass, you isolate the DNA, each spot, into a different reactor. And that's all there is to it. And basically what you do is you introduce the cell-free mixture into each of the reactors, and then you incubate and you observe. And because each spot has a different composition of DNA, this allows you to very quickly characterize a large number of, to obtain a large data set very quickly. And you measure the fluorescence from each of these reactors. So this is the assay that we did. So in each reactor, we had 2 DNA templates, one of which encodes for a particular zinc finger protein, and the other which encodes for GFP, but with a promoter sequence which has zinc finger binding sites introduced. So this looks like this. So the RNA, the E. coli promoter sequence typically has these things called the -35 and the -10 boxes. Whoops. And these are the locations where RNA polymerase binds. and we have a zinc finger binding site in the middle. So we expect that if the zinc finger can repress the promoter, if it binds, then we should see less expression, right? So we look at this quantity called fold repression. So when we incubate the reaction over time, if the zinc finger does not repress, we see this unrepressed state. But if the zinc finger somehow interacts with the promoter, we see a much lower expression. And the ratio of these is called the fold repression. And so on using our high throughput platform, we can very quickly obtain data sets such as these, where we have a set of zinc fingers and we array them against all of the possible targets in the library. And the two features to notice from this are that, first of all, the fold repression is high on the diagonal. So when the zinc finger is paired up with its correct target. And typically, and this orthogonality is pretty good. There are some exceptions where one zinc finger might bind a different target, but in general, it's quite orthogonal. So zinc finger AAA, for instance, binds its target well, represses its target, but nothing else. The second thing to notice is that the binding affinities, the zinc fingers all repress at different levels. And this can be correlated with the binding affinity, for which we've measured for these zinc fingers already. And what this means is that the more strongly the zinc finger binds to the target, the higher the repression, as expected. So we characterize this binding affinity effect further by making mutations inside the binding site. So as you make mutations, you change the binding affinity, this interaction. And then you see that actually, really, the binding is actually very well described by simple thermodynamics. So you see that this measure of this KD, which is a measure of binding, as the binding increases, as the KD decreases, the fold repression increases in a very particular way. So we can actually control, what this means is that we can control the amount of repression we require by tuning the binding site of the zinc finger. So this was the first effect we characterized. The second one is that the position of the site affects the repression strength, because I said the site itself, the mechanism of repression is steric blocking of RNA polymerase. So obviously if you move the site away from the promoter, you expect that the repression will not work so well. So of course, as you move the site away, upstream of the promoter, the fold repression goes back to 1, which is essentially there's no effect. But we can identify locations where the fold repression is higher, and this is where the sites are very close to the core, the central part of the promoter. The third effect is that we can introduce cooperativity. So cooperativity is when two binding proteins actually interact with each other as well. And this is a very, very common mechanism in natural transcription factors. So the way we introduced cooperativity was to use this domain called PDZ. And this is a protein-protein interaction domain. It interacts with the end, a ligand, a small polypeptide sequence at the end of the second protein in a very specific way. So if you have the correct sequence of polypeptides, a correct sequence of peptides at the end of your partner protein, you get binding. But if you don't have the cognate sequence, you don't get binding. And what we see when we construct such an interaction is that the repression on the promoter increases, but also the nonlinearity of repression increases. So as we introduce more and more zinc finger proteins, the repression changes very quickly, as shown in this red repression dose response curve. And this is a feature of cooperativity, which is taken advantage of in natural systems, this increased nonlinearity. There's some more detail here, which I won't go into so much, apart from to point out that that cooperativity itself is also sensitive to the positioning of the sites. If you move the sites too far apart, then you lose the cooperative effect. And this again makes sense from a purely mechanistic point of view. But the point of all of this was we characterized these effects and then we put them together to build a NAND gate. So a NAND gate is a logic gate, which is common in electronics, but obviously in synthetic biology, when people try to design these systems following the principles of electronics, you want your cell or your genetic system to be able to carry out signal processing. And in cell-free, this is no exception. So a NAND gate is something which requires the presence of two inputs to have a repressive, to produce no output. So it's a system where you always see an output unless you have two things present, right? And the two things here being the two zinc fingers. And NAND gates are ubiquitous in electronics, also in synthetic biology, and they're universal. You can construct other types of logic gates using these combinations of these NAND gates. So the first thing we did was we designed a promoter, and we had a first binding site, the upstream binding site, positioned slightly away from the -35 box. So in this situation, we see that if we have one zinc finger present, we do not get a large repression because the site has moved upstream. So the green is the amount of expression we get without zinc fingers. The blue has this one zinc finger present. Right? The second site is in the middle of the promoter. And the problem with this site is when you have it there, you always get quite strong repression. So here on the bar chart, you see the presence of this central zinc finger, where you see this repressive effect. The expression has gone down. So, and of course, if you put both zinc fingers together, you have this cooperative effect, and the repression decreases even more. So this is not quite a NAND gate, because ideally what we want is we want this situation to not have repression. We want this situation to also have high expression. But we know how to do this, right? Because we know that the amount of repression is related to the binding affinity. So if we make mutations in this binding sequence, we can reduce the repressive effect. So from this situation, which is not quite working, we can reduce the binding affinity slowly. And you see that this lifts the expression of the 0-1 state, of the presence of this downstream zinc finger only, until we get something like this, the 13A mutation, where in all three of the cases where we have no zinc fingers present, or only one zinc finger present, we get a high expression. But in the presence of both of them, the cooperative effect ensures that the repression is low. So this is an example of a NAND gate. And we went on to further develop a number of other logic gates and combine them together to show signal processing. So the conclusion of this part is that, well, for transcriptional repression, there are many things you can tune, you can adjust, such as site affinity or positioning, or things like cooperative interactions. And you can put these properties together in quite a rational way, in quite an engineering-based way, to engineer something with function, like a transcriptional logic gate. But the point of this project was also to show that you can do this very easily with microfluidics and cell-free systems, because this amount of data, to take this amount of data in vivo is a lot more work. So these technologies help accelerate the engineering design cycle. So in summary of my talk, I talked about understanding, which is when we try to build more complex cell-free processes, and eventually leading to a synthetic cell, these tell us what the minimal processes necessary for life are and what conditions are required for them to operate. So the hope is we learn something about how, under what conditions, we can make more complex biomolecular systems operate. And for biotechnology, we see that these cell-free systems can complement the in vivo synthetic biology approach because of various advantages I talked about: bioproduction, metabolic engineering, and prototyping. And if you combine cell-free with microfluidics, it helps control and accelerate the characterization of these systems. So that's all from me. I want to thank all the members of my lab, as well as my collaborators, Zoe and Barbara in particular, as well as Richard from Caltech, who helped a lot with our transcription factor engineering project, and my funding bodies. Thank you very much for your attention, and I'll be happy to take any questions.
All right, Dr. Hagenogan, thank you again for the brilliant presentation. We have a couple questions that were sent to us.
Sure.
Let me just ask you in a moment. So, first of all, we would like to ask, can cell-free reactions be used to simulate organ-on-chip processes?
That's a very interesting question. I've not really thought about this. There is, well, the answer is I don't know. I've not thought about this. But if I may speculate, there are examples of combining cellular processes with cell-free processes. Right? So this is where, for instance, if you have a cell-free reaction, but let's say you also have some cells inside the reaction, you can use the cell-free reaction to interact with the cells and vice versa. In terms of organ-on-chip, I imagine it may be a similar thing, but we would have to, yeah, I think you'd have to see specifically what you need for those processes and where cell-free can help.
Thank you. We have a more practical question now. It states that what should we do if we cannot find translation coefficients for different RBSs in the literature while modeling gene circuits?
Yeah. It's a very specific but very broad, I guess, a very common problem that people face, right? So if you can't find it in the literature, the other approach is to measure it. However, it depends on your system, how you measure this. If you take the cell-free approach, I mean, in cells, the methodology is basically the same as well, I think. But in terms of the cell-free approach, you would build your model and you would try to take into account the experimental signals that you can measure. Let's say it's fluorescence of the protein or maybe the RNA. You'd have to determine, first of all, whether you can actually identify that parameter. You can do this in silico on your computer. You can see if you can carry out this, it's called inference, the process of determining the parameter identity, if your experiment is informative enough, right? If your experiment, if it turns out that you can actually measure the parameters from your experiment, then you go and do the experiment. Otherwise, it remains unknown. Obviously, if your model requires that parameter to be known very well, and you don't know what it is, then you'll probably have to design a better experiment to try to measure that. Of course, at the beginning, from a practical point of view, at the beginning, you guess. When you don't know what the parameter is, you put in a reasonable value, right? But you have to be aware that you're guessing, and you have to come up with a strategy to try to find out to improve your knowledge, basically. So I hope that helps.
All right. We have another question. Inclusion of further functions in the pure system, would the regenerative capabilities stay intact? And if so, could we produce a self-replicating in vitro protein synthesis complex?
Yeah, so I think that's the idea. The idea that eventually we have a synthetic cell, or even a kind of a sustained regenerative system with pure at its center. So this, I guess, goes back to this idea of, what are the limitations? Why can't we do it yet, right? And my take on this is that the pure system is a minimal system for protein synthesis. But minimal systems are kind of by definition the worst systems. So the efficiency is extremely low. And of course, inside a cell, it's much more complex. There are many other factors, but the efficiency is much higher. So the question is, what other factors give pure, what other factors can be added to the pure system to make it more efficient? And of course, at all of this at this time, we want to maintain the function and the regenerative capabilities. So that search for factors, in fact, that is one major goal of our research at the moment, which is just beginning. So I'm not able to talk to you guys about the results there. But yeah.
Would you be willing to entertain one more question?
Sure.
All right. What would be the approach when identifying crucial factors in a biological process during the minimization of an in vivo process?
Sorry, can you say that again, please?
What would be the approach when identifying the crucial factors in a biological process during the minimization of an in vivo process?
Yeah, so that minimization process is similar to, for instance, what is done in these minimal cell projects, right? With mycoplasma, where they try to take cells, remove genes which are non-essential to growth, and try to end up with a minimal genome. I think historically there are many ways to do this. So the minimal cell project is just to try to find the smallest genome possible, and so you think, well, all the genes remaining in the minimal genome must be essential for life. The problem is maybe we don't know what they all do yet. But typically, for in general minimization processes, I think you need a lot of expertise. You might be hypothesis guided. You might have an idea of what factors are important already before you do the experiment, and then you can test that. But I don't think there's a general answer to this question. It depends on the process, and it depends on what you already know.
All right. We have two more questions if you're willing to answer.
Sure, of course. Yeah.
All right. This is a broader question that is directed to you personally. What was the factor that made you move from the field of physics to the field of synthetic biology?
Yeah, that's a good question. So I was always interested in biology. In my undergraduate, I specialized in physics, but I also studied biology and biochemistry. And I wanted to work at this intersection between physics and biology. I guess well, there are a few reasons probably. One is I want to be able to take control of my own data. So it's not always in physics that you get your own data. You can work in large projects where the data comes from elsewhere and you analyze it. I wanted my own data. I wanted it to be quantitative. So a lot of synthetic biology, and especially cell-free synthetic biology, is very quantitative. We build mathematical models of the system. And we can actually adjust the system to test these models in a way which is actually harder to do with living cells. And I found the problems very interesting. I think the problems in biology, are very complex, and there's still a lot of mystery in biology. So that's one. I guess maybe that answers your question a bit.
Right. Another question. It is a very broad question, by the way. Which scientist of our past would work wonders in the field of synthetic biology?
Which scientist of the past would work wonders in the... Oh my gosh, I don't know. Yeah, it's an interesting question to think about. Probably I'll think about this for the rest of the day now. If I don't answer with the name of a scientist, but actually I sort of answer with the name, the qualities that the scientist must have. I mean, synthetic biology is very broad. You have people who are, approach it very, from an engineering point of view. A lot of pioneers of synthetic biology were, engineers, computer scientists. But I think probably the thing that we realize from synthetic biology is that there's a lot of biology also involved. There's things like evolution. There's things like so an engineered biological system doesn't necessarily look like an engineered non-biological system. So if we can really embrace that, so I guess someone who actually tries to take advantage of the biology while having these sort of engineering goals in mind. I know this is a bit of a cop-out. I didn't really answer the question. But I don't know. I mean, synthetic biology is also a new field, so I don't know actually who. Yeah, my answer is I don't know.
All right.
Yeah.
Thank you. It seems we don't have more questions at the moment. Would you be so kind to join us in the breakout room?
Of course, yeah.
All right. Thank you, Dr. Lau.
Thank you. Thanks, everyone. Bye.
Our next speaker is Professor Dr. Richard Jäger, who is a researcher in Bioengineering University of Applied Sciences. Today, his talk will introduce us to the challenges of forensic DNA analysis performed with low DNA concentration in single hairs, explain the principles of PCR-based analysis of DNA degradation, and discuss NGS assays of forensic DNA loci in his speech titled Prospects and Challenges of Analyzing the DNA of Single Hairs. Welcome and thank you for accepting our invitations. If any of our participants have any questions for the duration of the speech, they may write it in the YouTube live stream chat or through Instagram DM. Thank you. I've assumed they checked whether your microphone works and the screen presentation works.
Yes, so can you hear me?
Yes.
Perfect. So before I open the presentation, I just would like to say hello to everybody. I cannot see all of you, so you're just on the top of my screen somewhere. Some of you I'm very happy to be with you. I find this is a great opportunity. Thanks to the pandemic, we can use Zoom, and I'm still in Germany, but I can talk in Istanbul, and that's actually nice. That's very good. So I will open now my presentation. I will share it with you. Where is it? Here we go. Okay, can you see it?
Yes, we can. Thank you.
Okay, so I will start now with my presentation. So the title of my talk is Prospects and Challenges of Analyzing the DNA of Single Hairs. I'm actually a professor of forensic biology, actually on a teaching level, so I do not work on crime cases. I try to develop and improve techniques, and I try to teach them to our students. I'm located in the Hochschule Bonn-Rhein-Sieg, which is in, our campus is in Rheinbach, which is a town very close to the former main capital from Germany, to Bonn. The talk will lead you through those main chapters. So first of all, I try to give you an idea why single shed hairs are of interest in forensics. And then we need to try to understand what are DNA profiles, how we analyze them, in order to understand why actually single hairs are a challenge, an analytical challenge. And I will then discuss with you a highly sensitive method that we developed in order to analyze the DNA quantity and the DNA degradation of single hairs, of human hairs, and also we have a look at dog hairs. And in the end, I will show you how we use this technique to, well, somehow predict the DNA typing success for single hairs that we analyze using the conventional forensic DNA typing analysis. And if some time still remains, I will give you just a glimpse on future directions, how we can actually use next generation sequencing based analysis of DNA in forensics, and how that might improve the DNA typing of single hairs. Now, why is the analysis of single shed hairs of a particular interest in forensics? First of all, I should mention that the modern DNA profiling that we do in forensics is based on PCR, on polymerase chain reaction. That means it is extremely sensitive. We just need to increase our PCR cycle number, and then we can even obtain DNA profiles, theoretically, from a single cell. This sounds good, but there is one drawback. The drawback is that this high sensitivity very often leads to showing up of irrelevant DNAs. Contamination, for instance, or secondary transfer. Think of you and I, we shake hands, then you grab a hammer. Then, of course, it's not only the DNA from your hand that we will find on the hammer, but maybe also my hand, which touched your hand in before. That is secondary transfer, and also DNA traces from individuals that are not connected to the crime case who used the hammer some weeks ago will still be detected. So this is causing some trouble. So as a consequence of that trouble, we often see mixed DNA profiles. The more sensitive the techniques are, the more often we see mixed DNA profiles, and it's difficult sometimes to sort them out. Now, there's one way out if we would like to maintain the high sensitivity but avoid mixtures. Well, we might analyze trace types that cannot be mixed, and such like traces are single cells, for instance, single sperm cells that we may analyze in a rape case, single shed hairs. This will be the content of today's talk. And also we did also some experiments with single skin flakes. So I will focus on the single shed hairs, but we worked on all three trace types. What is now the forensic relevance of shed hairs in general? So they are a frequently encountered trace type. Hairs are everywhere basically, and we all lose hairs all the time. Nevertheless, they are rarely used for DNA profiling, and I will explain you in some minutes why that is so, because they are difficult to analyze. But the advantage, as I just mentioned, is actually one hair is derived from one individual. So the DNA profile we get from one hair is exactly pointing to one individual, and there's no mixture. This shows us the root region, the root area of a human hair. This is a club shape, so this was the insertion part inserted into the skin before, and it would extend into that direction. But we also may be interested in analyzing animal hairs. Here would be an example of a dog hair, the very same area, and you see it has a totally different shape. Why would we be interested in analyzing dog hairs? Well, they can help clarifying the crime circumstances. If, for instance, a crime has been committed in the household of a dog owner, maybe the victim, even if it has been removed, may still have on their body or on the clothes some dog hairs. And then we can, of course, pinpoint the crime site if we can identify the original dog, the dog from which the hairs were derived. This is possible. There are DNA profiling of dogs is some work that we can readily do nowadays in the crime labs. And also animal hairs may generally be relevant, for instance, in wildlife forensics for clarifying wildlife crimes. Such as some examples why animal hairs might be of interest. Main part of the talk I will focus on human hairs, but I will show you some results with dog hairs as well. Now, hairs are quite interesting appendages of our skin. This would be a schematic drawing which I have from that article here. We can subdivide the hair growth phase into different phases. The longest phase would be the anagen phase, where the hair is produced in the hair follicle, which is down here. Here are proliferating cells, which subsequently will cornify, will differentiate, and will produce hair all over the place, and the hair will grow continuously for several years. That's the normal situation on our head or on the bed or wherever. Then after some years, for some reasons, proliferation stops here and the hair loses contact to the living tissue. So this cornified thing is no longer renewed, and with time it will be so to speak, will be expelled. This would be the telogen phase, so it will be resorbed here. It will lose the tight physical contact to the living tissue, to the hair root tissue, and finally it will be shed. We can subdivide that phase, very often is summarized as the telogen phase. We can also subdivide it into two stages, the actual telogen stage and the exogen stage or phase when it is actually shed. So the telogen hairs, these are the ones which are naturally shed and which we can find easily without plucking them out. So now what happens during the hair growth is actually, if we think in terms of DNA, which interests us, is that down here we have the proliferating cells of the hair root region in the follicle, and when they differentiate and cornify, the cell actually dissolve and the DNA will become degraded, also the nuclei dissolve. So along the hair shaft, we have almost no intact DNA any longer. And this is of course preserved in the later stages as well, such that in a shed hair, we have not much DNA, only little DNA, and the DNA is degraded, it's damaged somehow. We will discuss this in a minute. But of course, by that means the shed hairs are dead structures, cornified dead structures that do no longer contain cellular tissue. So degraded DNA, nuclear DNA, which is providing an analytical challenge for forensic DNA typing. And in order to understand the challenge, I have now to introduce to you how DNA profiles are actually analyzed, what they are and how they are analyzed. So on that slide, I first show you the diploid set of chromosomes of a human cell, numbered from 1 to 22, and here we have the 2 sex chromosomes, and these are the approximate locations of the DNA loci that are all analyzed in the German system. I'm not sure whether the Turkish crime system analyzes exactly the same. So within the EU, this is actually a consensus set of loci that is analyzed. 16 such like loci. They have complicated names, but the system is simple. This would be D1 as and the number. So D1 means DNA on chromosome 1, DNA locus on chromosome 1, somewhere here, D2. So on chromosome 2, we have 2 loci, on D3, D18, chromosome 19, D19, etc. And there are some loci which have non-systematic names, which are, well, historically justified. So these are the loci that we analyze, and we analyze those loci because they are highly variable. So there are many differences in those loci between different individuals. The class of loci is short tandem repeat, or STR loci. Now, what are these? Let's highlight the D1 as 1656 in a bit more detail, not much more detail. Here we have 2 alleles in that example, allele 9 and allele 12. So this would be a heterozygous individual, and as indicated by that orange area, the one allele is a bit shorter than the other. So it's a size difference. So these are actually length polymorphisms. And if we look into a bit more detail, we notice that actually what is making the size difference is the number of such like repeat units. So this marker has 4 bases that are repeated 9 times, THCA, 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 etc., 9 times. And on the other chromosome, the very same locus has 12 such like repeat units, which makes a size difference of 3 repeat units, which is 12 bases. So in another individual, we may have other numbers. So maybe another individual would have here 11 and here 13, and yet another individual may have at the same location twice 14 repeat units, might be homozygous then. It's a length polymorphism, and we analyze this simply using PCR. So if we apply PCR primers here and here and here and here in the flanking part, which is conserved, then of course the PCR product here is shorter than from that chromosome. And shown here once again, so the size difference of the PCR product, which goes from here to here, is 3 times 4 bases, namely the 3 additional repeat units we have included here. So 12 base difference. How can we resolve this difference? We use capillary electrophoresis. So basically some gel-like matrix in a, say, 50 centimeter long capillary. We load the sample here, we separate it along the capillary, and when it reaches a detector, we just record there's a signal. How can we record? We record a fluorescence because one of the 2 primers is covalently linked to a fluorescent dye. And this is what is recorded. So there's a laser and some sort of a CCD camera, and then we can record the yellow fluorescent signal here. And what we get are 2 peaks in the output, which is called an electropherogram. So it's actually like a gel electrophoresis, but it's not a gel, it's some sort of an electropherogram that we get. Peak 1 is the first one, the early peak, representing the allele number 9 with the 9 repeat units, so the short one here. And somewhat later comes the second peak, representing allele 12. And it's quite convenient in the human system that these numbers that we use to designate the alleles is just the numbers of the repeat units. It's a very simple system. And of course, if we analyze DNA from another individual, as I just already mentioned, the pattern may look different. So a second individual may have here allele 10, so 10 repeat units on the one and 11 on the other or so. We do not just analyze one locus, we do PCR amplification of all of them together in a multiplex PCR reaction and separate them simultaneously. What we get out here is then something like that. So in that color channel, I would call it now, we have the one locus. All peaks falling into that size class would be assigned to that locus, but we analyze in that example 3 others as well in parallel and in the same run, basically, the D18, which would reside here. Here we would, in that example, just see 1 peak, so homozygous, and these 2 other loci. Above here, we have the size of the fragments in base pairs. This would be the fluorescence intensity, which we measure in relative fluorescence units, RFUs. And the important thing here is that the size ranges of the amplicons of the PCR products must not overlap. Why? If here we would have an overlap, if the D18 would overlap with that range of that, then we would not be sure whether this peak is now belonging to that marker or to that marker. So in order to be able to interpret such an electropherogram, we must design our PCR assays in such a way that the size classes of the fragments fall in, do not show overlap. Of course, the more loci we use, the more base pairs we would need, which is inconvenient. This is why we do a simple thing. We use different colors. So the primers, for instance, for those loci are not labeled with a yellow fluorescent dye, but with a green one. And then we have a second color channel, which just runs in parallel. So, and then you may notice, for instance, here, these two peaks have the very same size, but we can tell them apart because this one is green, and this one has been generated with the primer labeled with a yellow fluorescent dye. So we immediately know this is the D21S11 locus and not the D18 locus. That's how the system works. And in fact, in reality, we use 4 different colors. There are systems nowadays that even use 5 colors. And by that means, we can actually simultaneously analyze all these PCR products. There are 2 more, which I forgot to mention, which are on the sex chromosome. So we can also identify whether the DNA is derived from a genetically male or chromosomally male person or female person. Now that is an example now of a real DNA profile, which you may understand now. So here you see the different color channels, the 4 color channels, the blue one, the green one, that is the yellow one, which we show in black, because otherwise there's too little contrast in order to see it properly on a printout or on a screen, and the red channel. And let's focus maybe on the green one. Up here, you see the bases, so the fragment length in base pairs or in bases, so 360 here, 80 here, and this would be the fluorescence intensity in some arbitrary fluorescence units. And for instance, and this gray rectangle, this is the size range into which the D10 locus would fall. So all fragments that we amplify with those primers are expected somewhere here. So those two peaks which we see here belong to that marker, and they are automatically assigned by the software as allele 13 and allele 15. This assignment, this automatic assignment, we call allele calling. So these are the alleles are called, and there is a certain analytical threshold such that these tiny peaks, which you may see here as well, they are not called. So we just see the really important ones, basically. So this is heterozygous. These 2 peaks we notice, these belong to the D1 marker. D1 is 1656. Heterozygous, and here we just have 1 peak. So here we have a homozygous genotype, and the same thing we can do for all the others. And if we just write down all these allele numbers and in conjunction with the DNA locus, that is what we get. Then that is a DNA profile. It's just a numerical information where we just collect all this. And once we have done this, we can forget all the analytics that we have done before. It's not always as straightforward as shown in that example. Sometimes the electropherograms look a bit messy, but basically that's what we have in the end. And if we have such a profile with numerical information, of course we can feed this information into a database, and we can also use it for a database search. That's why the system is so powerful. And the other thing why it is so powerful, I forgot to mention, is because there are so many variants in the population. So each of these gray bars, for instance, here depicts a possible variant that we can find in the population. So there are many, and that means there are many combinations. There are many possible combinations for all of these different loci. And if we combine all these combinations, we get astronomical numbers of options that we have. So each profile is actually statistically a unique one, but I don't want to go into that. I just wanted to explain very shortly. Now, let's come back to the single shade hairs. Why are they a problem? I may already mentioned the DNA is degraded and there are very low DNA amounts. Of course, there's also one technical obstacle. This is the PhD student, Sarah Hess, who did most of the work which I'm presenting today. And we have to work in some sort of a protected area, in a DNA-free area. So it's not just free working in the labs. We have to take precautions in order to avoid contaminations because we have so low DNA amounts and we have to apply very sensitive techniques. Now, this is the technical side, but now we go to the analytical side. So degraded DNA and very low DNA amounts, what's the problem? And I try to explain that using that slide, which is somehow similar to what we saw before. Just I highlight here the DNA with each of these strands is a strand of DNA. Of course, in a DNA extracted from the trace, we have thousands of such like strands, of course. And if the DNA is intact and there is much DNA, enough DNA, then each locus that we analyze is present in many, many copies. That means whether we analyze a short amplicon, like that marker here, or a longer one, maybe that marker here or that here, we will succeed, and we have balanced profiles. More or less similar peak heights, nothing is missing. Now, what happens if the DNA degrades? The DNA degradation means random gaps, double strand breaks or pieces are missing. If we just now statistically distribute such like random gaps now in the drawing, just write by hand somehow, then you may notice that there's a much higher chance that there will be an interruption or disruption at the longer amplicon. I highlight it like that, then you can see it. So here are many, many gaps, and each gap means if one primer sits here and the other here, means we cannot do the PCR. We cannot amplify, because for PCR amplification, we need an intact stretch. So the chance that DNA is affected increases with the length that we analyze. So the shorter amplicon is almost not affected, so still remains the same, but the longer ones, the longer amplicons, they get lost. We call this an allele dropout. So alleles are missing. We do not see the peaks any longer, or also a peak imbalance, which is also not nice because this is then difficult to interpret. This is what happens during DNA degradation. So if you have DNA degraded, even if it's a lot of DNA, we get somehow those effects. So the longer amplicons are more likely to be affected. How about now the second challenge posed by the hairs, the low copy number DNA. If you have only little DNA, say 33 picograms, well, what is a picogram? One cell, the DNA content of one cell is 6.6 picogram. Chromosomal DNA. So 33 picograms is less than 5 cells, so to speak, or 5 cells or less than 5 cells. That means if we have so little DNA, the chance is quite high that for stochastic reasons, not all chromosomes or chromosomal parts are present. So stochastically, we call this a sampling error. Stochastically, just by chance, some loci may be underrepresented or even absent. In that arbitrary example, for instance, from that locus, we have only the 2 chromosomes containing the allele 10, but there's not the copies of the homologous chromosomes containing the other alleles are not present. That means we have an allele dropout. And in that case, it's not always the longer alleles that drop out, it's just any allele can drop out. Same here, or here, for instance, we have, for that locus, we might have the chromosome bearing the allele 28 twice, and the chromosome bearing the allele 20 only once in our mixture. So just statistically, that means we get an imbalance, so a small peak here and a big peak here also. And there's one other problem depending on the analytical strategy. I mentioned to you with PCR, we can do more PCR cycles to increase sensitivity. The risk is then that we pick up some unrelated signals, which we call allele drop-ins. So this is the problem of low copy number DNA. Sorry, I'll show you now some examples from single hairs from our analysis. So for some hairs, we can even get a complete profile. Looks like here. So again, you understand now how to read that profile. So here we have the different markers. Here we have the peaks, and you may notice that actually it's a complete profile. All the markers have 2 peaks. Well, that is a homozygous. We know the genotype of the donor, of course, so it's a complete profile. But what you may already notice is, although it's a complete profile, the longer amplicons, the longer fragments are already very small in fluorescence intensity, which points to degradation. So although we have a complete profile, we can see the DNA degradation here. What is a complete profile? Which is nice. So some hairs can yield complete profiles, but only rarely. More often we see partial profiles like that. Here, for instance, we have some alleles still called here, but here nothing is called. Here just one locus, one allele is called. So it's a very incomplete profile. Basically, we have noise on the baseline. What you also may notice if you compare the y-axis, the fluorescence intensities, here we are in the hundreds. I just go back to the previous slide. Here we are in thousands. So the intensity also decreases. So this is quite typical for such a hair. And of course, it's such a profile would not help in forensics. And in other cases, and they are even more severe, we have a complete failure. So just noise on the baselines of all color channels, except here we have one tiny little peak, but we do not know, of course, whether that's a complete profile, so whether the individual is homozygous or heterozygous here. So useless. We do not get the information. Now the task will be to identify beforehand those hairs which are promising, from which we can expect a good profile. How can we do that? So our strategy was actually to look at the DNA. So extract the DNA and then check before doing a DNA profiles with a relatively cheap method, the DNA amount and the DNA degradation status, or how we call that, the DNA integrity. And how can we do this? How can we measure DNA amounts and DNA integrity of low copy number DNA? The answer is that the normal lab methods that we use in molecular biology labs, such as a photometer or fluorometer, actually are not sensitive enough in many cases. So the DNA amounts are too low. We do not get information on source of the DNA. So there might be a microbial contamination. We would not see it. We would just measure DNA. And we cannot get information on the status of DNA. Is it degraded or not? And also we do not get information whether there are compounds in our extract that would inhibit PCR. This is why in forensics, quantitative PCR or real-time PCR is the method of choice, because it's PCR-based, so it's very sensitive. We can, by designing species-specific primers, we can find out how much human DNA is in a sample. We can, as I show you in a minute, get information on the DNA degradation, and we can get information on whether we can PCR amplify our DNA at all. For the students among you who are not too familiar with qPCR, I included some slides explaining the basic principle. I hope that I will not bore the majority of you with that. So how does it work? Basically, quantitative PCR, qPCR, or real-time PCR is based on fluorescence-based measurement of the PCR product accumulation in real time after each cycle. So after each cycle, we measure how much DNA has been produced in our PCR. So we start maybe with one template molecules, and after the several cycles, we have an increase in number. We detect those PCR products using fluorescent probes, which bind to the DNA more or less specifically. We excite the fluorescence using a light source, and we record or detect the emitted light after each PCR cycle. And the fluorescence options are several. One can just use a DNA binding fluorescent dye, such like cybergreen. This works similar to ethidium bromide, which you may know from the lab. Or we can use nucleic acid probes that are complementary, such as Tachman probes or Scorpion primers. But the principle is always the same. So we measure the amount of product by recording fluorescence produced after each cycle. And then we get a curve like that. Here on the y-axis, the fluorescence intensity, and on the x-axis, we plot the number of cycles. And after each cycle, we get some light signal. And after some time, it crosses our analytical baseline, so to speak. And then all of a sudden, we get stronger signals. PCR is working, we notice, and after a while, we reach what we call the plateau phase. No longer amplification possible, no increase in product. And of course, we interpolate that, or the machine does this for us, and we have this nice curve. So this is an amplification plot where we can distinguish the exponential phase here, the linear phase, and the plateau phase, which you may know from your molecular biology lectures. The idea of quantitation is now to ask, to define a certain threshold in fluorescence and ask how many cycles do we need to reach that threshold. That's what we do here. So we draw a threshold line, usually through the exponential phase. This would be the fluorescence intensity, and we just count the cycles that we need to reach the threshold. This is the CT value, the cycle threshold. And if you have a second DNA sample containing a lower amount of DNA, fewer copies, then of course we need more cycles in order to reach the threshold. That means the CT value of that will be higher. And in the times of COVID pandemic and PCR testing, even the man in the street knows that a lower CT value means more virus, and a higher CT value means less virus. So it's the opposite of what one might think. And we can quantitate the copies by in parallel analyzing standard samples, plotting their CT values against the logarithm of the DNA concentration. And if we have done that, this is then a linear relationship. Ideally, we can then use this CT value, go up here, and then we can read the DNA amount over here in the y-axis. That's how quantitation works. Now the question is, how can we use this not just to quantitate DNA, but how can we now measure DNA degradation using such an assay? And the answer is quite simple, because actually you know it already. We do the very same thing that we saw as an obstacle in the DNA, in the forensic DNA typing. We analyze two different amplicons, a short one and a long one. And if the DNA is intact, the PCR will work equally well for both of them. The long fragment will be amplified as well as the short as well as the long fragment. So these are the measured copies that we quantitated using qPCR. So the ratio long divided by short would be 1. But if the DNA is degraded, as we saw before, then the long amplicon will no longer be efficiently amplified. So we do not record as many intact copies here as from here. So here we have less, and we can define a ratio, and that ratio long divided by short will be lower than 1 for degraded DNA, and the more degraded, the lower it will be. So basically we use a very short amplicon, which serves for quantification because it's only slightly affected, and the longer amplicon is degradation sensitive. That's the idea. Now there's a second thing that we have to think about. This is sensitivity. If we analyze DNA that is in the picogram range, we need to be highly sensitive. If we would use a standard template, some gene on some chromosome, we have actually just 2 copies per cell. That would be, again, the chromosome set. We would have just 2 copies. And of course, we need many copies in order to have a considerable signal. So we have only weak signals here. And if we have stochastic effects because we work with low copy number DNA, maybe our template is even absent or reduced in amount and no longer reflecting the true amount of DNA. So this is a problem. What we can do, we can analyze multi-copy targets. So genes or DNA loci that are present in many copies are distributed across the chromosomes. And there are several options. There are some transposons that can be used. There are the ALU sequences you may have heard of, which come in millions of copies in the human genome. Things like that can be used and have been used. The idea is then we get a much stronger signal even from a single cell. So we have a very high sensitivity, and the system is less affected by stochastic effects, so by stochastic losses of individual chromosomes or chromosomal parts. So the idea is to analyze multi-copy genes and a long and a short amplicon. Now there are commercial kits that are nowadays used in forensics. I just list 4 of them from different companies. They have different names. Very often there's a Quanti in the name, Quantifiler, Quantiplex, PowerQuant, InnoQuant, etc. So I don't take care on the names and on the companies. I don't want to make an advertisement, but they all work in, following the same principle. They analyze a small amplicon of a multi-copy DNA locus, different sizes, different loci, and the large one. And by the ratio, they determine the degradation. And the sensitivity is 1 picogram and even lower of DNA. Once again, 1 human cell contains 6.6 picogram of genomic DNA. So we need less than a cell in order to reliably analyze DNA. They have also some additional features. So they analyze also Y chromosome specific sequences. So we can analyze the presence of a male component, and they analyze what we call PCR inhibition, so presence of compounds that would interfere with PCR-based analysis. When we started with our single hair project in 2014, those kits were not yet on the market. So we decided we establish our own system. And of course, the idea was basically the same. But we analyzed ribosomal DNA sequences. This is why we call the human system RiboD, so ribosomal DNA and degradation, RiboD. And we designed a very similar system for analyzing dog hairs. This we called RiboDog. And we have small amplicons. The RiboD, the small amplicon is 67 base pairs. The large amplicon is 362 base pairs. And for the RiboDog, these are a bit different ones, different primers, etc. Our sensitivity, as I will show you in a minute, is again 1 picogram. So very comparable to what the companies put onto the market sometime later. So we have our own system. Why did we now use ribosomal DNA sequences? The answer is here. The ribosomal DNA sequences are present in multiple copies in our genome, roughly 400 copies per cell. Many, many copies. Why is that so? That is so because the ribosomal RNAs that are produced by those genes, the 28S RNA, the 5.8S ribosomal RNA, and the 18S ribosomal RNA, as well as the 5S ribosomal RNA, they're components of the ribosome. And our cells need many ribosomes in order to maintain protein biosynthesis. This is why there's a high demand on transcription, production of these RNAs. This is why we have so many copies of the genes, because then we can produce lots of RNAs. This is the idea. Now, strange enough, the 5S is present on only one chromosome, namely on chromosome 1. Multi-copies, one after the other, arranged here, roughly 400 copies. And the other members, the 28S, 5.8S, and 18.8S, their genes are located on 5 chromosomes. So in a diploid cell, actually, these are 10 chromosomes, chromosome 13, 14, 15, 21, and 22. And the genes are arranged in such a tandem array. So we have here an intergenic spacer, IGS, sometimes called, size 30 kilobases, 30,000 bases, followed by the ribosomal RNA genes, 13 kilobases approximately, and one after the other. So always the same array, one after the other per chromosome, 40, 50 copies or so, depending. It's not entirely has not yet been mapped perfectly, actually, because such like repetitive regions are difficult to analyze by sequencing approaches. Now, if we blow up that part here, then again we see here the intergenic spacer, and here we see now the 3 genes, the 18S, the 5.S gene, and the 28S gene. They are transcribed somewhere from here. Here would be the promoter region, regulating transcription of such a unit. And then it's first transcribed as one RNA, which is later on processed to cleaved into the individual parts. These parts are highly conserved, also across species. This is why we did not design primers amplifying within here. We chose the promoter region, which is sufficiently well conserved within the human species, but not, for instance, between humans and dogs or humans and cats. So I blow up that slightly. So that region, that would be what I call the promoter. And in that region, we have a long amplicon of 362 base pairs in the human system and the 67 base pair amplicon, short amplicon. These are the PCR products that we analyze in order to quantitate DNA and in order to somehow get an idea, not really quantitate, but estimate the degree of degradation. And here are results. So that's what we actually did once we have established the primers and have shown and seen that they work properly well, that the PCR efficiency is reasonable. Then we used DNA from HeLa cells, so human cancer cell line, and digested this DNA with a DNase-1 enzyme over prolonged time periods, and then we took out samples after 5 minutes, 30 minutes, 120 minutes, up to 720 20 minutes. And if we separate these samples on an agarose shell, here would be a lambda Hindi 3 marker, here 100 base pair DNA letter as size standard. We'll notice that the non-digested HeLa DNA somehow is a big blob of a bit over 20 kilobases. And if we digest the DNA, we get a smear down here. And the fragments get smaller and smaller and smaller. So we degrade our DNA. Let's degrade DNA, how it presents on a shell. And if we then use these at different dilutions, these DNAs, to test the system, we get the result shown here in that diagram here. Let's maybe focus on 1 nanogram DNA. If we use 1 nanogram DNA, the short amplicon, these are the orange bars, 5 minutes degradation, 30 minutes degradation, etc. So that would be the quantity of DNA that we quantitated using this assay. And in the first time, not much changes for the short amplicon for the short fragment as to be expected. Later on, it will also be slightly affected, but strongly only at the very end. The green, the degradation sensitive long amplicon, is affected right from the start. Please bear in mind, this is a logarithmic scale here. So this makes it already here twofold difference. Unfortunately, the ratios are somehow dependent on the DNA concentration, but that's just how it is. So for 100 picograms, for instance, it looks like that. Again, the orange bars, they do not change a lot, but the green bars, they are sensitive to the degradation. 10 picogram, it's a little more than 1 cell now, the very same behavior. So the orange bar remains the same. And also if you go over there, you see we quantitate almost exactly 10 picogram. That's fine. So it works in that range. And the green bars, so the amplification of the long fragment starts to become affected at later time points. And even with 1 picogram, we see in tendency the very same thing. So the longer fragments, shorter fragments are less affected than the longer. Here are some irregularities, but of course with 1 picogram, this is not DNA from not even 1 cell. Of course, we have stochastic effects and we do not know which parts of which chromosome is present in which sample. But basically it works and we can quantitate the DNA even for 1 picogram more or less reliably, and we can measure somehow still degradation. We can plot that. We can make a ratio, define a ratio which we call a D value, a degradation value, the quantity of the long fragments divided by the quantity of the short, and then we get such like curves. Here we have again the same time points in minutes, digestion durations. What you see here is actually, so it declines in that way. So we can use these D values somehow, if we know the DNA concentration, to assess the degradation or the integrity, to say it in the opposite way. We apply this then to hairs, and this is what I show you here. So this is our single hairs. We used the first 2 centimeters and digested them. We optimized the DNA extraction protocol. And then we have hairs with lots of DNA and hairs with little DNA. And we have quite different degradation values as shown here. So this is almost not degraded, so ratio is almost 1. And here we have a strong degradation, 0.003, so much less of the long fragment can be amplified. And if you look at the morphology, we collected the hairs using a comp. That means also those telogen hairs which were still somehow loosely inserted in the skin may have been recovered, and they very often have a little hood or nipple-like region of more or less dead tissue, cellular tissue, but most of it is dead. And the presence of that nipple actually does not tell us anything about the degradation ratio. Here, for instance, we have a strong degradation, and here we have only a moderate degradation. So these are just the hairs that we analyzed. And if we analyze them now using our qPCR and do subsequently a DNA profiling, and we just count the alleles that we can recover, so all alleles 100%, or less alleles, 1 allele missing 97%, and plot this here. This would be the allele recovery. So the success of the DNA profile. Basically, we see that our collection of hairs looks quite variable. Some hairs worked, some not. And if here we plot the D values, the degradation values on top, again, it's a logarithmic scale. So 1 and 0.1 here already. We will notice that if we have enough DNA, actually the DNA degradation is not so important. But for instance, here we have 500 picograms approximately here as well, here 5 and 4 and here 5. And here we have degradation, but not a too strong degradation. We have still 100% of alleles, but here with a strong degradation, only 77% of alleles. And if we go for the next pair, just to get an impression, we see again, we have same amount of DNA, so 300 something picograms, but here we have a moderate degradation, and here we have already a strong degradation, 0.067 as a D value. And we notice that here from an almost complete profile, with just one peak missing, we miss here half of our peaks. And so for the others. So if we have sufficient amounts of DNA, then the D value is very important. But if we have low amounts of DNA, so in the 20 picogram range, etc., then I think the DNA degradation still plays a role, but most likely the profiles will be incomplete irrespective of degradation values. So bottom line, if we have a hair and we analyze it and we get some D value in that range and a reasonable DNA concentration, or like here, then it makes sense to do a DNA profiling. If we have a high DNA degradation, like here or like here in that hair, although we have, in forensic terms, lots of DNA, 200 picograms, it makes no sense to analyze that hair. So this is how this type of analysis can actually support the forensic DNA typing of single hairs. Now I see I'm running out of time. I will conclude now by, in the end, discussing dog hairs. We did the very same thing for dog hairs using our RiboDog assay. These are the three suspects, Kimi, Motte, and Mowgli. Now to dog hairs, I would just like to explain to you that they have two types of hairs. They have guard hairs, they are longer, they are stronger, they probably provide some mechanical protection, and they have undercoat hairs, which are fine and thin, and one purpose might be thermal insulation. The dog hairs I already mentioned in the beginning, they look different, and these are the hairs that we analyze. So the K hairs are from Kimi, the M hairs are from Motte, and the G hairs are from Mowgli. That's what we analyzed. So actually those hairs do not contain much DNA, so mainly we get profiles from guard hairs. These are the hairs that make sense. And this is what you may see here. It's a very similar way of presenting the data as you saw before. Now the great surprise was that dog hairs do display only very little degradation. It's very different from human hairs. So the degradation ratios are all in the 0.5 or above. There are only some exceptions here and there. And the allele recovery, when we do DNA profiling of dogs, this can be done using different primers, analyzing different loci, very similar to the human system. It's not as elaborated as the human systems, but it works. Then we get almost complete profiles for most of the hairs. So dog hairs are much more promising sources of DNA in terms of forensic DNA profiling, and they seem to retain much more undegraded DNA. The biological meaning, I have no idea. I think, I noticed I took a bit more time than I thought. I think I skipped the NGS part. Is that okay with you, or should we spend some more time? What does the audience say, the organizers?
It doesn't really matter for us, but we also have a question regarding that topic. Maybe you could explain through the question. Okay, then I skip that. Maybe there will be some time during the discussion. I will be more than happy to show this data. Just let me summarize what we were discussing. So we were discussing DNA profiles that they are based on PCR amplification of STR loci. Single shed hairs are posing a challenge because they contain low amounts of DNA that is strongly degraded. We developed a qPCR-based assay for the analysis of DNA amount and degradation, and we analyzed ribosomal DNA sequences for that. That are present in many copies per cell.
And we noticed that in human hairs, the DNA typing success depended on amount and integrity of DNA, whereas dog hairs are strange enough different. They display less degradation, and the typing success mainly depends on DNA amount. There are many people who contributed to this project. Main person I already mentioned was Sarah, a PhD student who finished the PhD in the meantime, who did the human hair analysis and also part of the dog hair analysis, and Salvatore Trapani, a master's student from Palermo, who established the RiboDoc system. And there are some other persons in the lab who helped in the analysis. Also for the NGS analysis, which I did not show now, we have, of course, contributors from our Department of Informatics. And we have some collaborations with the Institute of Legal Medicine in Bonn and with the company Eurofins. They did the dog DNA profiling for us. Burkhard Rolf is the man here. And we have, these are the two funding sources which we had. And I would like to thank you now in the end for your attention. And of course, I'm happy to receive some questions.
Thank you so much, sir, for your speech. We have some questions.
Please.
First question is from our audience. Looking at your recent work, we found that you're working on the DNA sequencing of the next generation. She wonders if you can share with us ways you follow in determining this, sir.
Okay, let's then I go back to a few slides in order to explain. Yeah. So actually, we started using next generation sequencing in order to improve the DNA typing success of single hairs. And the idea is the following. If we determine the sequence of the very same STR loci and not just the length, well, the sequence information contains the length information as well. That's the number of bases. So we can do a DNA typing using sequencing approach, and the sequencing approach that can be used in for so many loci to analyze them simultaneously is next generation sequencing. Next generation sequencing means being able to determine sequences of a mixture, because each individual molecule will be sequenced essentially separately. This is what next generation sequencing actually means. So first we do a PCR of the very same markers, and then we sequence them. Let's look at this here. That would be a conventional analysis. Here we have again base pairs. These are the three markers which we look at. And for instance, the FGA marker. Normally we see peaks. If we analyze by sequencing, we get a sequence. This marker has these repeat units of CTT, CTTT, and many times for that peak, and then for another number of times for the longer peaks. These are the alleles. And the important thing is that by just sequencing, of course, we can determine the number of repeat units. And this is why we can determine the allele, as we would do using the capillary electrophoresis. But now we do it by sequencing. One advantage is we get additional polymorphisms that would not change the length, such as SNPs, which can reside in the repeat units sometimes. So in the population, some people have different bases sometimes here, or can be present in the flanking regions between repeat units and primer binding sites. So we get additional information for some of the markers, and this can be very useful because it can improve the discriminatory power of the system. And this can be important if we have DNA mixtures, because then we can assign a particular allele to a particular individual. And this can also be very valuable if we have incomplete profiles, because we get a bit more information then. But the other thing, this is why we find this for our research question, it was of advantage, is that when we determine the allele number by sequence, and we identify the loci by sequencing them, we are no longer dependent on separate size ranges. So all the size ranges now can overlap. It's no problem for us. And we can create small amplicons for all the markers. That means no longer big amplicons, which would be affected by DNA degradation. So, and we can even analyze small loci. So what we did, again, we were almost ahead of time. Well, the commercial systems from Illumina just came out at the same time when we wanted to do our research, so we developed our own system. And by doing that, we learned a lot about sequencing using NGS. These are the size ranges of the different loci. These are the 16 loci that we analyze conventionally. Now, these are the size ranges when we analyze them using PCR and next generation sequencing. The colors are meaningless, it's just for illustration. But what you may notice is the size ranges of all loci are smaller than 300 bases, and most of them overlap, which would not be possible if we would analyze using the capillary electrophoresis. And we do now the sequencing-based analysis of them. And DNA profile then looks a bit different. We do not have peaks, we have some sort of bars. This is now what our bioinformaticians did for us. They programmed a pipeline for automatically assign allele numbers to the sequences to sort out everything, which is very difficult because it's a wealth of data that we get. And on the y-axis, we see now the number of reads, not the fluorescence units, the number of reads. So how often have we sequenced that particular sequence? And on the x-axis, the assigned allele numbers that have been identified by the bioinformatics pipeline. So we here, for instance, have those two peaks as the main peak. So that would be a heterozygous profile, just as an example. That's how these data look like when we analyze them. And if we apply this now to the single hairs, this is just what I showed you before. So the DNA typing success that we saw before of the very same hairs. If we do the very same thing with next generation sequencing, we have much better allele recovery for most of the samples. This now sounds like a very huge improvement and it's very promising, but there's one drawback, and that drawback is our system. Oh, I shifted a bit. Our system is so sensitive, and also for some reasons of the bioinformatics pipeline, we have some peaks that have not been present in the original sample. So additional peaks, ADIs, false peaks. And this is of course not nice. So we are working on that now in order to improve that situation. That's the price we maybe pay, for our increased sensitivity here, false alleles. And in forensic terms, this is actually very problematic because a false peak points to the wrong individual. So we are working on that. But basically, we get, as you may appreciate here, many more correctly called alleles. Unfortunately, considerable number of wrongly called as well, in addition. I hope that answered that question.
Thank you so much, sir. We have one additional question. There is some early work of the 3D face remodeling derived from the genetic material of suspects. What is the current situation and how does its future look like?
What do you mean with 3D modeling? You mean some sort of a photo fit image, or what is the question? Can you say it again?
There is some early work of 3D face remodeling.
I see. Mm-hmm.
Derived from the genetic material of suspects. What is the current situation and how does the future look like?
Yeah. The current situation is that it is more artwork than science at present. So there are some markers that can be analyzed, for instance, for pigmentation, so skin color, eye color, hair color, things like that. Also, we can more or less precisely determine the age of DNA donor, but other features cannot yet be reliably reconstructed from genetic information, such as the length of the nose or things like that, or the chin or such like features. There is one, what's her name? There is one artist actually either in the UK or in the United States. I forgot her name. She did this as some sort of an artwork, and she creates some phantom photo fit images of DNA traces, which she just collected on the street from some cigarette butts or so. And actually, it's a nice work, but most faces actually look very similar, I should say. So what there is, what we can do is pigmentation of hairs. So pigmentation genes, the pigmentation is genetically very well understood. And one can use pigmentation genes, so polymorphisms, SNPs, SNPs in pigmentation genes in order to get some ideas. Does not work very well for some intermediate eye colors, things like that. But for the brown color, it works quite well, for instance.
Thank you so much, sir. And lastly, we have one last question. In one of your publications, there was a question stating, will you be a victim? He was wondering if the possibility of this event happening can be inferred from the current data that are available to us.
Which, can you say it again, please? I didn't fully understand the question.
In one of your publications, there was a question stating, will you be a victim? I was wondering if the possibility of this event happening can be inferred from the current data that are available to us.
Where you will be a victim. Sorry, I cannot make much sense out of the question. I apologize for that. Maybe the one who asked the question can clarify what she or he is meaning.
If there will be any feedback from the one who asked the question, then I will be able to conduct you in the breakout room.
Yeah, thank you.
Thank you for your speech. It was phenomenal.
Thank you very much for your attention.
We'll be waiting you in the breakout room.
Thanks. So you need to transfer me. I will close my presentation now here. And then.
Dr. Jaeger, you're expected to join the breakout room. You can find the entry button between the record and reactions bar at the bottom of your screen.
Actually, I don't see
it. Do you see the share screen button?
Yes.
Or the icon there? All right, it should be 2 icons right of it, breakout rooms.
Ah, I see.
Can you
see?
All
now
right.
I see. Okay, fine. Thank you very much.
All right,
I will join now.
Auf Wiedersehen.
Wiedersehen.
Our next speaker is Assistant Professor Abdülkerim Eroğlu, who is a researcher from North Carolina State University, focused on the impact of dietary carotenoids. Today, he will inform us on secreting beta-carotene in mammalian gut by enhancing beta-carotene secretion in yeast, while it is residing in the mammalian gut using different strategies such as CRISPR-mediated genome editing in a speech titled Engineering Beta-Carotene Secretion in Mammalian Gut by Probiotic Yeast. Thank you so much for accepting our invitation and welcome to our Congress. If any of our participants have any questions, they may write it in the YouTube live stream chat or through Instagram DM. I assume everybody checked your microphone and whether your screen presentation is working.
Okay, well, thank you so much for the kind invitation. I'm very pleased to be here sharing our findings with the participants. Is everyone able to see the slides? share screen, sorry, right there. Okay,
Yeah, it's good, sir.
just a sec. Let me go back, share screen. Okay, is everyone able to see the screen? I mean, the slide, first slide?
Yes, it's good.
Okay, well, again, I'm very pleased to be here, and the title of my talk is Engineering Beta-Carotene Secretion in the Mammalian Gut by Probiotic Yeast. So, my name is Kerim Eroglu, and I'm an assistant professor in nutrigenomics at the Department of Biochemistry, North Carolina State University. So on this slide, you see the representative picture of yeast and the chemical structure of beta-carotene. Let's start with some background information. On the right, you see the cartoon of gut microbiome. A special credit to Nature Outlook, which was published several years ago. You see, we are navigating these we are a holobiont. We are living with multiple microorganisms such as bacteria, viruses, fungi, and others, collectively all known as the microbiome. As you probably have heard, gut microbiota and gut microbiome terms in this past decade or so, more emphasis placed on gut bacteria. Fungal population is has been utilized or not been well studied, although they are also comprising about 10% of the total gut microbial mass. And our interest is lying on the engineering commensals, engineering microorganisms, and most to date, most engineered microbial organisms are bacteria. So that's the point that we are, from an in situ biomanufacturing perspective, fungi or yeast are desirable because they are not susceptible to antibiotics like bacteria. And moreover, several fungi are considered safe from health perspective, and since they can be used as probiotics. As I said, they are considered generally as safe, and they can be used as utilized as probiotics. And more specifically, we are interested. There are multiple probiotics in the market. More specifically, we are interested in Saccharomyces boulardii, which is a cousin of Baker's yeast, Saccharomyces cerevisiae. And the reason that we are very interested in SP or Saccharomyces boulardii is they grow better at human body temperature and they are highly pH resistant. So on here, what you see, SP supplement on the market. There are so many supplements on the market. I'm just showing one of them. And what you see on the front is some information regarding their ability to improve digestion and gut health. And on the back, you see supplement facts like they are in terms of colony forming units and other serving suggestions. So why we are into, what's the motivation? So we are interested in SP or Saccharomyces boulardii, which is a probiotic yeast. And probiotic yeast can be engineered for biomedical applications. And as I mentioned on the previous slide, yeast can be used as an in situ biomanufacturing purposes. And the main research gap that we are trying to fill on this study is to utilize these yeast for in situ biomanufacturing to produce small molecules like beta carotene. So to enable balanced high metabolic flux through small molecule biosynthesis pathway, we have to understand how to utilize finely tuned gene expression. So that's sort of like keyword. So I want you to understand finely tuned gene expression is absolutely necessary to enable synthesis of biomolecules, finely tuned gene expression. So therefore, in this work, we sought to elucidate the genetic strategies governing tunable biomolecule production in Saccharomyces boulardii, or SP. From now on, I will call it SP, Saccharomyces boulardii, a probiotic is that we are using in this, that we are studying in this project. And so there are several objectives that we developed. One is engineer SP to deliver biomolecules to the large intestine. That's hence it's called in situ to site. Then we will define the genetic engineering strategies for the tunable biomolecule production in this yeast. So, and our central hypothesis is we first hypothesized that SC, or Baker's yeast, since it's cousin of SP, the promoter strengths that are used in SC will exhibit similar levels of gene expression when implemented in either SC or SP due to the genetic relatedness between SC, Saccharomyces cerevisiae, and SP. So again, the keyword is tuning, how to tune gene expression. So when I started at NC State almost 4 years ago now, I was really not familiar with synthetic biology. In these last 2-3 years, with the collaboration from Department of Chemical and Biomolecular Engineering, I learned a great deal of synthetic biology, and I'm still fascinated the tools that we can use to construct for genetic engineering purposes. So synthetic biology is really placing a key role in designing genetic promoters. So what we did, we first began by performing the most comprehensive analysis of engineered gene expression to date in SP. So we asked whether the tools that are being used in SC can be applicable to SP. So what you see over here, normalized fluorescence values of different promoters. So we employ promoter engineering techniques to understand whether the promoters that we design can be used in SP as well. So at the end, we used a model fluorescent protein to facilitate gene expression measurements, since these exhibit a high signal to noise ratio. So going back, the height of these bars represent the mean fluorescent value of 3 biological replicates. You see the biological replicates, those are the black dots, and the mean of each replicates are shown in different colors. Okay, so how to tune gene expression? So next, we generated expression cassettes containing different markers. One is called oxytrophic, and the other one is called antifungal. So what are these? Oxytrophic markers are being used very extensively in yeast genetics, where they are used to select cells that have been successfully transformed with recombinant DNA. So just to give you a little bit background, a wild-type yeast cell that synthesizes its own nutritional requirement is called a protroph. while its mutant counterpart, which loses the ability to synthesize an essential nutrient due to a DNA mutation, it's called auxotrophic, autotroph. So these mutant, nutritional requirements is used to distinguish between recombinant DNA and wild-type DNA. Hence, we use antifungal markers and auxotrophic markers to test whether our strategies are working efficiently. And also, we used two different origins of replication to design and engineer promoters in SC. I'm sorry, in SP, in Saccharomyces boulardii. So what you see, a combination of auxotropic markers. So these are all different auxotropic markers, TRP. And by the way, these are very commonly used. TRP1 is a frequently used auxotropic marker for genetic modifications, and other commonly used HIS3 and URAT3. So we use these common auxotropic markers to test whether genetic modifications are feasible in SP. We also use several antifungal markers shown in here at different doses in a combination of two different origins of replication. So all in all, this slide tells us that auxotropic markers are very efficient in generating synthetic promoters that we utilized in this study. So we achieved a maximum fluorescence in using URAT3, which is right here, and with the background of with the 2 micro replication origin, which is shown in here. And the lowest fluorescence was in CAN-MX with a SAN origin of replication. So how to design engineering probiotics? That's the next question that we ask ourselves. The key thing is noise always should be minimum, as minimum as possible. So we used an extensive literature search while we were designing these studies, and we found out that SC cell variability is a function of promoter, terminator, origin, and selective marker. Hence, we measured the coefficient of variation in the fluorescence values. And then, as you can see, plasmids exhibits less cell-to-cell variability than the 2 micron plasmids. And also, besides, auxotropic markers exhibited less noisy expression than the antifungal markers. So takeaway points. So we used auxotropic markers as well as antifungal markers, and we found out that they interact with each other with origins of replication to control gene expression. All in all, in this first part, we can confidently say that the genetic circuits that we designed can reduce plasmid-based expression in variants. Another key point that I want to take your attention is to, in metabolic engineering, we have this iterative cycle of design, build, test, and learn. So first, we designed our plasmids using genetic engineering strategies, and then we used primer design, DNA assemble, and computer-aided tools to build. And then next, we will test these probiotic yeast in situ. And then that's what I'm going to talk about in the next few slides. But before moving on, I want to talk to you about a little bit CRISPR-Cas, what why is it so important, and the evolution of CRISPR-Cas in the next couple slides. Since it is extremely important in genetic engineering techniques and genome editing tools, and it's been highly utilized for genome editing purposes. So CRISPR is an acronym for clustered to regularly interspersed short palindromic repeats. That's what it is. And Cas is it stands for CRISPR associated. So what CRISPR-Cas systems, these are present in most archaeal and many bacterial genomes. So you see Streptococcus pyogenes, which is a bacteria to adapt and acquire an adaptive immunity by utilizing CRISPR-Cas9. So I'll walk you through as simple as possible in the next few minutes. So what we see in here, RNA-based bacterial defense mechanism to recognize and eliminate foreign DNA from invading bacteriophage and plasmids. So which consist of Cas endonuclease right here, and that is directed to cleave a target sequence by a guide RNA. And Cas9 is cleaving or taking out these recognized sequence or to distinguish between host between self DNA from foreign DNA. And Cas9 nucleases are using this protospacer adjacent motif sequence. And due to the, for the sake of time, I'm not going to talk to you in extensive about the mechanism, but just to let you know, CRISPR-Cas9 systems distinguish self DNA from foreign DNA and naturally functions as an adaptive immunity mechanism in bacteria and archaeobacteria. And then what on this diagram, on this picture, you see the Streptococcus pyogenes mechanism of acquiring adaptive immunity by virtue of the CRISPR-Cas9 system. So I hope you appreciated CRISPR-Cas9 as a genetic scissors, and this technique makes it very easy to change DNA. Hence, it's been used extensively in genome editing techniques and purposes, as simple as cut and paste. And that has been awarded for a Nobel Prize in Chemistry in 2020. And next, I want to talk to you about, there are several classes, and I want to talk to you about the difference between class 1 and class 2 CRISPR-Cas systems. So by now, it's obvious that CRISPR-Cas is an archaeal and bacterial system of adaptive immunity that consists of CRISPR array and Cas genes. And we have two classes. One is called class 1. Class 1 is CRISPR-Cas systems that have effective modules composed of multiple Cas proteins, okay, that form a CRISPR RNA binding complex. And class 2 systems that have a single multi-domain CRISPR RNA binding protein that is functionally analogous to entire effective complex of class 1. So we have Cas9, Cas12, and Cas13, and so on and so forth. So the reason that I prepared these slides to the audience is to help appreciate, to understand basic principles, and also to appreciate why it's being used widely in genome editing tools. And since we used the same principles in our SP or Saccharomyces boulardii. So what we have done in here, we have used 3 genome editing methods. Getting back to our study, one is called double-stranded DNA integration. The other one is CRISPR-Cas9, and the last one is CRISPR-12A. So to characterize and overcome inefficiencies associated with genome integration, we compared 3 different editing methods in SP: unassisted linear double-stranded DNA integration and 2 CRISPR nucleases. The reason you see SP and LB, it's simply it's referring to the organism that is being originated. Streptococcus pyogenes for Cas9 and Lactobacillus bacterium derived from the Cas12a is derived from Lactobacillus bacterium. Different genetically engineered promoters and synthetic constructs using CRISPR, RNA, Cas12 RNA expression vector, Cas9 RNA, guide RNA, and Cas9 expression vector. And this table shows us the guide RNAs that we used for genome editing assisted with CRISPR nucleases, SP Cas9 or LB Cas12a. So these integration sites were chosen based on high expression levels from previously characterized chromosomal sites in SC. Again, we used SC as a model and utilized their integration site and whether it's applicable to SP in our synthetic constructs. So that's why we chose those sites in our experiments. So in the next slide, you see genomic editing efficiency. So let me walk you through tables first. What you see on table A, number of colonies screened for each editing method. Successfully edited cells are defined by the number of fluorescent. Remember, if you recall the earlier slides, we tested different promoters. So we used fluorescent promoter colonies divided by the total, which is shown in those numbers. And on table B, you see the percentage of these efficiencies. And as you can see in this figure, we were able to generate genome integrants in SP with 80 to 85% in Cas9 systems and close to between 95 to 99% in LB Cas12a system. One thing that I forgot to mention is the reason that we use CRISPR-Cas and genome editing is in SP is because these organisms, since these are not model organisms like Saccharomyces cerevisiae, these microorganisms are not genetically tractable. So it's not easy to manipulate SB. Hence, we use CRISPR-Cas systems to make it genetically tractable. Okay, so here's the colorful part. So once we verified which CRISPR-Cas was efficient, then we moved on utilizing those. Then we moved on biosynthesis of beta carotene in those vectors, in those promoters. So let me guide you through step by step. So what you see over here is the beta carotene pathway, synthesis pathway, or more specifically mevalonate pathway. So beta carotene synthesis starts with mevalonic acid, and through a number of phytin synthase and desaturases and cyclase enzymes. We have beta carotene synthesized from mevalonic acid. So accordingly, we used these enzymes to design our SP to make sure that we are that SP is synthesizing beta carotene. And it's the reason that we chose beta carotene synthesis is because it's since beta carotene is visible with naked eye, the color change. If the genetic modifications is successful, then we can monitor the changes by just checking the transformation of colonies. So what we have done, we ask ourselves whether if we use combinatorial pathway assembly to utilize and to assemble a promoter library for each gene that you have seen in here. So these are the genes that we used for our combinatorial pathway assembly of beta carotene pathway in SP. So our goal was to assemble beta carotene synthesis, biosynthesis pathway in vivo. And I'll walk you through again one at a time. So bear with me for a second. So what we have done, first, we used a combinatorial pathway overview of the assembly steps. That's what you see in here. Genes in the beta carotene pathway were randomly cloned behind 9 promoters. For the sake of simplicity, I'm not going to talk to you about all the details, but we used 9 different promoters. And these 9 different promoters were cloned behind several keratinogenesis enzymes like CRTE, which was shown in here, right? CRTE right here, CRTB and CRTI, and so on and so forth. So that's what we have done. You see CRTE, CRTI, CRTYB, and HMG1. That's shown in A, and then what you see over here, B and D, these are the SP transformation plates for beta carotene pathway. So what we have done, we pick out colonies from these plates, 13 out of 28. See, it's nice and easy to distinguish between colored versus not colored base, not colored colonies. So basically, coloration indicates the presence of beta carotene in those colonies. So we picked 13 out of 28 and proceeded with sequencing and beta carotene quantification. That's what you see. So 3, 4, 8, 12, and 13. So these are the 13 colonies that we pick. And we selected isolate number 23 because the reason being is that one can ask why we have not selected the highest beta carotene titer is because although clone numbers or isolate number 6 may yield higher beta carotene titer, but due to metabolic burden, we have not selected these isolate, and we have selected isolate number 23. And then what we have done next, once we selected this isolate number 23, we have gouage. Okay, next goal was to institute delivery of beta carotene. So what we have done, first and foremost, we first measured SP's residence time in the germ-free mouse by delivering SB or Saccharomyces boulardii to germ-free mice via oral gouage. So the reason that we, rather than gouging those colonies to mice directly, we wanted to find out whether SP can be present or can be colonized in the gut of germ-free mice. So we selected different dosage, 10 to the 6, 10 to the 7, and 10 to the 8 cells on different amounts of SP, 10 to the 6, 10 to the 7, and then 10 to the 8. And these were gouage on day 0. Gouage means directly inject. And then what we have done, directly given, I'm sorry, not directly given. And then what we have done, we measured the residence time of SP in germ-free mice. That's what you see in B panel. And error bars indicate the standard deviation among different, biological replicates. And feces collected prior to SP gouage during days shown in here. So all in all, these proof of principle studies showed us germ-free mice are appropriate for testing of biomolecule production in the gut environment, since colonization levels were the highest in this setting. So due to the sake of time, I did not talk to you about I did not present any information about conventional mice and antibiotic treated mice, but but we have seen germ-free mice was the most effective in terms of colonization of SP. Okay, next we chose, again, if you recall from the slide before, we chose this isolate number 23 for gouage for the delivery of that to mice. Since that's a strong beta-carotene producer, so we gouage mice to germ-free mice. We gouage beta-carotene producing SP to germ-free mice. As a control, we administered wild-type SB to also another sets of germ-free mice. So same amount of cells were administered to both groups. On the top panel, you see fecal load colony forming units of both wild type and beta-carotene producing yeast. So this tells us that these data indicate beta-carotene producing SP exhibited a similar ability to colonize germ-free mice as compared to wild-type Saccharomyces boulardii. So knowing that beta-carotene producing, so once we verified that beta-carotene producing Saccharomyces boulardii colonized germ-free mice, next we ask whether the quantity is something meaningful. In other words, in other words, we can measure, monitor the changes during the course of experiment. And that's what we have done over here. We collected fecal samples throughout the experiment, throughout the course of this experiment, and we measured the levels of beta-carotene in wild type as well as the beta-carotene producing yeast. So it is very fascinating to see an increase in the beta-carotene producing yeast in germ-free mice in the gut of germ-free mice in the large intestine, suggesting that we were able to deliver SP to mice, and in situ biosynthesis of beta-carotene takes place in mice, as we have demonstrated in this figure, over 14 days in the mammalian gut. That's really significant. And to summarize, we established an extensive toolkit of genetic engineering parts and techniques for SP. We have developed high efficiency transformation and genome editing protocols and created a list of tunable gene expression over a wide range. And we have engineered SP to produce beta carotene, which is a major dietary provitamin A carotenoid. And for the sake of time, I did not talk to you about the what does it mean and whether that has any significance in terms of the amount of beta carotene. But keep in mind that amount that you have seen was about 50% of daily beta carotene that we can that recommended beta carotene in our diets. And finally, we have demonstrated that biosynthesis of beta carotene primarily occurs while yeast is residing in the mammalian gut, not prior to delivery. As you have seen, there's a nice increase in the synthesis of beta carotene. And again, as I mentioned, that corresponds to 50% of the recommended daily intake of vitamin A, recommended daily intake of beta carotene. So moving forward, this work really positions us that SP, which is a probiotic yeast, I think all of you now that appreciate it's a probiotic yeast that can synthesize biomolecules using this genetic engineered that we created. And SP can be used as a robust host for production of biomolecules in the mammalian gut and reveals genetic engineering strategies that can be used for in situ biosynthesis and delivery of therapeutics. So moving forward, we will use these strategies to enable and to develop further technique, further enhance, further the release of beta carotene in the gut of mice and for high titer production of a variety of molecules, such as not beta carotenes only, such as antimicrobial peptides and so on and so forth. So we will continue developing these strategies. And as a matter of fact, we are writing another proposal, which is we'll be submitting in the next few days to National Science Foundation. Fingers crossed for that. So last but not least, I am very grateful to Nathan Crook and his talented grad student, Deniz Durmuşoğlu, for their insights and motivating me to investigate these techniques and how to make it better in terms of production of beta carotene in the yeast, in the probiotic yeast. So as I mentioned, we are collaborating in a number of projects with Dr. Nathan Crook, and one of them is NSF funded study, and another one is, I did not talk to you about that. We are also working on understanding whether carotenoids are biologically active molecules in the gut within the context of low-grade inflammation, which is funded by USDA, and stay tuned for that project too. So last but not least, also, I am looking for talented grad students and a postdoc to join my team at NC State. I cannot emphasize enough that it's really a teamwork. Maybe I'm the one that going through all these slides, but it's dedication of all these students and a team of investigators that really producing this beautiful work, great, fantastic science. So hopefully someone will really get excited as we are and pick up and help us to promote recruiting students and postdocs in the coming near future, in the coming years and near future. And that's my email address. If you want to reach out to me with this job advertisement, or in general, if you have any questions, I'll be glad to take. Well, that's about it. I think from a time point of view, it's perfect timing. I finished in 40 minutes or so. I'll be glad to take your questions.
Thank you so much for your speech. It was really informative for us. We have some questions that we want to ask. One of our participants asked that, do you think with CRISPR technology, human evolution can be controlled?
Well, I think so. I think so, because that's been very extensively studied, as I mentioned a couple times. It's very likely. I would say it's very likely.
Another participant asked that there are concerns about gene editing and its possible effect on creating so-called superhumans. What do you think about this, or can it be pretended by universal ethical rules?
It is another excellent questions by the participants. So this is also has been very meticulously under the radar, or very meticulously investigated about regarding the ethics of creating these utilizing these genomic editing tools to create superhumans, so-called. I would say I'm not really 100% familiar, but to my knowledge, there are some ethical rules or ethical organizations in the States. I'm not really confident, or I don't want to say something wrong, whether it's the same thing in other countries or in Europe or elsewhere. But I know in the States, there are ethical organizations, and there are also some foundations that are not investigating, they are controlling and whether compliant about the general scientific principles.
Thank you so much.
Sure.
One of our last questions involves that what do you think about most advantageous powers of CRISPR-Cas9 comparing other gene editing techniques such as CAST-T?
Such as CAST-T, you said?
What are the main advantages of CRISPR-Cas9 over CAST-T systems? I mean, why CRISPR-Cas9 is mainly preferred instead of other gene editing technologies?
Okay, that's a good question too. So if you go back, let me just do this. Okay, there it is. So the reason that CRISPR is so efficient in editing, in editing, is because it really gives us to how to say. Okay, I'm trying to simplify. It really allows us to cut and paste easily than other genetic editing tools like TALEN or others. Just recently, when I was reading this literature, I found out the efficiency of genetic editing with CRISPR-Cas9 is significantly better than other methods that has been used in the last decades. Why? I think it's really stemming from its ability to recognize foreign DNA efficiently. I don't know whether that answered the question.
Thank you so much, sir.
Sure.
In the following,
and also one other final comment to that previous question is, again, there are multiple. The evolution is I mean, for the sake of time and simplicity, I did not go through all these details. But there are excellent review papers in Nature, excuse me, and elsewhere. So these are really guiding us the recent developments in CRISPR, and it's really hard to evade from the immunity perspective bacteria acquire adaptive immunity. It's really hard to evade, and that makes CRISPR-Cas systems as an excellent or superior genetic editing tool. And there are, as I said, multiple Cas systems like Cas9, Cas12, and so on and so forth.
Thank you so much, sir. Again, we are so appreciated by your acceptance to our Congress. And for the evaluation part of our Congress, we'll be waiting you in the breakout rooms.
Sure. Thank you so much for the kind invitation again. And I hope the students and all of the participants really appreciate our findings, and I'll be glad to answer questions that have not been directed during this in the last few minutes. Feel free to email me at aeroglu@ncsu.edu, aeroglu@ncsu.edu.
We will be having a 15-minute break before our next speaker. The Congress will resume at 6 p.m. Our next speaker is Batuhan Turhan Bozkurt, who, if I may say, we shouldn't be fooled by his title of graduate student, and he carries many knowledge beyond his title. We will be listening to a poster presentation of his while we wait for our next speaker.
Well, thank you, Elif, for that very kind introduction. Will be a little bit more than a poster presentation. Actually, let me share my screen. So, is it working properly? Well, meanwhile, let me introduce myself again. I'm Batuhan. I graduated from Yeditepe's Genetics and Bioengineering program in 2018, and I very recently graduated from the master's program of the same university, Biotechnology. Throughout my years in Yeditepe, I worked with Dr. Neslihan Taşlı on the subject of extracellular vesicles. I've been with our research group for the last 5 years, and today I'll be sharing some of our works that we made, our research that we produced during those 5 years. Now, extracellular vesicles are quite fascinating. They're tiny lipid bilayered vesicular messengers that are secreted by all known cell types. And these vesicles, these little parcels of cargo, they carry cell-specific payloads that can affect the physiology of other cells. That's why I like to call extracellular vesicles the social media of the cells, because cells secrete these vesicles to give little status updates to cells at their close vicinity. For instance, if there's a wound, the cells in the wound area will be secreting extracellular vesicles that will signal for inflammation and apoptosis, etc., and wound healing, most likely. So today's presentation will have these 4 parts. I'll start with a brief introduction of the background of extracellular vesicles, how they work, what they're composed of. Then I'll share my first research. It was an article we published in 2019 on an isolation protocol for extracellular vesicles. Next, for the characterization section, I will be presenting my thesis work, which is on the characterization of plant extracellular vesicles. And finally, I'll wrap things up with drug delivery using extracellular vesicles. As natural messengers, they have some great innate properties, and I'll be sharing a final project there as well. So let's start with biogenesis, composition, and properties of extracellular vesicles. Again, EVs are tiny lipid bilayered nanovesicular messengers. They range between 30 nanometers to up to a micron in size, in diameter. And there are even some studies that suggest that there might be EV smaller than 30 nanometers as well. EV secretion is a highly conserved trait between different types of organisms. At this point, all studied cells and organisms that we know of secrete extracellular vesicles. They're present in plants, fungi, prokaryotes, different types of eukaryotes. And the secretion, biogenesis and secretion, as well as the composition, is also highly conserved as well. So there are similar proteins in different organisms. The cargo of the EV is highly plastic. The main structural elements tends to be the same, but the signal, the message that the EV carries can change dramatically under different physiological conditions and by different cells. This is how EVs are used by cells to secrete a message, send a message to one another. So let's have a brief overview on their structure. They're covered by a lipid bilayer membrane. It's really similar to the cell membrane with an abundance of rough associated lipids, and which makes the membrane a little bit more rigid than the normal cell membrane. We have a lot of surface and cytosolic proteins that can initiate biological responses on their own. And we have nucleic acids, miRNA, mRNA, and DNA fragments, which can affect the expression of proteins in target cells. As an element of cellular communication, EVs are practically present in all types of biological functions. They're present in, they're strongly present in immune response, both in the stimulation and regulation of the immune system. They're present in development and cell differentiation. They're everywhere. And this is also true for non-mammalian cells, non-human cells as well. For instance, we know that pathogens such as eukaryotic protozoa, apologies, protozoa and even some prokaryotic cells such as gram-negative bacteria secrete extracellular vesicles in order to create cause immune suppression so that they can successfully infect their targets. Now, secretion of an EV is a highly complex protocol, and it can happen through a variety of different pathways. This is still very much up to debate. We are still discovering a lot of things about both how EVs are secreted and how they are uptaken by target cells. On the clinical side of things, EVs are quite useful because they are basically a mini cell, a carbon copy, not a carbon copy, but a similar tiny version of the cell. They can be used as a biomarker in order to detect different diseases. For instance, we can use EVs of a cancer to basically have a no-touch biopsy of that cancer, because that EV might have DNA fragments and mRNA associated to a specific type of cancer. They're great drug delivery systems, more on that later, and they can be used in place of a cell. So we can have cell-free therapy, for instance, using mesenchymal stem cell EVs to have immune suppression, to help with organ grafting, for instance. Now, EV isolation is quite difficult, with an asterisk on the top. We can easily isolate EVs using ultracentrifugation. It's a really simple method. It's the most common type of one, but simple ultracentrifugation-based methods will have high purity of EVs with very low efficiency. On the other side of the spectrum, we have low purity, high efficiency methods. And we need to decide based on what we are experimenting with and what our application is on the ideal, the optimal isolation technique. The ultracentrifugation methods are really simple. We sequentially increase our centrifugation speed and sediment more and more particles. At the end, we get to something around 100 to 200,000 G, which will sediment the smallest of extracellular vesicles. Now, the simple differential ultracentrifugation is the most common EV isolation method, but it's also a really poor method. The efficiency is really low, but in addition to that, because we are sedimenting EVs to the bottom of a tube with such high speeds, the structure of the EV actually gets deformed, and that could affect our downstream applications. Density gradient ultracentrifugation, on the other hand, uses gradients of different solutions with different densities, and EVs will slowly move through these solutions and reach a resting point at around 1.17 grams per milliliter density, which is their own density. So at that point, the forces cancel out, and we can just scoop out that density gradient and use EVs that way. This method is ideal for analytical studies because we are getting very little amount of EVs, but at a really high purity. So this is great for proteomics, transcriptomics studies, but it's not that useful for an in vivo study, for instance. And even with some in vitro studies, we might not be getting enough EVs for multiple samples. The middle ground is, well, it's rich at this point. There are quite a few new methods that are being developed in this field. This is in a chronological order, more or less. The first one came out in 2014, the aqueous two-phase systems. And we developed an optimization of that protocol in 2019. That will be the protocol, the study I'll be sharing today. It uses liquid-liquid fractionation. So we have two different liquids that can mix with one another, but form two distinct phases after centrifugation. One of the liquids will have an affinity for extracellular vesicles, so it will pull the extracellular vesicles in. And by taking that fraction out, we will have our sample. Then we have affinity capture, where we can use antibodies that will target different parts of the EV to collect those EVs. This can be done through chromatography or using magnetic beads covered with those antibodies. We have tangential flow systems. These are getting more and more popular. They are both quite efficient and isolate very little amount of impurities. In these, we will have parallel flows. I'll actually have a picture for that later. And we have macrofluidic systems, which use sonication to move EVs around and isolate them in a size-dependent manner. So here you can see a diagram of the tangential flow system. We have affinity captures as well. But the final methods, polymeric precipitation, I don't really like this one. Polymeric precipitation uses large molecular weight polymers in order to basically make everything in a sample sediment at normal centrifugation speeds like 10,000 G. Now, the problem with that is that a lot of the soluble proteins will be sedimenting with extracellular vesicles, and they will create a high amount of contamination in our sample, which will make it really difficult for us to determine whether something we're seeing is because of the EV or because of our contaminants. So most of the high impact studies actually simply disallow the use of these. The high impact journals don't like the precipitation based methods. However, most commercial kits are precipitation based. So that's something to be on the lookout for. So here is our first paper. This one is the first paper I published. It is on aqueous two-phase system isolation of extracellular vesicles. So our idea was that we didn't have an ultracentrifuge at that point. We didn't have one in 2018/2019. So we needed a way to isolate extracellular vesicles. We didn't want to use a kit because the kits are both expensive and has a lot of contaminants in their isolates. So we found this paper that was published in 2014 about EV isolation using this method, but we were working with plant extracellular vesicles at that point, and we were even exploring isolating the extracellular vesicles from parasite cultures. And that protocol wasn't optimized to isolate from those sources. So we were like, let's take this protocol and try to improve it somehow. And what we came up with was this whole process. It uses two different polymers, polyethylene glycol and dextran. And when we mix these two up and centrifuge them, we get those two phases. By removing the upper phase with the contaminants and adding fresh peg in, fresh upper phase in, we can wash away the contaminants from our EV sample and get a quite pure sample of EVs. Actually, the system is quite capable of removing 99 something percent of proteins away from the EV sample. So we were quite happy with that result. And we tested this with plants and parasite cultures that was novel, and as well as we changed the isolation protocol to use pre-made solutions instead of dissolving these PEG and dextran solids inside media, and that improved our speed, and it subjected our samples to less heat in room temperature conditions. So that improved our stability as well. So here are the results from that paper. I'll really briefly go over these. These results basically showed that our protocol was capable of removing lipid contaminants. We salted our samples with a lipid and went through the isolation protocol. And as you can see, the PEG phase is the one we discard, and all the lipids that we salted are in here, while the dextran phase that we're collecting is actually lipid-free. in this one. We tested for the ability for this protocol to remove phytochemicals, and for that we used ascorbic acid as an example and model phytochemical, so to say. And the protocol removed away up to 90% in the first step, and it simply became undetectable after the third wash. And here you can see that the ATPase protocol was quite capable of removing protein contaminants. After the second wash, the third wash, we reach undetectable levels. But I also want to talk about in this talk how we could have done these things differently. We have a lot of students in this talk, and I want to make sure that our work is never perfect. There's always room for improvement. Our biggest issue with this protocol is dextran. We get our samples, our EVs, suspended in dextran, and while dextran is FDA approved, it is fine to be used in vivo systems. So for future clinical approaches, we are probably fine. It's also really viscous. It precipitates with alcohol. So we had a lot of difficulties with isolating RNA, DNA from these samples. So we need a way of removing dextran away from our extracellular vesicles to reach, to use this protocol at a higher efficiency, at a higher purity, so to say. And dextran have biological effects in vitro samples, at least. In some of the studies we used dextran. We saw that dextran could initiate biological responses all by itself. And that was a problem because we couldn't be sure if the effect we are observing is from exosomes, extracellular vesicles, or dextran. The second point is blending. Now, at that point, all of the plant extracellular vesicles research that were being published used blenders. They took fruits or leaves of plants, they juiced them using blenders and took EVs from that. However, such harsh isolation methods break down cells and release internal contaminants, such as internal vesicles. Now, at this point, we weren't quite aware of this, and neither was the field. There is a standardization article published in 2018 called MySafe that fixed most of the issues, at least made us become aware of most of these issues in this paper. Finally, we have the non-standard characterization of plant and parasite EVs. In order to characterize extracellular vesicles, we need to hit several guidelines, and those guidelines were simply not published at this point. So we weren't quite aware of them. And actually, there are still no guidelines for non-mammalian extracellular vesicles. All the optimization was done with mammalian ones. So that was also one thing that could be improved in the future. And as a research team, after this paper, we learned that there's no ideal isolation method. We really liked the ATPase one, but it was really challenging in some applications such as DNA isolation and downstream proteomics, transcriptomics. So we instead started to look for other methods such as ultracentrifugation based methods, density Cushion isolation for analytical samples. And we noticed that the plant EV research was really lacking in non-mammalian EVs, which leads to my next point, characterization. Now, with that paper, with some other stuff, we became really interested in doing plant EV research. It's a really abundant field, in spite of the fact that plant EVs were actually characterized quite a lot earlier than mammalian EVs. The first definition of plasma human extracellular vesicles were made in 1941, but it took up to 40 years, almost 40 years, for them to take 10 images of human extracellular vesicles. In contrast, plant extracellular vesicles were imagined in 1957, and the field actually went quite fast after that point. They did some hypotheses in plant extracellular vesicles that were almost correct, which we now know that those hypotheses will also be done 10 years later in humans. But the field came to a complete stop at 2000-ish, while human extracellular vesicle research skyrocketed. And now in 2020, we're talking about, oh, we should improve the quality of our plant extracellular vesicle papers. And that really requires characterization. Now, at this point, we have around 40 papers on plant extracellular vesicles. And they report some really promising therapeutic effects against cancer, wound healing, etc. Plant extracellular vesicles can be a viable alternative, a cheaper alternative to mesenchymal stem cell human extracellular vesicles for drug delivery applications and modifications. And we have no idea how extracellular vesicles fit in for most of the plant biology subjects. EVs are practically everywhere in human processes, so they're probably everywhere in plant processes as well. At this point, we only know that plants secrete extracellular vesicles against biotic stress, so fungi infections, for instance. But we haven't really studied anything else. But they could be present in abiotic stress, development, transformation. We can use them for transformed cells, etc. So at this point, these are every single plant EV paper that was published. So we have asparagus, grapefruits, we have lemons, strawberries. There's 40 odd papers in total. Why is that? Why we only have 40 plus papers in plants? Well, the problem is with characterization and isolation. There is a paper called Minimal Information for Studies of Extracellular Vesicles. It was published by the International Society of Extracellular Vesicles. And this paper gave us the foundations for high quality extracellular vesicles research in humans, in mammalian systems. And there is simply no equivalent to something like that with plants. In order to publish an extracellular vesicles paper, we need to have these two characterization steps. We need to first characterize our samples physically using scanning or transmission electron microscopy. We should also have something like a nanoparticle tracking analysis, which gives us the size distribution and concentration of our extracellular vesicles. We also need a biochemical characterization, which will look at biomolecules, almost always proteins, to show that EV is part of the cell membrane, is intact, so it has some cytosolic proteins in it, and is not highly contaminated by soluble proteins, which are normally not expected to appear in extracellular vesicles. Serum albumin is a good example. Now, because these methods are mostly protein-based, we use antibodies to do these, and those antibodies are simply not available for purchasing easily in plant research for most plants. So that is the problematic part. Again, no plant biomarkers exist. So in my thesis, I wanted to create a workflow for isolation and characterization of true plant extracellular vesicles. In order to have true plant extracellular vesicles, I had to get my samples without blending. I couldn't use any harsh isolation methods. And I wanted to do bioinformatic analysis on these samples to find conserved proteins, which can then be used for characterization. So I had these three steps. I first had to isolate my extracellular vesicles cleanly, get proteomic profiling done on them so that I can see what proteins are in there and if there are any patterns that I can use to characterize EVs in the future. And then in order to get biomarkers, I compared well-known human EV proteins with the proteins I got in plants, because I knew that EV secretion was highly conserved between species. So let's start with the characterization part. I used these three different plants, tobacco, grapes, and stevia, which is used in artificial sweeteners. And again, I couldn't use a blender, so we partnered with the plant EV research group from of Professor Bahar Soğutmaz Özdemir, and we got our samples from plant cell suspension cultures. So instead of getting a fruit and juicing it, I would be getting liquid media that is entirely similar to mammalian cell culture media. I centrifuged my samples and isolated my extracellular vesicles using density Cushion ultracentrifugation, which is the Cushion ones, the density ones are better than differential ultracentrifugation. They isolate more extracellular vesicles and subject EVs to less stress. And this was what I got. These are nanoparticle tracking analysis results. We call them NTA. And here I was really pleasantly surprised that my EVs were all in the size range that I was expecting. Then I sent my samples for proteomic profiling. I could only send a single sample, so I choose tobacco because it's a model organism. It's better studied than the other two. And I identified 135 proteins in the extracellular vesicles of tobacco. Then I wanted to see what these proteins were about, so I did gene ontology analysis. Gene ontology is basically a categorization, a human readable categorization of proteins. Let me get gene ontology looks like this. We have different categories where our proteins can belong to. A protein can belong to one or multiple categories, and through these we can see what our sample is about, what proteins are present, what sort of function the proteins of our sample have. In addition to gene ontology, there was one paper, a real recent review article that put forth 8 possible plant proteins as plant EV biomarkers. They had a little bit more than 8, apologies. I found 8 of my proteins that fit in with those proposed potential biomarkers, so that I was really happy to see that. In gene ontology, we look at biological processes, molecular function, and cellular compartment, where those proteins are coming from, basically, of my proteins. And I saw that my proteins in tobacco extracellular vesicles had really similar patterns to human extracellular vesicles, especially in the cell compartment part, because EVs generate from the cell membrane. We expect them to have a lot of cell membrane proteins, and that was true in my samples, and have little to no nuclear proteins, and nuclear, ER, Golgi, and other intracellular organelle proteins. And that was completely true for my samples. And because EVs are basically messengers, we also saw a high enrichment of response to stimulus proteins. Then I also did a cellular sublocation analysis. This is an algorithm that looks at the protein sequence of a protein and guesses where that protein would be coming from in the cell using a motif on protein motifs. And in my sample, I was happy to see that my samples were enriched with extracellular vesicle proteins and had little to do no nuclear proteins again. So that was a positive response we got as well. Finally, I wanted to compare the proteome I got with the published plant proteomes. So at that point, there were only 7 other proteomes that were published. My tobacco one made the 8th one. So I downloaded all those proteomes, and I also got the 300 most abundant human EV proteins from an online database called Vesiclepedia. And I did blast analysis between comparing my plant proteomes with the human proteome, the 300 human EV biomarkers that I got. My logic was that these 300 are very abundant in human EV proteins, so they were most likely to be more evolutionarily conserved compared to the whole human EV proteome. And what I got was really interesting. So I identified 24 proteins in these 8 samples that were shared with between at least in the high majority of the plant proteomes that I got. If proteins were present in at least 5 of the proteomes I looked, I marked them out. So these 24 proteins are highly conserved between different species of plants. There's one, adenyl homocystainase, which is present in 6 different proteomes. So these are likely good candidates for plant EV biomarker research. And there's also one other interesting thing. Out of those 300 human proteins, around 40% of them had a plant ortholog. So there is at least something around 40% conservation in human and plant EV secretion mechanisms. And these orthologs tended to be quite highly orthologous as well. We saw something around 80 to 90% similarity in some of them, in a majority of them. So that suggests that those plant EVs, those proteins on the plant EVs, they could have therapeutic effects in humans because they would be acting similar to their orthologs. They are basically they could be a replacement for their orthologs. So looking at that MySave paper, there are different categories of characterization, and I identified these 5 proteins as potential biomarkers that could be used for a whole range of different plants. And the use of plant suspension cultures was also a really major improvement of this paper compared to the literature. With that, we didn't use a blender. We could modify our culture however we wanted. We got more EVs from a more stable source compared to buying fruits from local markets, which is what most researchers did in the field. We developed these identification methods that can be used for plant EV identification and identified an homology between different plants and human EVs. What could be done to improve on this? Well, I'm in the process of looking at the callus proteins. So I'm checking the source of those plants, checking the cell cultures of those plants, getting their proteins out and comparing that proteome with my EV proteome. I would like to finish with the remaining plants, the other 2, and did a comparative study between them, test out large-scale EV production using suspension cultures, and test out various in vitro studies. Finally, because of EVs' high potential as drug delivery devices, I would like to see if plant EVs can be used as a replacement for mammalian EVs in that. So more on that now. Now, EVs are natural messengers. They are our body's solution to the PTT system, the UPS cargo we have. Because of that, they're also amazing drug delivery vehicles. They are natural carriers after all. So in another study we did with our colleagues, we loaded an anti-cancer therapeutic cape, caffeic acid phenylethyl ester, which is a part of propolis from honey. We loaded extracellular vesicles with this agent in order to try and treat neuroblastoma. Now, why am I talking this much about EVs as drug delivery devices? Because they are great at that. Compared to synthetic drug carriers such as liposomes, EVs show little to no toxicity and immune response. They're produced by our cells, so we can do autologous production, but we don't even need to, because taking EVs from one organism and giving it to another organism causes little to no response. little to no negative response, so to say. They have an innate ability to target cells. So if I take EVs from liver, for instance, if I inject them to a patient, those EVs are more likely to be present in the liver. That is because of the surface markers present on the EVs. EVs can naturally pass through the blood-brain barrier, which is great to deliver cargo to brain, which is notoriously difficult to send drugs to. And they have a unique uptake pathway. EVs tend to be taken in by cells through fusion, which allows them to avoid the degradative, the enzymatic degradation processes in the lysosome. So this allows us to send fragile cargo, such as CRISPR-Cas9, for instance, from Kerim Hoca's presentation. You would remember that we can send fragile cargo with extracellular vesicles more efficiently compared to synthetic ones. They have some downsides as well. EVs are biomolecules, so they come with proteins on them and proteins in them, as well as miRNA, mRNA. So we need to make sure that our EV is benign. It shouldn't do something bad to the cell that we're sending it to. The EV production is highly susceptible to changes in the cell culture. So if we have a bioreactor, we must make sure that the reactor is kept in a real stable condition to keep our EVs happy. And the current isolation methods have low efficiency and they don't really translate well to large scale production. And we still have a lot of unknowns in the literature. We don't know how EVs are generated, not specifically. We don't know how EVs choose which cargo to take in, etc. So there are some example of applications. As you can see, EVs have been successfully used to treat different types of cancers through dendritic cells. EVs take major part in immunomodulation and immune stimulation. So using EVs from dendritic cells stimulates immune system cells such as T cells quite effectively. Other studies looked at passing through the blood-brain barrier. So these two, one sent curcumin, another sent siRNA, past the blood-brain barrier to treat diseases of the brain. And more recent studies looked at mesenchymal stem cells to reduce the cytokine storm or high immunoinflammation, immunostimulation cases of COVID. Now, we can load EVs in two different ways. We can either isolate EVs separately and use sonication, freeze thawing, etc. to open up their pores and put our drugs in. That would be the ex vivo loading method. Or we can simply modify the cells producing the EVs to produce EVs with therapeutic properties. So it will be a prepackaged drug, so to say. This works great with protein drugs, or protein therapeutics would be a better word, or miRNA, etc. For the ex vivo version, we have a few methods. We can use some small nonpolar compounds can simply enter EVs through incubation. We can use electroporation, sonication, or saponification to open up the membrane to get drugs in. Now, before we decide on which method to use, we need to consider several things. You must look at the cargo that we're trying to load. Is it a large cargo? Is it polar or non-polar, charged or non-charged? This determines which loading method is the most efficient one. We must consider that stuff like electroporation and sonication damages EVs more. So if we want to use their, for instance, cell homing capabilities, we need to use gentler methods such as saponification or the in vivo production. And will the contents of the EV be an issue? Are we using something like cancer EVs because we are trying to target cancer? Will that cause tumorigenesis on our target cells, for instance? We need a way to measure loading efficiency effectively. The source of the EV also matters. if we take EVs for mesenchymal stem cells, that will have different properties compared to EVs of dendritic cells. It will determine cell homing, it will determine the biological effects of the EV, so that's an important one. The isolation methods might change. How much EVs do we need? Do we need a lot? Do we need little? How are we delivering these EVs? Are we going intravenous, microneedle patches for targeted delivery, intranasal ones? EVs can pass through the blood-brain barrier, but getting them closer to the brain is always a plus for that targeting, etc. Now, in this study, we tested different loading methods to load CAPE, caffeic acid phenylester ester. Now, CAPE is a small non-polar molecule. So we could load it passively without doing anything else. We just gave an abundant amount of CAPE to our EVs, let them incubate, and it would pick some of it up. But as you can see, the more active methods increase loading dramatically, up to three times each. So we opted to test these out, electroporation and sonication. However, those methods greatly change the morphology and size of EVs. Normally, the EVs that we use are around 200 nanometers maximum in size. They average between 120. So a happy EV, basically, a undamaged EV profile would be a single peak around that. However, because of the stresses caused by sonication and electroporation, the EVs actually gained in size and mass, and we got these wild peaks that appeared after those protocols. And those protocols also caused some aggregation and breakdown of EVs, which led to us losing almost half of our EVs in the sample. So we learned that those EV ex vivo loading methods were a bit troublesome. We ended up going through with this method for this paper, but in future, for my own research, I will definitely be opting more towards in vivo loading methods. And we also noticed that it is really important to have a well-optimized method to detect loaded cargo, because without something that can detect very small amounts of your cargo, because EVs are so small, it's really difficult to detect the cargo that enters the EVs as well. So you need something like an HPLC for accurate measurements. Well, that is it for my presentation. Thank you for your attention and listening, and I'll be happy to answer your questions.
Thank you so much for your presentation. It was our utmost honor by your acceptance. We have a question. What do you think about the future of extracellular vesicles and its applications? And in which areas do you think it will make a huge difference? One of our participants asked.
Great question. First of all, EVs are really flexible. So they're present in basically every field of biology somehow, in some degree. But I think they will really blow up in drug delivery, because with improvements in CRISPR-Cas9 and similar gene editing methods, EVs become more and more attractive delivery tools to deliver those therapeutics. So I think drug delivery is one major field. Another one is with vaccines. Immune cell extracellular vesicles can carry MHC2 and antigen-presenting proteins, basically. So an EV can basically turn a dendritic cell, give a dendritic cell a specific antigen, which is what we want to do with vaccines. So that's another field, immunology. And I think there should definitely be more research with looking at the natural extracellular vesicles and what they do with cancer, because it's always a popular field, research of cancer, but we don't know how EVs really fit in. We know EVs of cancer can turn healthy cells cancerous, We know that EVs of cancer can cause immunosuppression. We still don't know really how. So that's also another field that could be interesting. But they're basically applicable in every field.
So in that sense, are we able to do modeling of certain diseases such as Alzheimer's disease or Parkinson's disease with exosomes? Is that correct?
Great for you to remind me that EVs are a major part in recent hypotheses on neurodegenerative disorders. So you know that most neurodegenerative disorders have a portion of protein aggregation in them. In Alzheimer's and Parkinson, we have those with plaques forming. We have ALS with a profile of protein aggregation there as well. And there are studies that suggest that a diseased neuron will be sending extracellular vesicles loaded with these aggregates to other neurons. They don't intend to do that, but EV secretion is basically a garbage disposal tool for the cell as well. So neuron is trying to survive by throwing away all those aggregates, but other neurons in the vicinity pick them up and they get hurt by those as well. So that's a major point of that, like the neurodegenerative disorder part. There's also a recent paper that I read that suggests Parkinson's might be starting from the gut. So in our small intestines, those cells get protein aggregates, and these aggregates are transferred to the brain through extracellular vesicles because EVs can pass through the blood-brain barrier. So that could be another part of pathogenesis of diseases, of neurodegenerative diseases.
Thank you so much for your speech. For the evaluation of the Congress, we would like to invite you in the breakout room. So that's all.
Thanks again for inviting, and I'll see you there.
Thank you so much. Our next speaker will be the Professor Andis Klegris, and he will be attend at the 7 p.m. See you then.
Our last speaker for this conference is Professor Andis Klegris from the University of British Columbia to deliver a speech titled Neuroimmune Signaling Between Different Cell Types of the Central Nervous System in Pathogenesis of Alzheimer's Disease. In this presentation, he will explain his in-depth research concerning the role of neuroimmune mechanisms play in Alzheimer's disease onset and progression, In addition to this, he will also be informing us about the main brain cell types orchestrating neuroinflammatory responses in the central nervous system. Welcome again, and thank you for accepting our invitation. If there are any questions, you may write it in the YouTube live stream or through Instagram DM.
All right, good evening, everybody. Can you hear me? Please confirm.
Yes, we can.
Excellent. Well, thank you so much to the organizers of this committee, of this conference, for this opportunity to present to you our research about Alzheimer's disease and neuroimmune signaling. I just wanted to kind of show that where we are. So I'm talking to you from Western Canada. I work in the University of British Columbia. So British Columbia is the westernmost province in Canada. And UBC's main campus is located in Vancouver, somewhere like here on the coast. I work at the Okanagan campus, which is about 400 kilometers east from Vancouver, about 100 kilometers off the border with the United States. And you may not know this, but Okanagan campus where I work is called Okanagan due to indigenous name of a large lake. And that's the Okanagan Lake. And that's how it looks. And I always felt that Okanagan must be looking somewhat like Turkey. I don't know why, but it's just how I see Turkey. But unfortunately, I was not able to visit this time, but I'm super happy to talk to you guys over the Zoom platform. So this part of Canada, we do have snow and skiing right now, but we, like as a southernmost part in Canada, there's lots of fruit growing, vineyards. So that's just kind of like to orient how this looks in a place where I am right now. And it is 8 a.m. in the morning. So we are kind of on the almost opposite side of the planet. So anyway, so I'm going to be talking to you about Alzheimer's disease. And you probably know that it is the most common cause of senile dementia. And like I'm sure that not many of you are thinking about this disease, but I still think that it could be relevant when you consider your career. Because of these graphs, right? So as we age, and clearly none of you are in this risk area yet, but as we age, the risk of actually acquiring this disease is very high. And by some estimates, like 30% or even higher, if you are lucky to live that long. Now, we all want to live long, and we all want to live healthy lives. And if we happen to live long, we are facing with these odds. And also, unfortunately, there's no effective treatment. So this is something what motivates our lab to research this disease. Now, when Dr. Alzheimer first described this pathology in 1906, in the post-mortem tissues, he immediately recognized these two structures, tangles, intracellular, and plaques. And at that time, Dr. Alzheimer wrote that he cannot tell whether these structures cause the disease or these structures are just a result of the disease or just a kind of side effect of the disease, which is not really relevant to the pathology. Now, more than 100 years later, we are about at the same stage that we don't know, like why these structures are, right? Are they caused? Are they not? And the reason why I'm saying that is because all our attempts to actually create treatments based on attacking these structures have failed. However, over the last 100 and more years, we've learned a lot about these structures. We know that neurofibrillary tangles are made out of proteins called tau. Plaques are made out of proteins called A-beta. We know that synaptic function is not working very well We know that the brain is shrinking, especially in several particular areas of the brain. And what is really exciting recently, that we have learned how to observe these structures and these deficits in live patients. Because just before maybe like 20, 30 years ago, we only could see Alzheimer's pathology in humans on postmortem tissues. So here you can see amyloid beta imaged by positron emission tomography, and you can clearly see that Alzheimer's disease patients have much higher levels of A beta compared to normal, healthy controls, age-matched. So all aged humans have some amyloid, but not to the extent as Alzheimer's patients. The second marker is showing that there's like disturbances in synaptic activity in the brain, in addition to general shrinkage. And these kind of positron emission tomography imaging studies allowed us to appreciate that the pathology of this disease starts much earlier, many years before actual symptoms appear. So here on this graph, you have a timeline on x-axis. So Alzheimer's pathology starts usually with symptoms of mild cognitive impairment. But here you can see that the structural changes, deposition of tau, A beta, and synaptic dysfunction happens long before the onset of symptoms. Now, there's one more curve which has been added recently to this graph. And this is this brown curve, which tells that there's an increased microglial activation, perhaps even before appearance of these protein deposits in the brain as an early precursor of Alzheimer's disease. So how we know that? Again, just because we learned how to image these reactive microglia in human brain by using PET scanning. So what are microglia? So brain, and you guys most likely know that the brain has not only neurons, which are responsible for our thoughts, consciousness, and so forth, emotions, right? There are several different non-neuronal cell types. And microglia is one of them. So here you can see other cell types, but in today's talk, I'm going to be focusing on these two non-neuronal cell types. So microglia are a type of macrophage. So it is an immune cell which can present antigens. It can launch innate immune attack. So it is essentially the only immunocyte, the only professional immunocyte in the brain, which is responsible for like our health. It keeps brain healthy, right? Astrocytes, they were usually thought as support cells, kind of fibroblasts of the brain. But we now know that they support also synaptic functions, help maintain blood-brain barrier, and also participate in neuroimmune responses. Now, how this Alzheimer's neuroimmune hypothesis was kind of stated. It started with these observations, essentially in a lab of Drs. McGeer, and they noticed that, say, in a normal brain, microglia, and you can see on this slide, they are very highly ramified cells with these extending processes. They are very territorial and doesn't seem to touch each other. But if you look at Alzheimer's brain, here you can see microglia as these black colored aggregates of cells which appear to be losing these processes and appear to be attacking the amyloid plaque. And amyloid plaque is stained in this light brown color. So here you can see this one of these resting or serving microglia. So, and researchers interpreted this as microglia as professional immunocytes. They detect abnormal protein structures and they become reactive and they try to remove these plaques. They are successful if there's not a lot of amyloid beta protein. But if there's excessive production of amyloid beta, they are not just able to remove this protein, and they become chronically active, and they start secreting substances which are designed to kill pathogens. But in this case, it is a sterile inflammation. There's no pathogens. And as a side effect, they essentially end up killing healthy cells. This initial observation was first supported by epidemiological studies showing that chronic users of non-steroidal anti-inflammatory drugs have reduced risk of Alzheimer's. Now, who would take NSAIDs for long periods of time at high doses? Typically, they would be humans who experience chronic pain, such as, for example, patients living with arthritis. So they would go to pharmacy, or their doctors would prescribe NSAIDs, and they will take them for many years. And it appears that this group of patients are protected from Alzheimer's disease. And there are more than 20 studies documenting that. So after these two observations, it was discovered that the brain levels of inflammatory mediators are elevated. And there was great enthusiasm about the neuroinflammatory hypothesis until several clinical trials failed to show benefits of NSAIDs. And there are many different attempts to explain why this happened, but these observations led to essentially kind of like a disappointment and loss of interest, perhaps, in microglia for several years, until about 10, 15 years ago, genome-wide association studies showed that the mutations in several genes increase risk of sporadic Alzheimer's disease. And sporadic Alzheimer's disease is the one which really, we don't really know the cause, but it is the majority of Alzheimer's disease cases. And these studies showed that many of these risk genes are either expressed on microglia or linked to immune responses. So this has renewed interest in the neuroimmune hypothesis tremendously and refocused many researchers on studying microglia and immune responses in the brain. So this has led to some interesting observations. The first one is the following. So we always felt that microglia are these static cells, and they kind of basically, yeah, they sit there, territorial, don't move. Until two-photon imaging was designed, where a small hole can be drilled in a skull of animal, and two lasers pointed into the depth of the tissue. And if to make these animals transgenic for microglia expressing green fluorescent protein, we can actually observe these cells live in animals. And I'm going to play this movie created by authors listed on this slide. And you can see that these microglia appear not to be static. Now, if you take a look at one cell, for example, this one and this one, and I'm going to play it again, you can see that the cell extends and retracts these processes all the time. And this is happening over a span of 6 minutes. So over 6 minutes, they can extend and retract these processes. And if there is suddenly damage, they would quickly move towards this damage and try to resolve it. So there's no reason for us to think that this is specific for animals. Nobody has made such movie, obviously, for humans yet. But like while you are kind of listening, hopefully, to this presentation, you could think that within your brain, this is what's going on right now. Microglia is just basically actively searching for any kind of trouble. They are helping remove synapses, what you don't need. They are helping to establish other synapses. They are essentially working non-stop, keeping your brain healthy. So that was a great discovery. Now, latest single-cell omics techniques also have demonstrated a couple of other interesting things. First, there's great difference between species. So mice are not men and vice versa. Microglia and glia are quite different between rodents and humans. Also, there's great functional diversity of microglia, and microglia, as well as astrocytes, have this really significant difference in terms of their spatial location, as well as how they look and function over the course of the development, as well as over the course of Alzheimer's disease, for example, pathogenesis. So, but just for the sake of this talk, let's keep this simple and let's consider microglia as these immunocytes which try to remove Alzheimer's plaque. They become reactive. Astrocytes do not really move to attack the plaque. They seem to try and encapsulate this whole thing to protect the rest of the brain from this. I also should note that both microglia and astrocytes, they are obviously needed cells, so they are able to secrete a lot of required and protective molecules such as growth factors, but in Alzheimer's disease they may lose this function and become reactive. So therefore, this Alzheimer's hypothesis goes as follows. So these pathological structures such as amyloid in Alzheimer's disease may be activating microglia directly, and that has been demonstrated for microglia to become reactive, and they kill neurons, and neurons release substances, molecules, which activate microglia. Once microglia are adversely activated, they start to secrete toxins, which are designed, as I mentioned already, to kill bacteria, for example. But since they cannot secrete selective molecules which are specific for bacteria, they end up further damaging neurons and other cells, and this basically completes this vicious cycle. So our lab first became interested in what kind of toxins are secreted by microglia. So recently we summarized and reviewed literature, and we were able to identify more than 20 molecules which are released by microglia, and they have a potential to kill neurons. So, and we hypothesized that it is very likely that microglia does not secrete just one toxin. It's most likely that they release mixture of toxins, and that this cocktail of toxins depends on the type of stimulus microglia encounters. So we decided to test this hypothesis and selected these 4 toxins. Glutamate is a kind of like a neurotransmitter which can become toxic. This is tumor necrosis factor, a cytokine. This is another cytokine, and nitric oxide is kind of like a reactive nitrogen species. In addition, we used like a simple cell culture system to assess the overall toxicity of microglia, where we take these cells, they could be either primary human microglia or animal microglia or some cell line. We stimulate them for, say, 48 hours with various stimulants, and then we take supernatants from these microglia, transfer onto neuronal cell model, and measure viability of these. So these simple experiments showed the following. So here we have these 4 toxins as well as overall toxicity of microglia supernatants. And we kind of added several different stimuli, starting with LPS, which is a bacterial product. Zymosan is a yeast product. These 2 are cytokines. This substance models virus. And ATP, as you know, is a molecule involved in the cellular energy, but it is also a neurotransmitter which can regulate microglial functions. So, and as you can see, several of these, or 2 of these molecules or stimulants induce secretion of several different toxins, while some of them induce secretion of a single toxin. Now, I must mention that we don't know how and what happened with the other toxins listed on the previous slide. It is possible that in addition to these 3, there were more of the toxins released, but overall LPS and interferon beta were able to induce overall toxicity on their own, meaning that there was enough of the diversity of toxins and enough of the concentration of the toxin for microglia to be able to kill neurons. So I also wanted kind of to mention how we model glia in vitro environment. And here you can see primary human microglia and primary human astrocytes. So how do we obtain them? First, we can get them from post-mortem human tissues. So when a human or animal dies, so typically that means that neuronal cells are dead. But glial cells survive for at least another 24 hours. And if they are extracted rapidly, they can be kept alive in appropriate cell growth media. Now, this is technically difficult to achieve. The other also possibility, and these particular cells on this photo, they were actually obtained from human epilepsy surgeries, right? So in epilepsy cases, quite often neurosurgeons go into patient's brain and remove small pieces of tissue, and we can collect those tissues, usually cortical tissues, and extract primary human glial cells. Now, more common is use of models. So here is like a human microglia-like cell and human astrocyte cell, which we use in vitro. With neurons, it's even more difficult because obviously securing live human primary neurons is not possible from post-mortem tissues. Also, it's hard from surgical tissues. So we use cell lines such as SHY5Y, which doesn't look like neuron, but it can be induced to behave like neuron by something like retinoic acid. And here you can see that their morphology change. They start growing these processes, establish synapses, and express markers of neurons. And this is how these cells are stained by markers of neurons. So I also wanted to mention, and this technology we don't have in our lab yet, but the discovery of iPSC technology, induced pluripotent stem cell technology, has allowed several labs on this planet to learn how to grow brain organoids. So these are essentially several millimeters large tissues where iPSCs are derived first from somatic cells, such as blood cell or fibroblast, and then they are forced into turning into brain. And these organoids have neurons and astrocytes, and some labs even are able to make them contain also microglia. So these are like these small models of human brains. Now, this panel, I also just wanted to show you because I find this very fascinating, is that HC is healthy control. So here again, there are, again, like don't worry about what these colors are, but they have neurons, astrocytes. But what I wanted to point out is these familial Alzheimer's disease cases where iPSCs were made from humans carrying mutation of Alzheimer's disease. And what these organoids show is development of amyloid plaques. So green structure is actually amyloid plaque, which we would expect to see in human brain from a patient who carries these mutations quite early in their aging process. So, therefore, this brain organoids likely is a future how we will be studying cellular interactions in vitro. So after kind of thinking about these toxins, what are released, another curiosity for us were actually microglial activators. Now, what are the molecules which are released by neurons and possibly other cells which instruct microglia to become toxic? In addition, in non-pathological scenarios, there must be like a broad range of such molecules instructing microglia to phagocytose, instructing them to remove synapses, instructing them to become quite different depending on where they are located in the brain. And if to look at the literature, there is not much information out there. So there are some molecules which we know, such as glutamate, ATP, cytokines, also some membrane phospholipids on cytolemma, which are recognized by microglia, and obviously pathology-associated molecules. So we wanted to extend this list because clearly it is not complete. And we chose to use this technique, which is called SciLac. And essentially what it does is that it is possible to grow cells in modified cell culture medium where all amino acids are labeled with this non-radioactive heavy carbon. And if you grow these cells from several generations, they incorporate this carbon in their proteins. And if we stimulate half of these cells and have another half unstimulated, and then afterwards we just kind of mix all the proteins together, digest by trypsin, do mass spectrometry, we are able not only to identify the proteins, but tell whether protein concentrations are increased, decreased, or they are staying the same. And output of this technique, which is actually very, very laborious, is like these long lists of proteins telling that some of them are upregulated, some down, and some are not changed. So after reviewing this list, there were several established toxins, but what caught our attention is cytochrome C, which was detected in this in the cell culture supernatant, right? So we collected supernatants from these labeled cells and noticed that cytochrome C is in extracellular medium. Now, many of you know that cytochrome C is like an essential electron carrier in mitochondria, and it is essential for ATP synthesis in all cells. Also, it is quite established that once released from mitochondria into cytoplasm, cytochrome C activates apoptosis. But what happens when cytochrome C then is released into extracellular medium? And there was very, very limited information flow. So we know that cytochrome C is highly conserved protein. There was evidence that it can be released by damaged neurons and some peripheral cells. And interestingly, serum concentrations of cytochrome C were suggested to be markers of several types of pathologies listed on this slide. And most interesting to us was that injecting cytochrome C into joint was shown by these authors to induce arthritis. Now, putting all this together made us think that cytochrome C very much looks like what's known as damage-associated molecular pattern. So these are endogenous molecules which are released from cells. They typically interact with what's known as pattern recognition receptors and induce inflammatory and cytotoxic response. So pattern recognition receptors, some are listed on this slide. The most famous are these toll-like receptors. And why damage-associated molecular patterns are called like that? This name is kind of was made analogous to another term, which are pathogen-associated molecular patterns, which are conserved structures of bacteria, viruses, and so forth, which are again recognized by these pattern recognition receptors. Now, another piece of puzzle, you probably know that there is this like hypothesis that mitochondria are actually ancient bacteria, which are now essentially living inside our cells, and it's like a symbiotic relationship. Now, if that is the case, then proteins in mitochondria are ancient pathogen-associated molecular patterns, and having DAMPs, therefore, in mitochondria would make a lot of sense. So we set out to actually test this hypothesis that cytochrome C is mitochondrial damp, or what is the same, some ancient bacterial pattern. First, we showed that if you kind of induce death of microglia by something like tumor necrosis factor or amyloid beta protein, and like viability goes down, this is accompanied by increased concentration of cytochrome C in cell culture. Next, we exposed microglia cells to cytochrome C and learned that on its own, it doesn't induce toxicity, but if we combine cytochrome C with some other pro-inflammatory stimulus, such as a lipopolysaccharide, that it causes microglia to start secreting toxins and killing neurons. So this is viability of these SY5Y cells, which have been exposed to supernatants from microglia in the present, which are stimulated by a combination of LPS and cytochrome C. Now, interestingly, astrocytes became neurotoxic when exposed to cytochrome C on its own. So here you can see that if to expose astrocyte to cytochrome C and then transfer medium onto neurons, neuronal viability goes down and neuronal death increases. Now, we also showed that the microglia of here, it's astrocytes, secrete inflammatory cytokines, and that the secretion is blocked by antibodies against one of the pattern recognition receptors, and toll-like receptor 4. So therefore, we think that we have demonstrated that cytochrome C is a mitochondrial damp. Now, in vivo evidence still needs to be collected, but at least in vitro evidence demonstrates that cytochrome C can be released by neurons and glia. It interacts with a damp, and that cytochrome C induces pro-inflammatory response of microglia and astrocytes. So therefore, it is possible that damage-associated molecular patterns, this group of proteins, is another kind of set of molecules which are released by damaged cells in central nervous system under sterile conditions. So Alzheimer's disease is a neuroinflammation where, as far as most of us researchers in Alzheimer's field believe, there is no pathogen. Now, there are theories that Alzheimer's disease can be caused by pathogens, and several candidates have been suggested, but I think that the most accepted scenario is that there has to be some endogenous molecules which are driving this neuroimmune activation in Alzheimer's disease. And we believe that damage-associated molecular patterns could be contributing to this. Now, if to look at peripheral tissues, high mobility group box 1 is like this prototypical DAMP, which is a transcription factor, and it's located inside nucleus. So the next molecule which we kind of described as possible DAMP in the central nervous system is mitochondrial transcription factor, TFAT. So again, similar to cardiolipine, we demonstrated that this molecule, if it is released into extracellular space from mitochondria, that it can activate microglial adverse activation and induce toxicity. And our most recent project concerns another mitochondrial molecule called cardiolipine. So cardiolipine is this strange phospholipid, because instead of having these 2 fatty tails, it actually has 4 fatty tails. So also cardiolipine, it resides in mitochondrial membrane. It also assists with stabilizing these complexes, which generate ATP, and it is also thought to contribute in a process of release of cytochrome C when apoptosis is activated. So what we have done now, not published yet, is to show that cardiolipine is released by damaged microglia. And we also discovered that there are a lot of different forms of more than 20 different forms of cardiolipine released by microglia. And they differ in the structure of these tails, right? So here, some previous studies have shown that there is difference in cardiolipine structure between bow and heart and rat liver, for example. And we are hopefully soon going to be publishing that microglia release cardiolipine, and that there are also like a number of different isoforms of cardiolipine. Now, when we attempted to demonstrate the cardiolipine is a damp, surprisingly, we actually obtained opposite result. So here you can see that if to expose neuronal cells to microglia supernatant, a microglia which has been activated by interferon LPS, they induce neuronal death and reduces viability, right? So once we start adding cardiolipine, the viability of neurons is restored. So cardiolipine appears not to be toxic, but quite opposite. It appears to be protecting neurons and reducing microglial toxicity. Furthermore, we realized that cardiolipine actually upregulates microglial phagocytosis. So here, these are primary murine microglia. So in absence of cardiolipine, they don't take up too much of latex beads, but if to add cardiolipine, they become more phagocytic, and this can be blocked by antibodies against TLR. So here you can see the quantitation of the data that cardiolipine upregulates phagocytosis, and this upregulation is annihilated by addition of anti-toll-like receptor 4 antibodies. The same is true for astrocytes, so that cardiolipine upregulates phagocytosis. So therefore, for cardiolipine, we can say that yes, it's released by damaged and stimulated cells. It interacts with, again, pattern recognition receptor, but it reduces glial neurotoxicity, and I didn't show this, but also reduces secretion of pro-inflammatory cytokines. Now, upregulation of glial phagocytosis, our current thinking is that it is also a good thing, because like a theory, one of the kind of thoughts about Alzheimer's disease is that perhaps this disease is caused by defective phagocytosis. So meaning that accumulation of amyloid plaques is because microglia, for example, is unable to remove the precursors of amyloid plaques. So therefore, upregulated glial phagocytosis is actually a good thing. So to summarize all this, what appears to be happening is that molecules such as, or some DAMPs, such as cytochrome C, TFAB, and there are other non-mitochondrial DAMPs which have been shown by other researchers to induce this adverse microglial activation, could be something which drives amyloid pathology and Alzheimer's disease. But there are probably some other DAMP-like molecules, such as cardiolipine, which inhibits microglial activation and promotes CNS homeostasis, thus basically reducing probability and slowing Alzheimer's pathogenesis. Now, this term was proposed by Shields and colleagues in 2011, and we have now also written supporting this concept that there are most likely a set of molecules which are also endogenous, which kind of act similar to DAMPs, but they have opposite inflammation resolving effects. And we are calling these molecules resolution-associated molecular patterns. So as a result of all this work, I think we can kind of conclude that there are a number of molecules, and obviously this isn't a full list, and we are going to be trying to look for others, but there are molecules which can differentially regulate glial immune responses. And the outcomes of this research, the practical outcomes could be that inhibiting some of these molecules, such as cytochrome C and TFAN, or removing them or interfering with the receptors they interact with, could be beneficial of Alzheimer's disease, while other molecules, such as cardiolipine, and some researchers have already created liposomes containing cardiolipine as possible therapeutic agents, that these kind of approaches also can be used to try and slow down Alzheimer's disease, just because cardiolipine appears to be having protective functions by inhibiting glial activation. So with this, I think I'm pretty much run out of time, and luckily my slides are also finished. I just want to kind of have similar, like a reflection in a mirror Zoom type picture of our current lab members. And all what's left, I just like to thank you for listening to my presentation. And I would be happy to answer any questions in a chat or other means what you use to communicate. Thank you.
Thank you, sir, for this amazing presentation. Actually, I'm going to ask the questions that we collected.
Yeah.
First question is that, can autography induction have a therapeutic effect on the progression of Alzheimer's disease? If yes, in which phase it can prevent the progression of the disease?
So can you please repeat the first part of your question?
Can autography induction have a therapeutic effect on the progression of Alzheimer's disease?
Autophagy induction, is that correct?
Autophagy induction, yes.
Yeah, so I think so, right? So the defects in autophagy, and I've seen grant proposals. We haven't ourselves thought about or kind of looked into this, but autophagy defect as a cause of Alzheimer's disease is one of the current actively, I would say, research hypotheses, not by us, but I know several labs are either doing that or planning to research that. So yes, the answer is yes, absolutely.
So the second question is actually a question. Can cardiolipin production be increased by viral vectors? Or are you doing any research about it?
Yeah, so I'm sure that viral vectors could be used to upregulate enzymes, yeah, so which would participate in cardiolipin synthesis. The synthetic pathways, cardiolipin is probably quite complex due to these like complex chains and so forth. So the short answer is probably yes. But right now, I'm quite excited about this liposome approach because liposomes, it's almost like a kind of also it falls into the area of nanobiology, nanotechnology, right? So one kind of important thing when thinking about treating Alzheimer's disease is to consider the blood-brain barrier, right? So it is hard to get therapeutic agents across the blood-brain barrier. And the liposomes is one way how to achieve this. And if we can get cardiolipin across the blood-brain barrier inside liposomes or even like something like exosomes, right? So I think that is a very, very cool and interesting approach.
All right, thank you, sir. We don't have more questions left. So if you are kind to join us in the breakout room, we have to accept. All right.
Okay.
Thank you so much. Can you join the breakout room or because you have to join?
Dear participants, our Congress now has ended. We hope that this Congress has been fruitful and beneficial for you all. We have had a lot of fun hosting it, and we believe that it has been very informative. We will inform you about the participation certificates, and we hope to have them delivered to you as soon as possible. Again, thank you for your participation.